Hello, good afternoon or evening or morning, wherever you might be joining us from. And welcome to Divestream 904 of the remotely operated vehicle SuBastian. We're back in the water for another dive of the Designing the Future 3 expedition. And currently we are passing, we're almost at 350 meters. And just like yesterday, we're hoping to make it down to 1,200 meters. But this dive will be staying in the midwater, so we'll get down to about 1,200 meters, and then we'll slowly make our way back up. And I believe this dive is shooting for another 12-hour length or so. So sit on back and enjoy, and I'll be bringing in the mission control audio.
This feels better. I like the one in science.
Let's see if we can pick
Yep.
up things.
The one in science feels better, if we're okay with that with everybody else.
Let's give it a try, and if we get the impression we're not picking things out, we can
Yeah.
switch it back.
So, yeah, we're just jumping the audio like this. Yeah, okay. Yeah. All right, y'all. Enjoy.
We're good?
You're good. I'll be back in a little bit. I gotta go clip.
Oh, you're good.
Past you guys.
This is not mine. If we were to increase the shutter speed. Hi, everybody. This is Heather Judkins, University of South Florida, St. Petersburg. I am one of the science team. I am currently hanging out while Karen Osborne is working on some logistics. She's our chief scientist for this expedition. We are headed down today to 1,200 meters again, as we did yesterday. And the ROV is in the water, obviously, and we're at about 380 meters at this point. We are looking for biodiversity. We are going to slowly descend and stop for interesting organisms we'd like to focus in on as targets as we go down to 1,200 meters and on the way back up. We should be here for about 12 hours for this dive today. So stay tuned and thanks for joining us. Get a little dark.
Do you want to just walkaroo? I fixed everything.
Ooh.
There you go. I fixed the computer. There's a do login there. So
Is that okay? I don't care what I said here.
you did such a nice intro. I was like,
There you go. Stand by for you.
all right. I don't think so. Thank you.
It's really great.
Yeah, okay.
Take a look, whatever you want to do.
All right.
There's been discussion.
All right. Hey, everybody. Switched over to Karen Osborne. I am an invertebrate evolutionary biologist at Smithsonian National Museum of Natural History. Here, taken an awesome team of expert scientists and engineers out to sea to explore the midwater off of Brazil. We are about 360 miles offshore, just below the bump out, and it is very oligotrophic here, which means not a lot of productivity up in the surface water. We want to see what's in the water column below that. Been seeing a lot of really interesting animals. Been getting some interesting things in our toes. Our objective out here is to build the reference libraries for midwater animals. Most midwater animals have not been sequenced. So even if we have described species, if you want to do eDNA on metabarcoding, you want to just take a water sample or a fish gut and see what's around, you cannot do that because the sequences have to be available for the animals you're looking for. And most of the animals in the midwater, there is very few sequences available for them. So we're out here to change that. We also have some really interesting new ways to look at animals, which makes it possible when we find particularly interesting ones that we want to describe, we can do that. Sometimes just based on the images that we see with our new technology. And as we use those, we will explain what they are and what the objectives of each of the different imaging systems is. So we are set running a bunch. Is that correct? Or are you guys still testing things being settled?
Playing with the camera. So
Okay.
just try to
Alright.
get the better
Yeah.
picture.
Yeah.
Just to bring.
It's a bit dark. Like I would want to see more particles out there, personally.
Usually when we take it out,
Or maybe there just aren't as much out there.
So like,
yeah, it does go on the clouds. Well, we also don't have uppers on. We just have lids right
you
now.
wanna try the opus?
Yeah.
Right?
It's better when we're going
Ooh.
down. What? Swordfish.
That's on the back.
Yeah,
In the back.
Now it's up top.
So a swordfish just moseyed past the back of the ROV. He was just
recording that feed? Probably not.
No,
So cool.
Fishing.
Very
that
slowly
is pretty
checking us
cool
out.
to see a swordfish just moseying past the back of our tether. We are at 470 meters, and he was just chilling. Can we look at these jellies?
Yeah. Wanna stop on us?
It's a bit like, yeah, please.
Stop it.
Looks like a genera, maybe?
There's something down, see?
That is a fish.
Something coming
Oh,
down.
can you see the fish below?
There's like, I think
See
it's
Or a
like
big
huge
long
siphonophore.
scythe. Yeah. We are
Ooh, there's a big size.
70. We're in a thick layer here. Actually, according to the EK80, that is a nice long scythe. Decide where the head is, one end or the other. Wow, there it is.
What's that?
Wraps around. Let's follow to the end, over to the right.
Yes, this is going to be.
We want to follow it up and see what's on the other end.
Oh, that's good.
Yeah, that's okay. We'll follow up to the right, see if we can find the top, so we can get an idea. And then if we don't find a head here, maybe we'll go over and try to look at the other end. This is the head end. You see it just down there, yeah. Can we zoom in on the head a bit? It's quite long.
It's got an awfully long neck to some.
Is that Bartmania? Bartmania? Yeah, what is it?
Is dropping swimming bells. Just looking through the curtain of tentacles. It's actually really cool.
Zoom in some more.
So what is this thing called?
Siphonophore.
Yeah, working on that. Is it arena? Just really spread out? That would be my guess, but I am not a siphonophore expert.
See, that's what happens. I call it apolemia. It doesn't have the little.
It doesn't have the tentacles to
No,
be
so it's not
apolemia.
I am going to the deep sea guide.
Well, Dougal sure picked a bad time to be gone. You got tentacles all smeared all over the camera.
So, you guys ready for the verdict as per
Where to go?
no?
Alright, so.
Arena.
Yes. Look down with your camera.
My guess is arena.
We could. I just wish Dougal was here to let us know.
It is a very long swimming part.
Yeah, it's like, no.
Oh, I'm not sure.
Here, let me zoom out.
You guys are not going to want to try to scan this, are you?
We probably would like to.
Okay.
Let's just do it.
All right. Let's try to do this.
Why am I not?
The tail is broken off, so it's really this contracted section with the head that they want.
Here we go.
Scan it.
Yeah. Yeah, I mean, with the laser one. Just this bit. Contracted part from the swimming bells.
Back off and just call it a price again.
So apolemia has little tentacles in between the
Okay, it doesn't deal with it at that level.
swimming bells, and this one doesn't have those.
Yeah.
Now, we got the lids right now.
So if you can, let it settle a bit, and then just really slowly push it through. So put it out in front and let it settle. And it looks like it has a twist in its.
Maybe a Mars. I put in Apollonia first of all because it's so small.
It's got some pretty hefty bracts back there, like an arena.
You're gonna suck them up?
We're gonna
Done the scanning?
scan it. Maybe we resist sucking up big sites.
Once I'm logging in.
This is less robust than the one that.
So if it's moving like that as it goes through the scan, it's super hard to reconstruct. So if you can like just let it sit out and settle a bit and then push it through, that would
I think it's a Mars, but it's
probably
not
make it
so-
easier for them to.
It works a lot pretty long, siphonophores.
Oh, okay.
Place of stone. Nectar stone.
Siphonophores, that's what you use.
Yes.
Oh yeah, it's very long, isn't it?
It was really long. We checked, I think we got lucky and we checked. We could have gone February.
The business end.
So I wouldn't record that as a DPI.
I don't know.
Already.
Oh, you did already. Okay.
got a note.
Yeah. No, that's fine. We just I don't think they got any usable data from it.
Alright.
It was too rambunctious. Lots of tentacles on the bill.
Yeah, I've seen this before somewhere. It's either a Mars or it's this new family we've been calling, depending who it is. I've been calling a Jezebel. I think Steve's been calling it a Harbisonian.
The Harbisonian?
That's what Steve's been calling it.
You guys see the, okay, so you don't have the different viewpoints here. Jimbo, can I get,
Or it
Well,
could be
Named
Mars.
after Herbert Lynch or Harbison?
Harbison.
wait, where do you have it?
Thank you.
Right here?
The second one from the left?
No, yes.
But I already have been published as the
And then
Jezebel.
that one, we had it to where it was, yeah. So then the other one would be the citation for, citation,
Far left?
far left.
HD4, yeah.
Right, so.
Yeah. Those perspectives. There we go. Yeah, and then up to you, but I find it helpful to say the perspective. And then one that's in the right spot.
Share times. I don't really have enough time charging yesterday. There's something wrong with the charging circuit that end. We only have 2.8 hours worth of data. The first step in India was there. So it didn't matter that there was a lot done. It'd be nicer to have it clean for when you would use it.
Yeah.
Should put a power jack on it.
Yeah, I'll plan to do the next one. This was a prototype that had to be re-engineered at the last minute.
Just the mids.
Wait, every.
And there was also not a lot of info on what was nature. And there was also not a lot of info on what was hydrocarbon.
Yeah, no, it's fine. So then you would go, you
This one? It's the same one. No. It's just contracted. That we don't know. But when they talk about siphonophores, when you first come across, yeah.
By the time this is sort of already
Yeah, it could have been.
So what is it, the contraction?
The stem has muscle on it. It rolls because it goes.
Let's get a little bit of that. So
Mm.
I have no idea.
Mm. Okay.
Just my, but I hope we'll still be able to see that.
Just stuff that move in the water.
Some of the tensilla from the tentacles seem to have stuck to the camera.
Oh, it's not dangling in the water. It's on our.
It's on our
It's
camera.
on the screen.
Yeah.
I see.
No, that's likewise. Turning.
Was that a good one? Was that on the screen?
Turn off now.
Turn on the months.
Turn off. Oh. I don't know. There's kind of lose them.
Yes.
Turn the mids back on.
Some people
Once I located there.
We'll just, yeah.
muscle coordination and stuff as well. But
I'm just gonna keep the muds on. That's what we'll toggle
yeah,
between
very
the
hard time
muds.
transferring.
For real depth as well. Look at that one. Not right now.
You can see it happening not as like a coordinated whole, but you can see some bits contract more and the other ones not that contracted, and then these ones start to contract and these ones start to contract.
Today's better than yesterday because I remember when we let up on a joystick, the vehicle went down or up and back up. There you go.
Now it's
I don't know, so yesterday we were a little buoyant. Day before it was hot meat today. Wasn't feeling.
Mm. Yeah, there was a high freedom.
We were gonna take it off.
No, you can leave the stomach, the tentacles, and people
Hopefully
a day or two be in there. But they lack reproductive services. Keep them afloat, etc.
Yeah, this is it, making her breathe.
Take a look at Casey Dunn from Yale. Not very much. Not very much else.
Ooh.
Is there any neural tissue that's specific to this? Like, so if I was to say beyond if there's muscles in this tissue,
Look up Dunn and Nanomia and development.
So the instrument that we're using now is the DPIB. It has a red laser sheet, and we're using the main camera. It also has another camera that records the cross-section of that laser sheet. The objective here is to line the animal up outside the laser sheet and pass it steadily and slowly through the laser sheet. Essentially what we're doing is doing a micro CT scan of the animals, or a CT scan of the, oh, it just dropped its head. Oh no, didn't, just went through it. Wow, that was weird looking.
That's such a long.
It's got such a long, it's so super long. So what happens as the animal passes through the laser sheet is we get a slice of the structure, which gives us three-dimensional morphology of these animals. To reconstruct those, we need them to stay essentially straight through so that we can tell which piece goes with which piece next, and they don't shift as you scan through them. Typically, if any of you have had a CT scan, you know that you have to hold really still in that machine, and that it moves around you to take the images. In this case, we're moving, the animal's moving, the water's moving, and we're trying to get everything completely still in that laser sheet to collect the data. So it's incredibly challenging flying. You see a few tentacles stuck to our windscreen. Those are tentacles from this animal. It had its tail spread really far out in a beautiful cascade of stinging tentacles, and when we peeked through that veil, we got a bunch of tentacles stuck to the hood that surrounds our main camera. So that's what the little orange bits are that you're seeing flying like air flags or something across our screen.
itself contracts, and that coordinates.
Nice krill scale now.
Does it need to receive a signal first?
That's what I'm saying.
Yeah.
I think it's calcium or some other, because
It's interesting to look at the tail and the way when it contracts the tail, it forms a spiral, how everything folds together in this beautiful spiral.
Gastrozoa is
Once they have it here.
Yeah.
the muscle that you really care about is the tensilla. They twitch the tip of the tensilla, and then you need to pull that up to the mouth of the gastrozoid. So that's important. The muscles along the gastrozoid and the pulpon, which is like the devout gastrozoid, which is for tasting and feeling and stuff. They're the two most important. And then there's fluid whales.
but then the why do this?
They need to be able to share energy, which they do via doesn't have to be close.
So when we turn the lights off, when the animal gets close to the laser sheet, it reduces the other imaging noise in the picture and allows them to have a much cleaner scan. So what they want to do is get the animal really close and to put it through the laser sheet with no other lights on.
Yeah, these don't manipulate anything.
Ready?
The common people who we haven't actually done anything really in situ for over 100 years, like of the structure of the histology, etc. But common wisdom is that inside of the stem is like a tube, and they share their nutrients.
Some of them are very small, right?
Yeah, some of them.
You could get like, yeah, the small ones. Obviously, this is hard.
Expansion of the colony to kind of provide
The way they stay stretched out and you see them in situ makes me think sillier.
Super, what did you guys decide to call them?
Maris.
Maris.
M-A-R-R-U-S.
Yeah. Any species name?
Undescribed.
Okay.
No. Mars, you have is red.
Yours
Google
is
Lindsay
Maris cornelianus.
Suggests that this is Mars, an undescribed species of Mars. M-A-R-R-U-S.
Is it like a,
It's in all over the place actually.
The night field
Types,
camera
that's it.
and the 4K original data had enough resolution to see the lines of nematocysts on the graphs, which is what you need at present to tell
I
you.
read it, and then
UTC. Can you get UTC off of the readout
was
up there?
that last night? No.
Oh, it would be a ISOS.
Yeah,
Siphonophore.
yeah, I was trying that out.
Oh,
That's all what you need. It's like,
there.
Yeah.
and I was like, I can't find
And then taxa other.
That's
Siphonophore Isonic.
awesome. Such a specialization.
I think it's down. They're arranged in
Especially
it's not really alphabetical order there, are they? That's helpful.
That's the struggle.
We did do that, so we can't really complain. Siphonophore, there we go. Isonic.
Dougal, do you want to collect this?
Unfortunately for my sample processing afterwards, but fortunately for my shadow cam.
Are we okay? Oh, so you want to do sections here?
Yes,
Yeah.
I want the zooids.
Whoa.
But then when it comes, before we fix it,
And we have a
So we can
scene
do
over
live.
here with,
I wanna do live stuff on this. Because I have followed labels also, and then just watching stuff.
Up.
Is actually,
You have to fix it.
yeah, but that's post, but that would be small pieces of tissue.
Just to see the
It
structure.
would be nice to actually
It'll look great.
see the
It'll
attractions.
look awesome. Yeah, good work.
So something that would be interesting if you're into histology, etc.
That's all we do.
So the Physonics somewhere in their evolution. Apolemia is the old smallest group, and there's a hell of a lot of different kinds of zooids, but they're jumbled up without very much.
Passing the animal through a laser sheet actually provides three-dimensional information by giving us a slice through the animal. If you for transparent animals, we can get a slice, and all of those slices together can be put together to produce a 3D morphology of the animals. We can understand how all bits and pieces fit together. When we suck it up, like we are about to attempt to do, all of the pieces and parts of the colony will mostly come apart, and you won't be able to see how they were connected before. But doing the scan of the animal through the laser sheet allows us to reconstruct.
And then the other one, opposite side of the stem.
Did it go from a right hand?
It goes from, well, it just goes from, if you had it straightened out, then it would be like a line or a muscular or another attachment. So and the body was
That one,
Yeah, sorry. Still trying to find a time code.
so that would be interesting.
What bucket are we on?
Don't remember which one was the skip.
Twisting. Yeah.
I mean, it's
So there were three different groups of fossils for this.
That's impossible.
The group that's dorsal ventral.
That's right.
That's dorsal.
The resolution on the laser scans is about 1.5 millimeters.
Where it's the only one
So it gives us the shapes of the individual pieces of the colony. Unfortunately, not enough detail to get down to cellular level or sometimes even tissue level.
and the necrophores are kind of
That's really- Yeah, do you see the head? The more transparent part?
Number 5 already. Do we want to index?
Right, okay, you want me, but
Yeah. So section sampler 5 has that unidentified Bisonex.
did you catch it?
It's in there.
We're now in, we're now on number 4.
Yes, please.
What can you down?
Yep. We're headed to 1200 today. Actually, when we're going down, I think we're lower. That's
Notice,
kind of
yeah,
what's
we
ahead
didn't
of
have
us.
a solar eclipse today.
That's kind of what we ended up on last yesterday, unless it
That's
gives us
really
too much
funny, it showed
reflection
up
from
in
all
all of
these
them.
equipment in view.
Alright, we'll cut the numbers.
The Burrowie?
It's pretty good. We just
have months and numbers on.
okay. Can we move the camera to not see the equipment any more than we have to?
I'm missing
Do you
the.
have a time code somewhere?
I don't have the sheet.
What's up?
The sheet to keep notes. Oh, you have it?
Yeah,
Oh, okay.
I'm doing it. I just, can we also get some way to see time code over here? If we see, is there, right, we had it on the bottom of the screen yesterday. Today we don't have it.
There's the
Simple.
undescribed Mars species. The one you get and the one I get a lot is Mars.
Any over here?
There's a couple like this
Over
one. This
where?
is what I call
There is a flat line.
white but there are a couple that are this.
That's what we have.
Oh, you see the
themselves.
We don't need to have the EK here because we have it right here. So you could put it right there.
The other Mars.
2:30, you wanna stop now?
Mars needs to be split.
All right. Thank you.
ROV Bridge.
And one of the animals we bought yesterday is presently in Maris.
Go ahead, Rich.
Have its own family.
Hello, you're going pretty quick. Do you want me to keep up with you?
Yes, please, you can follow some.
Yeah, I'll get another engine on. Cheers.
Yeah. I'm going back to the center, so that particular.
Okay.
Either the site, the nectipores or the nectosome matches, but the siphonophores super short, or vice versa.
If we're able to find a fairly large medusa that would be big enough to abrade off all the pieces of siphonophore stuck on the inside of the hose and in the tank as it goes through, it would be good.
Oh, so I forgot to.
It's probably also.
We did do that. Just some other plan. You could still see there's all the tentacles all over the
Yeah, I don't know on the screen of the main camera.
So if we're gonna do down and forward, that'll give us the best chance to see some, unless you're running back with a boom or something. Yeah, sorry. Yeah,
So down and
down and forward, great, thanks. Is there much current to it?
That's the
That's not more, but these are very
And I'd like to get some medusa
That's not bad.
while
It's
the telecam's still running. Something big and red? Coming up middle to right. Oh no, it's a piece of, it's out of focus tentilla from the siphonophore.
Yeah.
Sorry.
There's several of them, just so you know.
I saw the one in the middle. I hadn't realized the top right one was one as well. Oh, tracky. Oik. Oh,
Stop.
whoa. Is that a, what is that?
What is that?
Oh,
That's
Tetraphalia.
Beautiful.
Will be Tetraphalia.
Okay, so.
Spelling, please?
It's Mertensidae, M-E-R-T.
Look at that. Oh.
Oh, this will be nice for a scan.
Nick.
Mertensidae. It's a sediped ctenophore. Ctenophore,
You can keep that at a
sedipede.
DPIB view. Very-
Right.
It looks very nice.
Yeah.
It does.
This one's ripe for description. 3D
Okay,
data,
so
the
we're
canal
gonna-
structure. I've done a description in
Yeah,
Japanese.
if you get it a little
Science,
closer, then it'll come really into focus on the.
What's the rest of that spelling?
Mertensidae,
No, your camera
M-E-R-T-E-N.
doesn't look
SIIDAE.
But you said a different name first.
Yeah, that's
Tetra-
grey literature.
Tetraphalia?
Tetraphalia, yeah, grey literature. It's not published.
good.
Okay.
You put it in. I know you guys are focused on the science, but that camera, the grain gain is so bad. Put it on the DPIB.
It's eating something.
Yeah.
Okay. You could put in the, well, yeah.
Oh, that's the.
This is the more specific name, but it's not published, so I actually shouldn't use it.
So it's got to be called Mertensidae.
Look at that. Look at that.
Right.
So nice.
Right.
The tentacles on
And
this
you're
guy are
recording that.
beautiful. So this is Mertensid sidipid. It is an undescribed species. So you're done with this one, Dougal. You don't need it. Is what you're saying.
No, we need it.
You said it's all described. You've
No, it's
got all
not.
the canals and everything.
It's
Right,
ripe,
so-
ripe for.
Oh.
Uh-oh.
It's ripe for description. If we get, oh, we lost the tentacle.
I've got the one caught in the camera, lights.
Alright, so we would like to do a DPIB scan on this, please.
Sure.
I'm still recording.
Just
If you can get
hopefully you can
look
see
at
them
that.
from pretty
It's
far
so
away and let
nice.
them just get
No
set
doubt.
up out there.
Siphonophores are very good targets for genomics.
We know.
Yeah. This
We're
is
there.
Okay, thank you.
this I really want, and a suction sampler if possible, so we get shadow
Suction
cameras.
sampler? Seriously? It's gonna beat it
Also,
up.
that jelly, we have 400 megabase genome coming.
Oh, great.
Looking better.
That's good.
Yeah.
I think its tentacle is stuck to the
Still got
TPIB
tentacle
laser.
attached to it? Is it
Oh,
trying to
yeah.
pull it in?
So it's gonna be a weird
Trying to pull in the
scan.
camera.
I mean, it'll be tethered, so.
It's caught on the camera.
It's
Almost loose.
inside. It's inside.
Stronger than it looks.
That must be the tentacle.
Turn 90
Oh, no.
degrees, you bloody xenophobe. Oh, it's big.
Oh.
Back up.
Yeah, like. I think it's stuck to.
We kind of got it tethered to the vehicle.
Yeah, so the goal of scanning these animals is to see their three-dimensional structure. Many of these animals are too delicate to collect and image in the lab. And so we can image
Yeah,
them
you're
down
definitely gonna wanna
here.
put it to the
We're
right.
essentially doing a CT scan of these animals. By shining a laser through a transparent animal, you get slices of the entire animal. Put those slices together in 3D software, and you have the
This
3D
thing must be like
anatomy
this
of the
size.
animal. We get about 1.5 millimeters resolution.
Yeah.
So it gives
true.
you an idea of the whole animal, but fine detail.
It'd be too big to
But
fit in
enough.
our laboratory-based
We can
imaging
extract
systems too.
Yeah, the tentacle is stuck on the instrument, so it's tethered to the instrument where we're trying to scan it. Pilot has to try to fly this animal through the laser sheet
How good can you see the
slowly
actual
and
canal
steadily
in these
without
3D
animal
scans?
going up and down or side to side.
Normally, we don't normally do xenophores because the telomeres that we looked at just scan all the way. But this section looks relatively good.
I think a lot of the terms that come up with like getting cameras.
So big. The one on the right, yeah.
Sort of on.
What was on the inside?
Yeah, little baby, little bit. Oh, that was a good one. Oh yeah, that was nice.
Okay.
We have light field as well.
We can.
Yeah, you have the light field. No. Okay. Can we do the light field scan? You rate it? Yeah.
Not sure this is the. Not sure.
This is a good one to get the entire set.
So for the entirety of this expedition, we will be doing Midwater work, which is what you're seeing today.
It's gonna be one of the most
All
difficult
the
tentacles
amazing
around.
animals that you know very little about.
Probably yours
Many of
as
them are
well
very
as mine.
delicate, many of them are gelatinous. They live in the deep open ocean. It's a really, truly three-dimensional habitat that they live in. There's no place to hide, nothing to dive behind or sit on. They spend all their time swimming out in plain sight. So there's a lot of really interesting adaptations these animals have made to be able to survive like this in this habitat. And we're interested in all of those and the animals that are here. So we'll be doing midwater dives for the next week or so. The animals like this.
Fairly good photos from one we put in plankton that once and some other video and stuff to flesh out the construction.
Down.
I think they change a little bit as they grow. The size of the keels especially.
Yeah. Do it. Get back up. Sarah can say that. Oh, wow. I would say it if I was.
Hello.
Yeah.
We're headed down to 1,200 meters today. Focusing most of our work in the mesopelagic, which is about 200 to about 1,000. We're going to dip below that to see what else is there. We got some instruments on board, but can't go deeper than 1,500, so we're going to bottom out at 1,200. The water depth here is about 5,000 meters, if I remember correctly. So we're well up above the bottom. Everything here is spending its entire life up, well away from the bottom, living off of rain, snow, and all the other organisms that are here. Oh, look at that little jelly over there to the left. I think it's in the city. Try not to oh.
It's a little hot, but that was straight.
Google said it might be nice to try it on this.
If you can try the light field camera as well, so we have a full set of everything for the project.
I think it likely will be difficult to draw anything but like the team knows.
One thing I was gonna propose to you was maybe finding a good spot for Iris to look for it maybe. So that we don't have to like beat it back and forth.
We have a pretty low chance of being able to count the cone plates on the Pyrosome in any of these scans.
Sorry, what's that?
We would have a pretty low chance of counting the number of cone plates in each of the cone rows in any of these scans. That'd be a fair assessment.
I think we have been able to in the past, where we're limited sometimes is when they start to converge, and they get brighter and start like to illuminate each other. That's where sometimes it gets a bit blurry.
Alright.
Do you need
But you can
integer
count.
counts, or do you need like you'd be off by 4?
As long as I know what the error is, I guess it's okay if we have a whole set of everything. Like respective lengths of the cone plates versus where the tentacle opening is and stuff like that, I think you could probably do. But I always, because you don't know how many species there are in this genus, it's easy to describe it, but there could be different species that you need to tell apart. So I try and get the width of the cone rows versus the distance between them, and total number of cone, like what cone plate number does the tentacle sheath opening actually occur, or does that was fine scale data that I think is
So this
So,
one,
and the iris.
Yeah, so
Small
what I
relative to the
wanted, I was curious if we'd be able to try this today when
In
we
this case,
pick a good spot for Iris. Keep it to port of DKB and we leave it there.
Down left,
but
down
is not
left, down
something
left.
we could maybe glean from the close-up 4K science cameras? So we can measure relative distances and distraction.
You guys, I would rather, I'm worried, we have to get Iris in the right position and I'd rather us not risk losing the animal.
okay.
So.
Can we do some tight 4K with the main science camera shots of it again then, maybe?
Unless you're comfortable at all, I just want us to be for adjusting Iris.
Alright, let's see, I'm gonna bring it down there. It's gonna open, where do you want the iris? Right where it is?
I think we might need to be a little bit further to port from DKB because we're gonna open that light ray.
Okay. Are you good if I adjust a little?
Yeah, go for it.
Alright, coming up.
Give you that camera.
Just right about here, you think?
You can move the camera as well, focus on the
Yeah.
subject.
Yeah, yeah. Right there? I think that looks pretty good. And we just roll it straight along.
I can adjust it after.
That's all I guess it's.
Alright, let's turn on. Yes, can you turn on the left hand? For Iris?
Leave her alone.
Wait, shall we?
Yeah, we got it. I think we need a lot less lightness.
Bridge, Alvin.
RV bridge?
Yeah, could you come closer to us, please?
Yeah, it's getting up.
Thank you.
I think, what does that set it down?
5.
Yeah, we feel like it's to 50. And you might want to change this OC. That's the refocus view, so you might want to do the
We move it a little bit away, like forward and out, and then bring it a little bit to the center. Don't disappear too fast.
I think it's still
get a
out there in
visual
front
of
of
it
her.
first and keep it out front.
There
Yeah,
you go.
I'm gonna bring it to the center, so you.
Yep, alright, I'm gonna extend out. Extending out. Kind of like up here. It's
really similar.
Skewed a little bit.
Yeah, what are you thinking?
Yeah. So I need to bring this into the center, right? What's
No.
that?
One of
Of the
This.
animal to where the like do you want it? It's at a bit of an angle though. Do you want it more straight on? Works for you guys. It's good if it works for tomorrow. I find it works.
That's fine. Just so soft. That blue makes it at least fairly easy to see.
Jimbo, I need the iris speed.
Which
If
one?
you could
Second one?
put it, yeah, so it's 12, 3, and the second one, please. See where all of the lights are going?
Yeah. Oh, you want it there?
Yeah, so it's about like half a meter. Instead of
That's the, where is it?
No, it's down just behind the back
Where
virus.
the manipulator
Oh,
hold the
there it
things.
is.
Yeah, there it is.
Do you need the
Coming
pointer?
back up.
There's a pointer. They want it straightened up.
Yeah, sorry, about the.
Sorry.
Pencil works, okay.
Like, alright, so
There you
it's kind of straightened there a bit. Alright, line
There
it.
we go. Down.
Actually, this one doesn't.
There's a shortcut for the record button.
Ooh,
Shift
right.
Alt. Hold on, let me press 3 buttons.
Yeah.
That direction, buddy.
Yeah, ditch the very last cruise. I don't know. Do you want to? Careful. I mean, they're giving it to themselves. Never our camera.
It's big.
Yeah, it fills the frame. When it does. It's right on the edge.
Yeah, it has to be because seems like we're going to be right in the pool of that red
Yeah, I
light.
can see it.
There's a ton of flow.
Standing right here. Didn't trigger that.
Down the door. Scoot back out.
Did earlier too. I wasn't fast enough, but I already got it. They are ready for us to preserve the seducer. What do we want?
Did you take some for sequencing?
Yeah, as long as you get some
Yeah, I
tissue.
I was be given to me to put it in full.
As long as you get some tissue. it's ready to go.
Do you want tissue from the smaller one as well? Yeah. So you'll have to cut something off there and you cut off on the super.
Kind of hard to figure out what to cut off of.
Something that includes some muscle bands, I guess. The ring muscle has got a sequence, I think.
Too close or too far? It was either too close or too far, but I can. So it wasn't resolving as much depth.
Perspectives out there.
Some there. Back off a little bit, I think.
So, how am I do this before?
We haven't quite gotten a hold of the body.
It's too close.
Yeah, I think we're just
As long as we get the, as long as we get the half, the tentacles are coming out. Ah, there we go. That's probably enough.
Or you put it around the center.
Well, I think Dougal, you're happy with that last?
That's enough to tell the internal canal branch, and I think.
Okay.
Okay.
Over to you. I'm gonna follow the camera.
What's the genus on the big one?
On the big one?
The big one.
So this is a undescribed species of mertensid cytopid we've been looking at. It's a beautiful blue color, very sticky tentacles. Peter Lindsay, one of the scientists on board, is in the process of describing this species. It's quite large for a centipede. The body shape is fairly unique to these animals. And the four lobes that go out in different directions.
If we're going to sample
It's a
now,
mertensid, undescribed mertensid.
yeah. We were, yeah.
What's that having back camera should be?
Did it regrow its tentacle?
No.
We might not have cut the entire thing off before. I wonder if
It's
some of
indeed
them
a
have double
tetrapod.
tentacle bulbs, like a
Yes.
tentacle bulb pair.
Not in a
So
blue.
maybe that's a. So maybe that's a, like,
properly
if
described.
one gets cut off, it still allows you to catch prey, kind of an evolutionary trait. I hadn't really thought about that before.
Pretty sure there's a tentacle from this T4 still on my DVR.
Yeah.
It's not soaking in.
I've seen the bubbles. Yeah. It's big. It's just gonna be gently moving forward.
Not enough pump strength.
Oh, there we go.
Okay, time to pop this off.
And then just drive forward slowly.
I know.
Sorry, I guess.
No, you're.
That was
What kind of
great.
is that?
All that was happening.
Number 4.
I'm not going in. Those are such-
All right.
Turn them on.
Can we turn
See,
the...
it's
no. Something's going on.
Alright, something just came in.
Oh, there it is,
Here
okay.
All right.
Hey.
Suction off,
Something's off.
Rotate.
and it's just sitting there.
Just the right size, yeah.
Alright, index. Oh, it's beautiful.
Beautiful.
All right.
And it cleaned the inside of the shadow cam tank on its way through.
Nice catch. Nice catch.
You did great.
Awesome.
Like,
The shoulders, the back.
All the tension can release.
Yeehaw.
All right. Let's
Right.
set our camera back
Alright,
up.
so
If
if it is catching us, I'm gonna turn a little bit. Start heading out and down.
Okay. And can we set up our science cam?
Oh, yeah.
All the equipment out of it. Look at animals. Probably turn it down right. We have these lovely spiderwebs today.
Mm-hmm. Oh, which, with who?
Probably be with us for a while.
Those like creatures right here.
Have fun. Okay.
Do some.
All the leaves now.
A little.
Oh, looking back, copepod.
Turn the mids down.
Who knows what's going on?
Oh, that's a cute little calico.
We put something in November.
Not calico. It has 2 bells. Weird. Oh,
Or we have something
AI
else.
programs are still safe.
Yes.
I mean, I haven't started. I'm just trying to get my head straight here.
A little bit training day on that.
Right about there.
I don't know what it's gonna come because it's so dependent on the using system. That was so
That's
brilliant.
a problem for future systems.
I've never seen it so good.
When we went to an aquarium, when we planted a balloon to the phone.
yeah.
Okay, it's not,
That would be
it's gonna be
good.
hard to get a piece clean and off.
Yeah, do you have
Alright,
a
so we're
razor
good.
blade?
Well,
No,
I mean,
no,
it could,
I
scalpel?
can come back.
So it's like-
I think
We
it's
got
got
no
some
feel
small
for
scissors.
weight
You're gonna
wise.
get right, but now
So we are
studying
studying
history,
history.
it's the cleanest.
Yeah, I use scalpels.
Yeah, we are a little.
It's a little hard to pin it down, but you can do what you want. I have scissors.
Not bad though.
As we
Move forward now.
They are also not the sharpest ingredients, nor
Yeah.
very big enough to do anything like that. But yeah, a bit of
Yeah, it's
tissue would
just,
be great.
yeah, it's just a shame with it.
We're going
Take a nice specimen like that. Not have an easy piece to take off.
Yeah. But at least it has redundant bits forward and down. Let's see this guy. Salp chain. Let's see who's riding along with the salps today.
Some Vibilia in there, perhaps?
Usually. It's very irregular spacing on those guys.
Salpa? Salpine?
That's what's going on. It's missing a few. It's nice regular pattern.
You know, Brennan really likes salps.
That's it. Can we get a close up of these guys?
They're so fast. You ever try and chase one? You scuba
It's got a
dive
nice
on a chain like that.
parrot in it. Third one down. So this is a salp chain, a blaze salpa. I'm interested in the little crustacean that's hovering in the middle of the fourth one down. Brown Bibilia. They have kind of boring
Yeah, we're
eyes
trying to
for
look
a
at
high
that.
parrot, but interesting comparison to the ones that have much fancier eyes. They're pretty much always associated with salp chains, and sometimes in very high numbers. So they don't have to do a whole lot of their eyes since they seem to be living on salps as if they're just the substrate.
On here? I'm not sure if I can see any hybrids. Most of the orange is the gun.
Look at that.
And the brain, the white things.
They have
Oh,
really
yeah,
interesting
yeah.
antennae
Oh, yeah,
that are
it's got an
scoop
amphipod.
shaped. Sorry,
ID on the amphipod camera?
that's Bibilia.
That's a Vibilia?
Bibilia.
Look at his head. Wait.
Very distinctive little
Oh,
scoop
yeah,
shaped
yeah, yeah. I
antennae
see. Yeah.
and eyes that are quite simple and flat, wrapping around the side of the head.
Surprise, surprise, Vibilia on a salp.
Who knew?
Species.
Probably no interest in collecting this. I think we can let it go.
Forward and down.
Vibilia is the third branch of hybrids, right? Its own
Oh, no,
thing.
we let it go.
Derived
No, Bibilia is a
independently from gamma.
physocephalata.
Okay.
They're in that group, pretty well inside that group. Paraphronema and cystosoma probably form a group that is probably, oh, there's a radiolarian. to the
Vibilia
left.
is a separate incursion in the pelagic zone by a benthic group. right?
No,
No.
all of Physosomata and all of Physocephalata are monophyletic.
So the CO1 data has been updated by something else?
Yeah, we're way past that.
Okay.
Cystosoma and paraphronema are the weirdos. They may be a separate group. They may be sister to the Physosomata.
Cool.
Still remains to be seen
I have to
whether
update my
they're out there.
database.
Ooh, look at this.
I
Off to
believe
the
you.
right.
You do not
Spice
want
next.
to sample this.
We do not want to sample this. Okay.
Not want.
Good.
We do want to get some nice tight zooms, but we
Get
did
some
not
nice video of
resemble.
this. All those beautiful speckles around are the
I never want to do it again.
tentacles.
So is this another siphonophore?
It is a Physonect siphonophore. All those tentacles
Dispersed chlamydia.
are nicely spread out. Can we look up at the head up to the left? Get a little close up of
I
that.
got stung.
You got stung
Yeah.
last night.
Yeah.
On the head.
That is the head. We can't quite see the details we need.
We need to see if there are any small tentacles coming out between the nectaphores or not on the head there.
I don't see any little tentacles.
Almost as transparent as Brazilian.
It's not apple-ing.
No tentacles between the swimming bells up here at the head means it's not an
There's
Apollonia.
yellowish
But that doesn't tell us what
around
it is.
the ostia.
Just tells us what it's not.
Weird.
Could be another Mars. Quite as long.
Okay, not apple-ing, something else. Yeah, the ostia have this yellowish green pigment patch on them. It's a
All right,
kind
what
of
else do you
bug.
guys want to?
Alabama wants to be on YouTube.
I think they're just hoping for as much of a close up of that head and just behind it as they can
No,
get.
it's different. There's no orange gastrozoids, for example. It's not coiling in on itself in that.
Just to have some nice
It could be
imagery
a
that might start at the head and scan, kind of fly up slowly. Which
All together,
Yeah, definitely
actually.
not
Yeah.
apple-ing. Which means it won't be as sticky.
doesn't want to lose any more bits.
Right.
Well, I think. Are you guys satisfied with what you saw of the head?
Not quite yet, no.
A bit tighter. What part are you trying to see?
Both a close-in zoom, like where we are now with one of those chlamydia units, and also the head there, yeah.
Yeah, so the
Okay,
top
so
part,
it's
a little bit of
T-shaped.
the, just below the
Make
head
this
is what it wants to see.
Hear that,
I'm
Joel?
gonna say a rhizomia probably.
Just a
Okay, and then
The
a
head
close-in
and
on the
just below
tail
the
somewhere.
head.
There we go.
all over here. Where? I don't even know where
Prepare
it
for
is. You can zoom out.
speed. So you see
Okay,
how it
thank you.
just- Did you get the
I think we're
tail?
okay. Not
A
really.
little different.
Not really. I think it's a rhizomia. Do you think it is? They were apolemias. They're apolemias and then a maris, which needs to be moved to its own genus. Especially
Wow.
Unless you can see the radial canals, it's kind of hard to, like in the nectaphores, it's hard to tell.
It dropped its cycle sound to make it like
See it right there on the right.
we do need to get a better look at the tails.
Oh, that would be nice. far away from the garden, from the place.
Let's hope we can pull something from that. So what it is. Alright, so
It's a little amphipod, looks like Christina.
we'll label it as what we labeled already. Link it. Yep.
Thank you.
All right.
I want to rename
Okay, happy with that?
And it's also the pigmentation. That's 2% connection force.
I think it's a Rhizomia, Resomia.
Yeah, center me up. Thank you.
All right. Let's find another creature.
Did you zoom in there? It's built up, right? You want this one in there.
What's up?
You want the,
Yeah, if possible, but they don't have to be completely out. We just need.
Spicy kiss.
Works well to look a little bit down when you're going down. Makes it seem a little straighter. You get a bigger field of view.
I think the autofocus in the main camera is like getting tripped up by that siphonophore.
mm-hmm.
Yeah, right now it's
Yeah, I think we keep it manual.
Period.
Yeah, the manual focus works better usually in midwater unless you have a really obvious target. Especially when we're hunting, because then you can keep it focused pretty far out and have the best chance of seeing something.
Black on the DPOs.
Oh.
That's the DPOV. Terrible. So green, yeah. That's the lower.
Looks like there's a lot of ISO.
So we're at 470 meters, headed down. Believe it or not, we're headed to 1200 today. See what we find in the water column. We're just below a second.
Not a lot of light out there.
Did you bump the data? I always had that around too.
Yeah, I did earlier.
That seems fine.
the fear of the deep. What?
Oh, little oink up above. Saw the last minute. Oh, there's. Look at this.
Nice area.
There's also a little jelly here. Either one. Maybe the jelly.
Was it the same? Oh, this
there
is
it was.
different.
It's another
That was
different
very
one, yeah.
different. Did you see
Alright,
the
we have
spines
to
were
zoom
much
in.
stiffer?
Yeah.
Yeah, very stiff and not
Yeah,
many of them.
So that,
That's
that's
Right
different.
there.
No. Let's just call them rice areas now and not.
I have a teeny drive
Oh,
on the desk.
did we lose it?
Yeah.
On my desk. The blue pouch.
You have a picture, right? Timestamp.
This one.
Yeah, this is totally different.
You have two other big ones.
Elevation house.
Elevation house. Yep.
Tiny little one. Too small to scan. So we can probably let it go.
So we're still just trying to go down.
Yeah.
So
Up above, there's a little jelly, that white dot up there. This guy might be too tiny to deal with. Oh, it's long from that direction. Wonder what that is. Can you get a little zoom on that?
Just try to get a little closer.
Oh, it looks like right there.
Zoom in too much.
No.
Do you think I might be able to dive
Yeah.
under there again?
Sure.
Apparently we can not get into NAS. Do it.
How big do you think that is?
Half a
There it
centimeter.
is, half a centimeter. Oh, it's definitely right there, yeah.
What was that bonk?
See the silicate spikes?
Yeah, I can see that
It's
it's
spinning.
connected. I think that's, it's not spinning. My
I
intuition
think
is
it's
that's,
an effect of the lights and bouncing around.
Yeah.
oh,
We should just see what we don't have spared for yet. It's really just the small stuff.
might be spinning.
Actually,
No, it's either.
bulkheads.
Oh.
Like it could be quite pretty. It is quite tiny.
Yeah, this could be a solitary coloidarian actually.
We can, but it's.
Yeah,
What's
with
that?
the, you know, the egg dropping in the middle?
Yeah.
Yeah, this is a,
Is the
it's
balance
either a
not as
colosome.
good today?
Sequence, which is sure.
Yeah.
And a nucleus. Or do some analysis on it.
Pretty tiny dot to stay on.
Just make it dedicated.
All right, we can move this guy.
Why did we have to recycle that? Say 5K.
Oh, it's red in the stone. Yeah. It's the problem with these tiny things. We zoom, and keep getting closer, and then bonk. They pop us in the forehead.
Keep your eyes out for those Phaeodarians that hold hands.
Oh, yeah.
I think Manu
Maybe
would
we
like
gotta
one.
try to catch one.
Would like some of them, right?
Is the focus on Daniel?
Mano,
Yeah.
In the troll from today morning, I have a new.
In the middle, coming up in the center.
Big jelly,
Yep.
straight out.
Right in the center right now, yeah. I think it's a Halotrephes, maybe.
There's indeed
Like the
something
guy we
like
got
a
yesterday,
Halotrephes.
perhaps. And that's a Tusker. Is that a Tusker? No.
Not
It's
with
a
his tail
Lensia.
like that.
It's a Lensia, yeah.
Lensia siphonophore to the right, and probably Halotrephes dead ahead. His tentacles are beautiful.
It's supposed to be pretty good, but.
Like with the largest one of these yesterday.
Lensia
Ever seen.
had an orange
Only meant to
cyst.
collect. The whole thing went in. So, oh, he's ready to go.
Went a little to the right.
oh, it's here.
Should be hydrated. Doesn't even say how fast it is.
And back up.
Canada just showed up the side cam.
Yeah, he's in the
wants to go in the
sit
jar.
pin. He wants to fly right up through the middle from the bottom, which is a real trick. He's definitely in escape mode now.
Krill. Platyspheloides. That's the tentacle from the Mars.
Do we need another one of these?
No, I think we're okay. If we can count the number of tentacles, it would be useful. But
Well, he would have to. Ooh.
oh, stop!
Look, this
Xenophore,
guy
xenophore.
over here.
On the right.
Ditch the Halotrephes.
This sparkly guy.
What is that
here.
xenophore?
very like the other Mertensia.
It looks to be Euramphea, maybe.
He's off to the right, going up. There he is. Or she. Oh. Who is this, Dougal?
I think it's Euramphea. Where the two pointy
Euramphea.
bits come out the end, if there are filaments, like threads coming out from it, we can get an idea.
Oh,
The lobes look small enough that I would imagine Euramphea rather than
can potentially
Chiromaea.
just back off and let him settle a little bit.
I thought I
maybe.
saw a filament just
We were
then.
going really fast. Such tiny little lobes.
I'd say that's Euramphea. Yep. Those two triangular projections, whether they have a thread coming out of them or not. It's Ctenophora lobata.
Threads coming out from the pointy bit at this end.
Euramphea. Euramphea.
I do.
Is this still?
Supposedly there are cryptic species on there, but there's a couple of cryptic species that are already described as well. So I wouldn't know where to start doing anything taxonomically with it.
Yeah.
I mean,
I mean, those are all, I don't really want to drink them, but I told you so basically.
One more. Oh, yeah, I'm pretty sure I saw the thread there.
then I learned that you can actually do
You want to collect this, Dougal?
No, I'm happy. Thank you.
Okay. There are
Oh,
definitely
there are
threads.
threads. Yeah, you can see the threads. Yeah,
Great.
that's a positive idea.
Yep.
Euramphea vexilligera.
All right. I think we're good with this guy.
Karen's sorry to distract. Lots of the
Oh,
basically
nice. Thank you.
against the dog.
Yep.
They're
Yeah.
getting really good.
Perfect.
Jar can for anyone who's here.
Yeah, that's
Turn that
nice
next
to
to
see
it.
over there.
Yeah.
Yeah. All right. I
So
think
good
we've got a
training footage
positive
for
idea.
AI. That's good.
Let this guy go.
Okay, let's go find something new. Something new and shiny. With bells and whistles.
Oh. Too small. He came out the edge bar. Can we? I think the direction is good. It's just a bit fuzzy out
Focus
there.
out a little bit to
Things like put the focus out a bit further, maybe.
South
Oh,
chain.
south chain up above. John said salps are a great target for genomes.
very bright lights.
Yeah.
Coelodendron, maybe?
Oh, there's a little jelly up here. It's an oink.
Oikopleura.
It's a larvation. Little tiny one, probably too small to scan. It's very spherical compared to some of them.
It's pretty organized.
Sparkly. Alright, we can leave it. Yeah.
Is that a fish directly in front of us? Or just a string? Just a string.
Just a string.
Also arm.
Another oink over to the right. No, it's the small ones are too small for me. Oh, Tomopterus down below us.
Tomopterus.
Look at that one. Yeah. A little video of this guy.
There we go.
Yeah, Wiley. He went up. Oh, he's over to the right. I haven't seen him this one at all. They are incredibly maneuverable. Yeah, that was an interesting one. It had
You know what
almost
pick
looked
we
like
need
an
to develop
Apterus,
with
which
them?
is the second genus
Besides the track
of
UV,
Tomopterids.
To get rid of those.
a windshield wiper.
I want to for dome for.
To a port windshield wiper. Isn't there a group that chasing coral?
We're not the only ones. There are others
Uh,
different.
What's this guy? Or is that just out of a dot close by, but way out of focus? It really is trying to focus on these strings all the time.
It's on, I think it's on autofocus and it, the
Does it
science.
keep getting itself back to autofocus? Yeah, and nothing's moving, so it can't be real. Ooh, let's see that though.
Focus,
What is
focus.
going on with our main camera? It's like zoomed way in.
There we go. Oh, Pyrosome.
Pyrosome?
Our first Pyrosome.
Oh, talk about bioluminescent. Probably Pyrosoma atlanticum.
One's already been made. We need it.
We need it. There's usually some really interesting amphipods on these guys.
Food.
Atlanticum,
Is it?
typically.
Pyrosoma atlanticum?
Yep, Pyrosoma atlanticum.
Take your word for it.
sounds.
Yep. Pyrosoma.
Pyrosoma.
Any interest in this? You guys want to scan it? It's pretty dense gelatin.
Yeah, I think it's probably.
What about iris? Might be interesting, iris. It's nice and dense, so it would
Let's do Iris.
give you some depth.
Give us
It's going to be pretty small.
It's not going to run away fast. That's good.
might be good.
Were you happy with the setup of iris, or do you want to straighten it out?
Yeah, Sarah, what are you thinking? I was worried that it being so far to port, you just have so fewer perspectives to get things aligned.
Thing is,
But I don't
it's
know,
not new, so it's
Well, also,
Yeah.
Iris. So I can try
like, close
could,
to the center.
bring it to the center, but then it's gonna be hard to resolve that.
It's kind of it is.
I can bring it.
yeah, technology.
It's kind of it is. It's more like, situations are on a new animal,
We find a way.
like being able to go from one region
Sometimes they have like shrimp sitting on them, for example, and that would be cool.
We did do a fun
But this isn't
experiment with an iris before seen die
Yeah.
in the lab.
Right.
Yeah.
Oh, and have it
Very
shooting
fun.
at
I
the
haven't,
end,
very
I
like,
haven't tried the iris
Yeah,
yet,
yeah.
so I'm not sure.
like those,
I'm
chimney of
game
dye.
for
like those
whatever.
chimney sponges.
Whatever was
Yeah.
helpful
Yeah.
for you.
Well, that is already up for you, and then I'll turn the
We're gonna, can I take this?
lights
one?
on.
Sure.
Oh, right, but then-
Which one?
That must be my core.
Maybe get closer again. Okay.
Now,
Can I take the science?
Do it.
When I was
Let me
looking
see.
at lights dimmer,
No.
Wait here, you're good.
it's probably still a good setting for the lights.
So last time it was really hard to tell whether it was, seemed like it was always too far or too close. Like
Yeah.
figuring out where the sweet spot was incredibly
Right,
difficult.
but I have found that sometimes in order to the
There's
depth map to really
cyclophony,
show
hard
up,
to tell from
we're
this angle.
adjusting the gradation line to make sure that it shows up okay. Run full side place on.
Clear.
How's that?
Better. So that,
See?
so that left is down.
There, it's gonna be a hot.
We can just grip it in the
They
jaw
can
from the
just-
side tomorrow. So that you can do this and have it point normal.
See that? Oh, there it is.
All we can do is give her a go.
I mean, do you want it straightforward or does it work in this way? Have they
You want
done
out
it
front?
that way? So that you are seeing where you're putting the animal?
Pyrosome or Atlantic.
This,
There's no like laser or anything.
Yeah.
yeah, but you want
Yeah.
the axis of the
And it
view
doesn't
will
seem
be
to have
good.
shrimp sitting on it, or it's fish living inside it, unfortunately.
I think trickier here is the 90 degree.
I mean, I see it
You
from
can't see a
here, so it's
lobster
gonna be
wrapped around
trying
it,
to
like a
get it
lobster.
in there and
Yeah.
know where the sweet spot is.
With this setup, like a down looking camera would probably be. So the instrument that we pulled out now is called Iris, and this has a
Oh, yeah, there it
planotropic
is. That was-
lens,
Very close to where we want.
or something similar to that. And that means that it has 2,400 micro lenses embedded in the lens, and each lens has a different focal depth. So if you can do the calculations and put all of those individual images back together, you get the 3D structure of the outside, or the depth information on the outside of the part that you're looking at. This imaging system works really well with things that are not
There it
transparent.
is.
It prefers to have systems that it can't see through to give us depth.
Might have been too close, actually.
And what it does is give us a color-coded image of the animal where cool colors are far away and warm colors are close up. You can see the details of the animal. Has these extendable arms to light the area really well where you want to put the animal to image it. And this instrument produces incredibly large amounts of data very quickly because it's got information all from all of those 2,400 lenses.
Oh,
So it takes a massive computer to run this
it's above.
system.
It's above.
Because the thing is they could take advantage of our crossing. It's
Hello.
if they put iris to the walrus.
Oh, yeah. Good move.
I didn't see any depth, but
We have sequenced a lot of these animals from various areas in the Atlantic and the Pacific. And they all seem to be a single species, which is a fairly unusual situation. It was the assumption for a long time that most oceanic or midwater animals were the same throughout the world oceans. But when we go in and look more closely, accumulate more samples. We use genetic tools to tell them apart or to tell if they're the same. We end up finding out that most midwater animals, like most other animals we're familiar with, actually have quite specific distributions. Pyrosome atlantica, which is what we're looking at now, is quite different from that. They seem to have a nearly global distribution and the same genetics everywhere we look. They're a colonial organism. They start out with one little cell and quickly produce 2 and then 4, and then they grow into this long tube-shaped colony where they are talking to each other with the light or the bioluminescence that they produce. And each one is pumping water from the inside to the outside or the outside to the inside. I'm pretty sure it's from the inside to the outside, but I'm not totally sure. And the whole colony moves together by pumping water through the zooids or through the individuals of the colony. And they coordinate their movements so that they can grow. They can get really large enough that a diver can actually swim into the inside of the colony. They bioluminesce like crazy. Some have tried to implicate them in being mistaken for torpedoes. As you see the large torpedo-shaped objects moving through the water at night, glowing as if they're hitting a bunch of bioluminescence, but they're actually producing the bioluminescence themselves. They're a really fantastic place to raise your young if you are a hyperiid amphipod. The young are often dropped off on the colonies and burrow into the colonies. They'll eat a single zooid and then use the gelatinous matrix that the colonies are embedded in as a little hiding place. They come out and climb around on the colony and eat various parts of the colony or eat things that are caught on the outside. So right now we're just practicing. It's pretty tricky. It's a small area you have to get the animal into. It's pretty tricky flying to get this little guy right in the sweet spot so that we can see it in the iris camera.
This far down the predation line, or is this just to see?
Just if I can see something.
Once we're not fully blowing out with the predation line straight.
He's just to the left. Center is coming over. There you go. Yes, why do you guys use red lights on iris?
Sorry, what's the
Why do you
question?
guys use red lights on iris?
To not scare off animals.
No.
Many animals can't see
Yeah.
the light.
Yep.
The camera itself is monochrome, so the color
So it
doesn't
doesn't matter.
Um, And actually, you can see the yellow box. We get a nice view of iris here. The yellow box to the side is the light circuits. And as opposed to many of our other housings that are one atmosphere air-filled housings, this is an oil-filled housing. So it doesn't have to be as bulky.
Don't have to worry about it leaking.
Well, you do worry about it leaking, but it's much less disastrous
Yeah.
if it does.
And if
You-
water gets
how's
in,
yours?
does it not proliferate?
Yeah, you don't.
Because the oil kind of keeps it out or no?
Then it very much matters of, like, depends on the orientation because of the way water and oil interact.
Okay, yeah.
But because of the hydraulic systems, you can over-pressurize the housing slightly
Uh-huh.
so that you're leaning towards
Pushing
leaking
oil
just a
out,
slight droplet of oil out rather than seawater in. So that if there's a crack or something like that,
Despite the current.
your instruments are still in oil and not
Yeah.
salt water, which is great.
And is there anything you have to think about in submerging your instruments in oil? You're probably pretty happy with it.
So for many it works. Heat connectivity is a little different. But
Slower?
some things like optics don't like oil. So the main housing, titanium housing we're looking at here, that one is air filled because we're doing a lot of fiber optics in there, and there's camera and lenses. we wouldn't be able to see that. But that's why we separated like the higher power light circuit outside of there, which is pretty nice design. It is independent. The instrument would work without the entire light shroud too. It's just to give it more light on the actual animal.
Which is probably fairly useful when you have so many little individual lenses. They need a lot of light for each.
Decent
One, or-
amount of light. But we're finding that, for example, benthic dives, we basically only use ROV light because it makes a really nice, fairly even field of illumination due to
Yeah.
like wide spacing.
Mhm.
And then we often don't even turn on the iris lights
Oh, okay.
because they tend to be too bright. As soon as you get something reflective like a sponge or just like bottom substrate or even
Yeah. Team rotors.
there's octopus, they're like
Oh,
so
even
bright.
the octopus. Okay.
Yeah.
So why can't it do transparent animals so well? It just can't pick
It
up
needs,
those
yeah, so
contrasts very
yeah,
well.
the calculation depends on like matching patterns.
Mhm.
So any sort of texture works pretty well.
Yeah.
But transparent isn't
Not so
really
good.
giving you texture.
Yeah.
Plus, for each location, it tries to put in one distance. So if you think of multiple transparent layers, like there
Yeah.
not one solution of where the tissue is.
Right.
There are multiple. So it works very well for opaque things.
Yeah.
It works for things like teen rows, but then in between the teen rows, well, T4 you might not resolve really anything. You can still do valid measurements on that, right? If you're, like Dougal was saying earlier, what you want to know is the distance between the plates on the
Teen
teen
rows and all
rows
that, yeah.
or the relative teen row placement. Doing absolute measurements on that is really cool, and it works. But, you know, following the actual shape of the transparent tissue might not work. And for that, we go to DPIV.
And as you've developed this iris over the last few years, have you kind of fine-tuned what you want to get out of it and types of things that it's most useful for, and kind of gotten rid of a few things you thought you might be able to do with it, but replaced it with other things that it works well for?
Yeah, we found it to be pretty versatile, but we've also found the analysis to be extremely time-consuming. Because with DPIV, we spend a lot of effort to make sure things are lined up really well with the laser
Yeah.
sheet. You know, with the help of skillful pilots to like make that happen, so that we collect exactly the data in the plane where we want it. With iris, we're not constrained by that. But to analyze it, we still have to do that. So finding a reference plane, especially for midwater stuff, has been one of the main challenges.
Okay.
So you have the data for it, but now you have to do a lot more work after the fact.
So the computational software that you guys are working on for the Ocean Job grant with John, that is for iris or for DPIV or
Both. Yeah, so for
Great.
both, we are working on improved pipelines for data processing, but we focused on different areas for the different instruments.
Yeah.
And it's a multi-stage approach. For iris, for example, one thing that was really limiting is it's a very high data rate, like 100 gigabytes per minute. So we can't really record continuously.
Mm-hmm.
So we were like not trigger happy enough. We miss important
Yeah,
things.
which is why we have the auto when it comes in with it.
Yes, so
view
we
it records, and when it's not there, it doesn't.
Yeah, a very recent and not so trivial addition was the pre-buffer. So we can essentially record things that happened 5 seconds ago. It's just continuously, temporarily
Replaces.
saving them into a buffer, so that we have time to press like, oh, this was the good time, the good moment we wanted to record. And now we even got these events, so that when we get good depth maps, so we're resolving something that looks like it's a good, gives us good data, automatically triggers. So the computer is constantly running and trying to do these calculations. And but so far it seems to be working quite well. There are times where I was too slow to get to the record button, and it already did trigger.
Yeah.
This one didn't record, right? No, because I don't think we resolved depth on that one. It was just outside of the volume.
Don't worry, I've recorded things.
Okay.
This
Yeah.
thing hasn't automatically.
But I'm not sure we resolved the the depth, and I'm talking about depth, but depth here is distance from this camera. So
Depth
in
of
this
the
type of
object.
computer vision, talking about depth is common, and it's not so confusing when you're doing like in a factory measuring the screw pitch of your manufactured nuts and bolts and stuff like that. It's a little bit more confusing when we're at depth talking about depth measurements, which is distance from the sensor.
Yeah.
So when we get a good depth map on this iris view, you'll see different colors overlaid, and that is what we call a depth map. So blue would be further away from the camera, red would be close to the camera. We're aiming for something in between, but just getting there is pretty challenging. I agree, Katanya. I think we need to bring the box camera back.
Yes, I just.
Have the mount for it.
Do you? Then we can ask if we can have
Yeah.
another
Put a tiny camera next to one of those lights so that you can see when it's coming.
Yeah, so basically it's just a little backpack for Iris with an additional camera with a slightly wider field of view. It lets in a bit more light. So you can use that to have a more intuitive view as a pilot. And I don't know why we kind of dropped it on the last expedition. We had it on initially, took it off later.
But why is it why is it because I thought we were doing one?
Did I hear it all right?
Almost.
Almost. It's in there.
Just hold it in the manipulator at the end.
Yeah, put it over.
What if we catch it first? In the D sampler. And,
Animal tracking.
I mean, that did work pretty well. Right? But for the product, for the sense. This is true, we didn't bring enough hardware.
Oh, just to the bottom left.
We still think it's spiders.
Close enough.
But we still think it's spiders.
Yes. We're trying to go to 700.
RIS. RIS stands for Remote Imaging System. The transparent layers do not tend to degrade the capture capability. You really just look through it. And yes, there's absolutely benefit in capturing those. Before we were looking at team rows, but also like gut structure and things like that for siphonophores. So we do try. Oh, there's a good image. And it just depends what we're after. If we really want to resolve the transparent structures, we'll go to DPIV.
I'll take that.
So what is that thing that just came by that we did resolve?
I don't know. I thought it was the Pyrosome.
No, that was the Pyrosome.
That was the Pyrosome.
Yeah, the Pyrosome.
And there's some krill every now and again.
No, it looked like, I don't know.
We don't know enough to know if it's a really hard thing that they're asking to see or not. So it's really interesting to learn about how they've developed and watch these technologies grow. These have been out on the first two Designing the Future expeditions a few years ago, and now this is a evolved version of it. They can do a lot more post-processing, have some ideas, as you can
There's a
tell.
second foot, eh?
There's still some things that need to be worked on, fine-tuned to get this so that we can really get the data that we need from this. Oh, look at that in there. Right. Handing it over to Heather Judkins. Mollusk researcher extraordinaire.
I think we need.
Maybe you can get her to talk to you about Pyrotutis, one of her favorite topics. So,
So it needs to be a long distance away from those lights.
And we always say this, but we also need the 50mm lens.
I agree.
The
Write
zoom-
it down, yes.
The
write it
zoom
down.
lens is nice, but
Yes.
also not. You have one big summary of things to
At the bottom. It's been a little while since we last did.
You have a little auxiliary camera. It takes so little bandwidth. I wonder if we can splice it into Iris.
you just early.
Yeah.
Right.
It was just to
Take
the right.
wait till I'm here.
Huh?
I want to wait until I know we're actually doing
Yeah.
it, and then I get involved with watching it, and then I don't write it down until we're done.
Fun.
That was waiting the whole time.
Cancel. Okay. Oh yeah, that was a good one.
Took a long time.
Is that what you were looking for? I'll take it.
Did it also count as an event?
Thanks, Sarah.
This
No.
is more difficult.
Yeah?
I can always come in and show you how it's
I wasn't paying
done.
attention. Thank you.
Find out.
Hi. You have somebody coming up.
You ready?
Yeah. Are you done with this one?
She's putting together that.
It's actually kind of a nice way to do
It's like we got a little siphonophores.
Great. So we have another.
Okay. Zach would love to
So-
hear about it.
Here's a picture I just got.
Are you still recording?
Is it sensing?
Science is
That
on.
just went straight.
Extra.
Sarah's still
Science is on.
Are we moving on? Or are we?
Have we
Jump in.
done anything? We have one bucket.
We just got one. That's the event trigger.
And it didn't trigger?
It did.
Great.
Okay.
Girls.
All right. So, Katani, we're good to move. Everybody good? I know.
Whoa, this is,
It's good.
this thing's smoking hot.
Yeah, sorry.
She's been working hard.
I'm
She's been working hard.
ripping
Oh
it.
my gosh.
She had some minute detail to work
Sarah's
on.
like, I want to.
Earned your dinner tonight, didn't you?
She wins. the
Gotta
game.
burn calories somehow.
Like,
That was a
Crow.
9 points.
Wait.
really big camera.
All right. Hi, everybody. This is Heather Judkins again, University of South Florida, St. Petersburg, invert zoologist. I'm taking over for Karen for the next few hours. And we have just finished some really cool, intricate work, working with Pyrosome, getting it on their camera system. We are now going to continue moving down towards our 1,200 meter depth mark when the pilots are ready to go.
Oh, we're done with this guy?
Yeah,
Okay.
we are finished here. We can close up the camera, yeah?
Please.
Yes.
I saw that too, dual ink, right?
That, okay, so that two together, that's how it's supposed
There's
to
something
be.
on the
Looks
right.
weird, huh?
Mid screen.
Oh, yeah, I see it.
I
What? I'm sorry.
Heading left.
we want the camera to do this, right?
Yes. Is that the Pyrosome still?
There's still a pyrosome in the view, yes.
Yeah, different one.
Just the.
Collected on the camera lens. Let's go.
Yeah, I don't know if the 90 degrees was helpful,
Ours is
but we're trying all
three.
sorts of.
Feed area, Coelodendrida.
We could really couple between
Coelodendrida.
All right, so the goal, nice and slow,
Where'd you go?
a slow snow.
Somewhere
As we slow snow as we angle and descend slowly towards 1,200
You
meters.
heard me?
If we see a great target, that would be good.
We're looking. I want to get one because we see enough of them. I want to get one that you've got two
All right, moving ahead and down.
No, let's
Yeah,
get
just
one.
one.
Let's not be greedy.
Single one.
Trust me.
It'd be good.
I'm
Still
not
a little
even
light, huh?
sure we would know. Have they been captured before? Because I
I've
can't
caught one.
find. I
Yeah.
haven't.
I put one in the
Do you
ship
have
in
a silica
2000.
structure?
Yeah, back in.
Yeah, because I
year
tried to map
2000 or
it.
something.
I don't map it to any of the described species.
Now that you're having fair in the top right hand corner just there.
It would be cool if we can do two and we can check whether they're cytoplasmically.
And then if we have the
connected
zoom as far
or
as
just-
possible for search mode, that would be great.
Okay.
So-
You're confident that we'll see.
Dougal, we're okay with the
Oh, yeah.
light?
Let's do that.
Well,
I
we
see
wanted it.
almost as many pairs.
We
as-
had it.
Let me set it up like yesterday.
No,
UVC,
nope, we did
what have
not.
you
We
been
changed it
calling
from
for
yesterday
Minifero
because yesterday was
line?
too
It's
bright.
not a 4-hour.
Yes.
So
Stand by.
even the stuff that we see.
Let me cross check
we're
with
not
Dougal if they
describing.
changed anything from what we started today. But what we
Oh, I
started,
see.
did you change things from what you started today?
It's a Theodarian.
Yeah, I didn't think it was on the science skin.
I've seen forums
Today.
very similar. Like
Right, that's what we wanted, right? And we were happy with that. That's what I thought. Yeah, so
but
however
it's
the
a
settings are
simplification,
now, we are
suitable,
happy with the
yeah.
camera setting.
Like, everybody calls it the Hespetina, but those have straight spines.
What is this? Is this siphonophore?
These are the ones that kind
It's not
of
stuck on that.
Yeah.
Correct.
Yeah, that's in the
Now,
Nakamura.
this is what we
Right,
want.
it'll be
It's
that.
totally different. Like it's completely new in terms of that's the actual structure. Spines, every tip of them then has, and the only description.
And if we went a
is
little
actually
more
naturalist
forward,
Is that something you
I-
imaged?
Yeah, I just imaged yesterday. That's the Theodarian echolitron. This is the only one that comes even close to it. But every one of those lines has a, this is like 10 micron. These little lines are maybe 2 microns.
Are you in contact with Nakamura Yasui's? Oh,
stop,
baby
Just see what that
Apolemia,
is.
maybe.
middle, just to see,
Yeah, this was the species. That's
Moving
like
whatever it is.
right? Just to see what it is. Might just get a zoom quick and keep going. I don't think it's anything to hang on to.
Looks kind of like that thing
That's the thing that it closest to.
Mm. Maybe
RV makes more sense.
All right, give me a little zoom if you're not there.
that'd be great.
And then we know it's a Theodarian because it has to.
Might just be snow.
No, it's an Apolemia, a baby Apolemia.
Oh, it's a baby apolemia. Too funny. Okay.
It looks like it might have lost its nectaphores. So it's a bit of stem,
Yeah.
a piece of stem of,
Yeah, it's something part of them. So it's a part of a siphonophore, right?
yeah.
Yep.
Something eat it.
So their stomachs, the brown things are stomachs, the other white things are palpons, and they seem to have some kind of a spherical body in the tip of each palpon.
Would this be a remain?
It's
if
it's a little
predator
piece of
might-
tail from
Yeah.
that's come dissociated.
Early stage, they say, is they'll just follow them.
Oh, this is just small.
Yeah. Before it's formed a big colony.
And then so far we've only used one bucket, right? And no samplers?
Two suction samples we used.
What bucket numbers?
4 and 5.
5, yeah.
4 and 5. Okay, so this is just not completed. Okay.
We're looking for the guy, the coelodendrid, that guy with the 4 arms coming
Yep.
out, and they occur in pairs holding hands.
Yep.
'Cause
I'm
we've
on
been
it.
trying to sample that the last couple of dives, and
Right.
That's a
Not
high-
successfully.
a high priority target.
Okay, perfect. Great.
Don't really want any more siphes that are going to attach themselves to our camera lenses.
Right.
It's
All right.
aggravating.
Lower priority at this point.
Yeah.
Got it.
And then we should try getting the hair-like rhizaria again, the big ones.
Think we should try one more time?
But for this week,
Do you-
Yeah, because it's not a foram.
Yeah.
I think if we really now want to confirm it, 2 samples would be amazing.
Yep. And you've got one from
I
yesterday?
have one from yesterday.
that
Okay,
from
but
the
2 would be
sample?
better. Okay.
Yeah,
So you did
only
find
the
it.
desampler, only from
Yep.
the rotating
Right.
sampler. Otherwise, it didn't make it.
The rotating, so the suction sampler, not the
Suction sampler, sorry, not the desampler.
Okay.
It's not possible to get it in the desampler.
What's
Yeah,
that?
I think
Left
the
hand,
blue,
middle,
left.
left middle.
water
Yep.
breaks.
Coming back.
Ctenophore, it looks
If we
like.
could zoom in on the middle. Yep. Sorry. Yep.
Looks like a ctenophore.
When you are ready. Yes. Yep. We have a ctenophore.
You ever see close and down to the third gun?
Yes. They do. Bless you. Bless you.
Interesting approach to return.
Get a bit closer. Does it have oracles?
I don't know. I don't see them. Maybe it could be the angle.
Maybe a bit more focus.
Any ideas, Dougal?
I'm just waiting for it to come in focus.
Yeah, yeah. There he moves. Okay.
I think.
How do you feel about focus? There you go. Perfecto. Thanks, you guys.
There we go.
If only he would. There you go. Perfect. So this
It
is
looks to be a
ctenophore.
lobate without oracles, which
Yeah.
presently there's no described species that are like that.
Okay.
So it's new.
So, does that? Do I hear suction sampling?
Are we? Is it big enough to scan, or is it like too small?
I don't know that answer.
So lobate without. I think I just need
Oracles. AUR. Oracles.
It's
Ickles. I see. Yep.
its pet name is Lyria, Llyria, but that's a grey literature name.
Gray.
What's grey literature?
Closer to our immune system, just so I can.
Google, what's grey literature?
So it's a
Like literature.
You got that, Zach?
magazines,
Oh, I
daily
see.
scientific journals.
Going real slow, so
No. I just want to. I didn't want to. It was coming from Kakani.
It's like the
It's
dark
perfect.
web for scientists.
The DPI
Magazines.
Or actually, I can do it.
you can do it.
Dark. Oh, there's a second one now.
Possibly, yeah.
Oh, yeah.
There's two
It's the
ctenophores.
top left one that we want.
No,
It's up now. It went up.
we were working on this one close.
Here it is.
I think
This
one
the
in
close
center.
one's the
The
one we want.
close one is the one we started with.
Yeah.
Can you pan down a little bit with this?
Pretty tiny.
Itty bitty,
Yeah,
teeny tiny.
looks small, but Kakani, you decide.
You can't see a good scan.
No, I think it's still a little
Far away.
My guess
So is it that?
My guess is like 8 centimeters.
Oh.
I think it's smaller than that.
I just don't see the DKB camera, which is the
Mm.
thing that I will have the best.
It's still a little bit far away, man.
Yeah.
Can you hit DPIV mode on
I'm a
that?
bit closer.
You got it down.
Already done? Oh, it is, okay.
Oh, you mean the crossing camera?
No, I got
You
crossing
got
on.
both. Yeah, you got both
Okay.
of them.
Oh, okay. I'm used to it being black and white.
Yeah, it's a setting. I need to change it.
Okay.
You focus there? Okay.
If it's that big, then I think we should be able to. Structure's so deformed.
I'm moving around a bit. I can't see it in that camera at all.
I think it's still far out.
It's, yeah.
I think it's about there now.
He's closer
Or a little
now.
bit in, yeah, closer now. Is that it there?
Nice, too close, now he's out of focus.
No, it's close.
Can we just concentrate maybe on the 4K in focus zoomed up stuff maybe then instead?
You want to focus on pictures?
Yep.
Okay.
Let's focus on pictures. There he is. Yeah, he just occurred in the side camera.
Yeah, I'm
Good.
coming
Can
to
we
the right
zoom
queue
in
then.
even more?
Could we get a little more zoom on this?
Let's see what those white spots are at the ends of the canal.
Yeah, the canal ends look different.
Cool. So it could be bioluminescent organs. They could be something
It's a little
else.
close.
Get it out of there. Up off screen. There he is. Middle.
One
Do you
more
wanna-
one more nice zoom and then suck him into one of the canisters, please.
Exactly what I was gonna ask. Thanks, Dougal.
Yes.
All right, let's get a zoomy zoom one more time, and then we're gonna attempt to do suction sampler. Yeah, of course.
It looks like the canal's coming from the infundibulum, meeting halfway along the meridional canals rather than at the apex.
That's great. That's great.
Bright white spots at the end of the comb road.
That helped, Dougal?
Very nice.
Yeah, that one just helped, right?
Okay.
Is that enough,
looking good.
Dougal?
Yep, let's suck him
All
up.
right.
See if we can get some
Suction attempt.
shadow.
No, it was here. Karen had it, so whatever works.
What bucket? Three right now?
Dougal, that's
Three's
well.
good.
Wait, three's that was super fine mesh.
They're all super fine.
All of them.
Did you
They're
make
all
them all
superfine
super
mesh.
fine mesh? Oh, okay.
Yeah.
Great.
Chained it.
3 it is.
Look at that.
See it? Still raining.
Yep.
Going a little bit of suction, but not too much.
Yeah.
I got both the second part.
Okay, come out.
First dive and the first part of yesterday's dive. Two more labeling runs at CMS.
I don't see him.
Oh, how many?
Just to the right.
Oh, there he is.
The last one is part one of yesterday, 644, but some cool ones with the galaxy side, but also a lot of
Small.
It's a fair bit
VIP
small.
scans.
Yeah,
8
he's
centimeters,
small.
Isn't it?
Some of them with not much
I think like 4 or 5 centimeters.
difference or hard stuff to look at, but also some are kind of cool.
No. I think he went past.
Maybe spark some
Is he?
discussion on
Yeah,
these.
he went past to the right. To the right, right, Dougal? Yeah. There he is. Coming in the middle. It's back in the middle.
No.
Right? No.
I don't think that's him.
Nope, that's not him. I lied.
We might have lost him. But there was another one in the same frame before, so I think we'll find more.
Yeah.
Stop and suction.
Do you want to
Yeah,
stop searching?
Stop suction. Yeah.
And
Look, it's in front
Oh,
of
there he is. There he is.
there.
In the middle.
Middle.
Yeah. The center.
That.
Thank you.
We haven't been looking.
He's there just in front.
Now that you're here,
Yeah,
will you able to do
tell
that?
us if you see
Oh, it's on the
fish.
side camera now
For us,
too.
that's just background noise.
Repel in the mouth.
The light catch.
He's back there still.
No, I haven't seen it. Was that your little container on the porch? The little black thing? No, you didn't put it in.
I think it's hard to see.
Someone put
Yeah.
a little black
I think we-
printed.
That might be him right there.
Yeah, they're right.
Do you have any success
There.
making a trap?
Yeah. That's him, right?
We'll try.
Come off a little
Yeah,
bit.
same species at least.
Close.
Yeah, you could see him on
Yeah.
this camera as well.
Oh, yeah.
Come off.
Yeah.
Out and left.
The little Lyria there.
Yeah, okay, another good spot there.
Yeah.
It's a Lyria.
Suck him up, it's almost invisible.
No,
isn't
no,
it?
no, right there, right.
He's
Come
off to the right. Yeah. Coming back.
Yep.
Back.
Okay.
He's a bit tiny, isn't he?
Yeah, he's pretty small.
So come up a little bit here.
I think since that isn't quite done either, it's so slow.
We haven't done
anything
but try to use
No. Yep.
I didn't know if you wanted to switch out.
Maybe, maybe not.
I don't think so.
Okay, let's go find something bigger, maybe.
All right. Are you good,
Pretty
Dougal?
hard, yeah.
Oh, yeah. He's right there. We'll go one more shot.
Sorry.
So I was using this count.
Yeah, kind of bouncing back and forth.
Hot. And then stop it as soon as it goes.
I don't see him now.
Yeah.
Starboard and backing up a little bit.
There he is again.
Oh, yep.
Oh, he's in.
Yeah?
He's in the suction sampler.
You think you see him in the
Something
section?
just went in, yeah, that looked like.
Yeah, okay.
Oh, yep. Okay. So we'll call that a ctenophore.
Lyria, Llyria.
In suction 3.
Section 3.
All right. I didn't see it.
But I believe you.
There's another one. That was another one right there. All right, we're gonna continue our way down. If we could,
Rotate the.
yeah, rotate the cups, right?
Yeah.
The canisters.
Okay, do you want to wait till he gets in there though, or?
Probably say.
I saw him come into the canister.
Oh,
I
you
didn't
did?
see him go in the pipe.
Oh, okay.
I didn't see him go in the filter, but or into the suction.
Okay, rotate
Two.
to 2. Other 2.
By the way.
That 2.
That's the one. Thank you.
Okay, moving ahead down.
That would be great.
That's 3, right?
All right. Crustacean. Oh, that part? Oh, yeah.
It looks like we're in space.
Nice to see what those white spots on the end of the cameras are. Stevens.
And the, all this water, all this stuff.
Velocicalis, maybe?
Oxygen with a little clear solids.
Yeah,
On the right,
oxygen
yeah.
minimum. Yeah. Interesting.
Straight ahead.
Yep, middle.
Is that another pyrosome?
Broad. That's what it looks like to me. Looks like a pyrosome.
Oik on the left.
Do we want to zoom in on this pyrosome?
Yeah, may as well.
Yep.
Drive through it, collect it as roadkill, move on.
Okay. We're gonna zoom in on this pyrosome, get a zoom, and then we're gonna keep moving
See
ahead.
if there's anyone sitting on him.
Sometimes they have some great symbiotic relationships with organisms that will live in and on these pyrosomes, so super fun to take a look to see what could be there.
Let's look inside.
Somebody on top.
Yeah, it looks like it, doesn't it?
Yep.
Tenant?
Yeah, somebody, the white spot. Oh, there's a few. Great Zoom, gentlemen. Thank you.
Awesome. If you see the two line, there's like each one of those little colonial animals has two lines flanking the mouth, which is sucking water in. They're luminescent organs, which supposedly are bacterial luminescence.
Oh. What do they use that for?
These are really bright. They're called fire bodies in English, their common name. And if you, that big hole on the right hand side right now, that's where all the water is expelled. They can actually swim really fast, even though they don't open and close their body like a regular jellyfish does.
Mm-hmm.
They just shoot along. It'd be a really good design for an underwater vehicle with all these tiny little pumps pumping water in, and then one exit where all the water comes flowing out.
Almost like a Venturi or something.
It'd be a,
Mm.
it'd be a,
It's not even pumps in this animal, it's all ciliary.
it's all silly.
It's cilia.
Yeah, right.
Right.
But you couldn't do the ciliary in the robot.
Cool.
You do
Everybody
pumps.
happy with this?
No, we get the miniaturization.
and
We're
Nice.
happy. We can move on. Thank you, team. Moving on down. We're at 500 meters. Thank you.
And
Crustacean.
if you pour beer in the opening, the whole animal brightly lights up, and the beer tastes slightly salty.
Oh my god, dude.
The adult
Just
fun
beer
fact
or
for the
any
day.
liquid will work?
So you've done
God.
this? I've done that, yes.
Oh wow.
Just went up
I had a
there.
couple of krill to chomp on, and then a beer poured into a pyrosome, and it was my midnight snack.
I think you missed us last tell.
How many centimeters long is that?
This particular one's probably only like 3.
Right. So not much beer then.
No, well, depending on the species, some of them are big enough that a scuba diver can swim inside and touch the inside with their hand.
Look at online, there's some really cool photos.
Fish.
Fish? What?
Yeah.
Oh, cyclophonic?
It's a cyclothony.
Oh, I do this so fast.
They're
There's some
swimming.
good photos
Yeah.
online
The
of
way they swim is
pyrosomes
pretty.
being eaten by leatherback turtles. And they're bigger than the turtle. Bottom right,
Yep, middle.
2/3 across.
Yep, this guy.
Another
Looks like a looking on a teen of 4.
It could be, or it could be a halicreated, it looks like.
Oh,
it
maybe.
looks like Halicreus minimum,
Yeah,
maybe.
yeah,
Go
I take back my teen of 4
slowly zoom in.
observation from afar.
The distal tips of the tentacles are bright white, proximally more transparent. It's got a pointy bell.
Halacria?
Halicreus minimum. We should be able to see on either side, like the corners of the bell, there should be little spiky projections if we can get the contrasty enough.
Yeah, you can see him now.
You can see him now.
There you go.
He is. This is the tall hat morph. Halicreas. It's a Trachymedusa. Nice tight zoom.
Where would I find this under?
Tricky Medusa.
Yep.
Yep.
Right now he's been
Go
a
up
tricky
to
Medusa.
Oh.
We have some Medusa options for you. Tricky. Yep.
Top right.
Top right.
Yep. Say goodbye to Halicreus.
This guy right here.
We've got a hippopodid colony.
I thought you were
see
done
which
with
one it
siphonophores.
Yeah.
Never.
We're just gonna take a
I'm only
squeeby
kidding.
look.
Never.
Nor should we.
You want both of them in?
And that would be
in the
lovely.
frame
Get both
at
of them in the same frame.
time.
Thank you. If we could have
Six
all
of
the
both
gelatinous
together.
organisms hang out together.
The Vogtia, it looks like it might be Vogtia serrata. Does it have lots of little spines, or is it smooth?
On the main part.
Yeah.
Yep. This
That's
guy
we
here.
need to get to that
We
zoomed
need to
in
be
if
really
possible.
zoomed in, yeah.
Forward all the time.
Yep, yep.
Stretch towards you there.
Yep, he sure does. Tentacle towards you. We go behind him.
It looks like serrata.
We're working on it, Dougal. Stand by.
You're gonna have to pay me out a little.
And grills.
Will you thousand?
It's got the
Oh, yeah. If we could do this, that would be
morphology
great.
From Alvarino's paper. Kindly synonymized. Here we go. Now it's okay. Okay, that's good. I got the frame there.
You're not seeing all the little green spikes just.
Yeah, so it's Vogtia serrata. It doesn't have all those little
Dougal,
spines
you're
on
good?
it. Yep, I'm good.
Okay,
Thank you.
we can continue moving on. Thank you.
Roadkill.
What's the other one? Serrata and
Spinosa.
Spinel.
Spinosa's got the spines.
I was thinking this is serrated.
Localize some of these guys for
I was
Kakani.
just making sure my iris is open so it's not minimum noisy, you know? It's a bit noisy today.
Do we finally get rid of the siphonophore tentacle on the right?
I think we did.
Awesome.
Finally. Finally got rid of that
It's
thing.
been a while. There's still some on the top left. Yep. Oh yeah, you're good.
Right?
We're fine.
Yeah.
Thank you for that.
Nope. Nope. We are good.
A little bit more forward if possible.
All right, a little more forward.
Yeah.
Right in the middle, the coelodendron. Looks to be only
Right here?
one.
Middle. Is this the
Oh,
one you wanted,
well,
Dougal?
let's take a look at this. What
Yeah.
on earth is... It's caught something.
No. Yeah. Something is. It's not
It's a
another one. It's not-
coelodendron that's caught a colodarian or something.
Yeah, there's a colodarian, the colonial.
It's one of those we've been seeing, but it's caught something.
It's caught
Correct.
something, yeah.
And they're not together in a pair.
It's caught a milky
I think we
crayon.
should get this.
It's not in a pair though.
Right,
You
not in a
sure?
pair.
Think you want one
Yeah.
that's in a pair? We'll see heaps of them. There'll be heaps.
Heaps.
Do you want the second thing that it's attached
Yeah, I think if it's a colonial, Zulano, we
What is
have
that,
so
egg?
many of
Is it an egg casing?
No, it's another colodarian.
Okay.
It's
Does look like.
A lot of little dots.
to? What's it doing
Yeah,
with it?
it's
It's
like
not
egg.
actually.
no, it's
Oh, it's like holding it.
It
It's
looks like
probably
eggs
eating
to
on
me.
it. Yeah,
Saying.
I reckon it's eating it. Because it's not actually caught on anything.
they're very sticky. All of them.
Let's see if there's anything coming out of that central spherical body touching on the thing that it seems to be holding. Holding it in place with its parapodia.
Yeah, see, you can see the spines and the central capsule connected. Right there. This is beautiful.
I don't see it.
No, it's not.
No,
Yeah, it's
I think
just
it's like
kinda
it's either
stuck on the
holding and
inside
stuck
there,
on it.
Yeah. Pretty cool.
Are we good with an image, or are we going to collect this?
Your
We've already
call,
used
Manu.
3 buckets, and we have not hit to
Have we
1,200 yet.
seen any pairs today?
Not yet.
We will see.
We will, though,
pairs.
we're not.
Maybe
We're
we're
only
not
at 500,
yet.
right?
Yeah, I think it was 800 is mine.
Yeah,
Okay,
that's where we
let's
had them
leave it
before.
then, and we'll try and get a pair.
Okay, we can keep moving
N
on. We're
equals
gonna look
2
for
is
that, but in a
always better than
pair.
N equals 1, right?
Yeah.
Looking
So-
for a pair.
No, it'll resolve the mystery whether they're cytoplasmically connected. Because it doesn't make sense that they would find each other.
No,
So
they can't.
Sweet.
it is kind of sweet. We haven't
Against
done
all
that.
odds, they found
I'm so excited about this paper because just for the title, we did a male and female diatoms in the gravity machine. We call it Falling in Love
Middle.
because they use gravity to actually find each other.
See what this
We can
is.
mix
What's
them.
that in the middle?
That's
It's
what
a-
I'm stopping on.
It looks to be a praying siphonophore of some kind.
Yep.
It's insane.
Oh, there's
Like,
somebody
they
that just went
use,
away from it.
the
Oh, no,
Stoic
it's-
drag.
It's got 2 swimming
It's
bells
got
on the
2
right
swimming
hand
They
side.
bells.
have
Yeah.
no appendages.
We need to zoom in on those and see
Yeah.
what the internal canal structure is.
Very cool.
Diatoms. Yeah. The title is going to be Falling in Love,
What?
colon, call it whatever else. But it's a little bit. What is the red?
Definitely is something.
It's got fluorescent red
Red,
does.
tentacula tip.
right?
Yes,
Oh,
it does.
Yeah.
And
Swimming bells look different somehow.
yeah, we need to zoom
If
in
we
on
can
those
get the
swimming
big
bells.
body up there when you get a sec, or
And
as
where
it
that
works,
bright white
as it
spot
agrees
is
with you.
in the center, is there stuff coming out from there or not?
If it agrees, there you go. Didn't we see something similar yesterday, but not without the feeding tentacles?
Yeah, it's the same subfamily yesterday
Okay.
we caught. Still can't quite see that.
Those feeding tips are great.
I can't see any somatocyst intrusions into the mesoglea.
Uh-uh. Huh.
Ah,
Dougal?
can we suck him up?
Even though you said no more siphonophores,
Yeah, well,
I feel like you're taking it. We're gonna
How
suck
big
this
is
guy
this?
up. He is small, I think. That was
Quite
pretty far
small.
away.
Yeah, no.
I think he's small, is my guess.
yeah, he's pretty small, I think.
Yeah.
Which jaw do you want to?
The jar it's on now would be
2
fine.
is fine. Here he is. Middle.
What type of siphonophore?
It's a praying siphonophore. I'll look offline now.
Got him.
What's the temperature today?
It could be
We're
a
at 6.49 degrees right now.
You're right.
Put it on the mids or the uppers. He just kind of disappears when he gets
Oh, yeah. Good call.
Oh, that's a cell.
Right. What's the closer?
Correct. There's a cell there.
Yep. Right. Just like.
Yep. Pretty sure we don't have a sequence for this spraying yet.
Yeah, I think this
Or
is
at
needed
least-
to be.
I don't have a sequence. Oh, nice.
Okay, kill it.
Good stuff. Oh, did he just go in? Flying ahead. When he gets in there.
There he is. Yep. Oh, he just went on the outside.
The bone shot.
Mm,
Did you see him going?
no,
Back quick.
he's way up on the.
Pulse. 1, 2 clicks. 1, there's 2.
Uh-oh.
Go for it.
Still haven't come through.
No, he didn't go in.
You didn't go in?
Well,
I don't think
the
so.
siphonophore did.
Okay.
I saw that one going, but the second.
Oh, okay. Has he gotten in the canister yet?
No. Another pulse, maybe?
You want another pulse on the suction?
Yep.
If you guys
How
could
hard?
pulse.
Like half?
Sure.
Half. Off.
And another?
I got nothing yet in the canister. Oh, maybe something's coming in. I haven't seen it come in.
Might not have gone in the field.
I don't think it didn't. I don't know that it did.
So maybe let's keep it on 2 for the next sampling.
Yeah. Sounds good. 2 with a question mark.
So it might come through, it might not.
And it was a what?
Are they pretty sticky?
I just said siphonophore.
Baby. Okay. Dougal, we're good?
Yep,
Okay.
thank you.
Moving on, my team. Nice job.
Copy.
See if we can get 10 meters without an awesome siphonophore or ctenophore. Might be difficult today. Might be difficult. There's one there.
We're
Bye-bye,
the-
Medusa. You
Where are the
didn't see it.
cranchids? Come on. Rosaria?
Yeah, it's a Rhizaria in the middle, right? Are we stopping for these? Quick zoom and go?
Yep, quick zoom and go.
Okay. Quick zoom and go on the guy in the middle, please.
Copy. Zoom in on that.
So was this siphonophore praying, maybe?
Mm-hmm.
Nice shot.
The
That's
last,
the one we saw yesterday too.
Yeah,
man.
Prayed, right?
the last one.
Oh.
The siphonophore.
Oh, that's what you're telling me. Got you. Thanks.
Mm.
Yeah. It's Rhizaria. Okay.
Okay.
Thanks,
Thank you.
team. We're good. Yeah.
Okay, wait.
No. We got some
We
fabulous
got
zoom.
We got some good shots. Yeah.
This is not the foram or anything
Oh,
we're
I know.
looking at.
It's a different one.
This
Yeah.
is
Do you
most
want us to
likely
stay for a minute?
cytocladus.
We
We
got
should
some
just
good-
at least get a zoom.
We did.
We did.
We got zoom, video, and still.
But you were saying it's at least
This is not this is
Can you zoom on
It's in
the
the
Rhizarian again,
Thalassinidae.
please? We're coming in, Manu.
yeah, see the spines don't actually wobble, and you can see that it doesn't have the core has 4 capsules.
it's crazy.
You're gonna suck it on top of the.
Why don't we
Prayed
suck
we
it on
just tried?
top actually? It'll be totally fine. You
On
think
top,
it would
in number
remove
2,
Yeah, in the same one we
Okay.
just
Yeah.
sucked on.
if
This
you
will be
guys
totally
can
fine.
go for this for number 2
Is
again.
there a risk of losing what
What-
you have in there?
Can you spell what you think it is?
I think my intuition is this is a
This guy, right?
cytocladus. Cytocladus. Cytocladus.
DUS.
DUS. Cytocladus. And this is the guy I told you after a million liters of water I have 1 cell.
Oh, really?
Yeah, in the Mount Ness. I have it live.
more light.
These four
You got him.
It's going in.
You
Yes,
got
you got in.
Nicely
Okay,
done.
wait,
Let's see him if he goes into
Number two.
number 2 is the goal.
Suction's off, and I'm just flying forward.
I didn't see it come in.
Not yet. It's still going. Give it 10 more seconds.
It'll take a minute.
It's a long transit.
Yeah.
Yeah, it's got to get through the shadow graph, right?
Yeah.
And all the tentacles and whatnot.
Right.
Maybe it's just stuck in there. It didn't show up still.
No,
It
it's
takes
still
a
going.
little while.
Takes a while.
What else is in
There's
there?
something in there.
Is there a
There may be a
siphonophore?
braided siphonophore in there. But there
Yeah,
may not.
that.
I don't know anything that needs something like But how come if there was water going through, the sheet still be moving?
Here it
Here he
is.
is.
We got it. We got it.
We got it.
Yep. You can change
Oh,
canisters.
That's fantastic.
Yep. He's down low. Change canister.
It's a lot bigger than I thought.
No, there's something
The
else
next-
stuck on it. No,
This is true.
this size
Yeah.
is
Perfect.
actually the
One.
original
Yeah, one's
size.
available.
So they become like a little grape.
Yep.
How far they are then?
Sampled in
Correct.
SS2.
Just to let you guys know, we're 500 meters still, and we only have. Oh, we have
There's
6.
another
We have the
Rexarian. You can just
decouplers
mark
as well.
it. It's
So we
right
have
in the
4
center.
containers
Dead
and
center.
2 decouplers. Okay,
So Heather,
we're moving down.
we're trying to get the samples while the shadow cam battery is still running.
Nicely.
Yeah.
Nice idea.
Probably it'll be stopping
Another.
soon. So
Yep, got it. Makes sense.
so
Gotcha.
we don't have to worry so much about it after that. Like
Come on.
when-
Got it. And you win. And you just win. It's perfect. It's good. I like it.
No,
All
but
right.
if you don't suck anything through it, then the shadow graph is not
And we're still on
collecting any
gentle
data, right?
suction.
Yeah,
Yep.
You have to capture.
We can, yeah, we need it
Because
back
we
on
don't
for
have
the
much of a
shot.
charge
You
in
want
between
the
dives.
suction on gentle all the way down still, yes?
It's probably run out of battery. by
Oh,
now.
we don't.
Yeah.
So you want it off. You tell me what you want.
That's fine. If we have it on, you'll catch your little crustaceans and things for your experiments. So maybe on is better.
Okay, that'd be great.
On is better
Gentle
also
on
because
would
we
be
now
great.
have
Gentle on.
fine mesh
A gentle
on all
on.
of
Yeah.
them.
Yeah.
Gentle on.
So a gentle on is always good.
Okay.
But you know, it's ironic.
That's
It's
where we're
been-
at.
I mean, last couple of days, I haven't seen any of the small stuff getting stuck in.
Nobody's really looked through the
I
suction
did.
samples.
I
all
do.
that. You do?
Every
Yeah.
day,
Okay.
yeah. Before I throw them out, I look through the small stuff. No, but the mesh, today is the first day we put the right mesh.
What's
It's
that?
possible.
Why do I not? Oh, there it
The
is.
Nacellarian jelly.
Bigger than that.
Yeah.
Below.
Oh, come
Let's,
up below.
yeah, that's what we want. Yep. Let's see who we have. Ctenophore? Looks like a ctenophore.
Is it a big Thalassocalus, maybe?
No, it doesn't. Where are the cold plates? No.
What is this?
Oh,
I
there
think
is
it's
the
a, yep,
cold
it's a
There it is.
Thalassocalus. Yeah.
There it is. It was looking at the position. Nice zoom, pilots.
What are those that look like combs but are not combs?
They're the gonads.
Gonads.
Yeah.
The 4 on the side.
4 of the canals have gonads in them.
And this is lobated. It's folded.
Well, it's yeah, it's folded together.
It's okay.
Thalas-
Do you have a species, Dougal?
Thalassocalus inconstans.
Elassicalus.
THALASSOCALYCE,
Yep.
as in the cup of the sea.
Yep.
Inconstans.
Inconstant.
And you can see the eggs
Nice
are towards the center, those
Yep.
rounded things, and the other side of the meridional canals in the diverticula are the sperm.
Yep. Nicely done. Are we good with this?
Yep, we're good.
All right. Thank you. We're moving down. We've made it 7 meters.
Another coelodendron.
Ah.
It's gonna be a long.
We've got this, team. We can do this. There's lots of good stuff in the ocean. Cyclothony.
Cyclothony.
Bye-bye, Cyclothony. Stop.
Oh, stop! Is that a squid?
It might be a squid. Ooh is right. See how calm I am? Like that noted for the record.
We don't need to look at it if it's a squid, right?
It might
What?
not-
Oh no, it's a molt.
Yeah, I was
It's
gonna say
another-
it's not a squid.
With the what?
It's another shrimp.
It's a molt of a shrimp or
that
something.
shed its.
That's a stalked
carapace,
eye going down, and it's his tail going off to the right.
Oh, it's a phylosoma.
No, it
it's
is
a lobster, phylosome.
larvae. That's exactly what it is.
Is
So
it
this-
dead?
It's awfully deep.
It's very deep.
I think it's dead.
I
and
think it
sinking.
might not be alive. Right.
Well,
And he
you'll
only has
find
one
out if you
eye,
blow
from what
it,
I
go
can see.
into it, it'll move.
So, okay. So this
Yeah.
is a great... Oh, no, he's got both eyes there. I don't see him moving on his own, though.
It's just kind of flying.
Right. I think... Okay, so this is a lobster larvae that's usually found up near the surface. 500 meters is a little deep, we think, to find these.
It'd be nice to
you
see if
just
it.
need to disturb it, see if it moves.
Yeah, because it might be heading to the benthos, and it might be a deep sea lobster too, right?
True. If we could get another zoom on this. Yep.
I don't know what the scientific name of lobster is for my annotation. I'll look up
Homerus.
Panuliridae, and then I'll.
Well, Homerus is the...
No,
Tunnel
we
up.
know this. Yeah.
I'm
Pleocyamata.
gonna say we have a phylosoma.
Phylosoma,
Oh
yes.
my goodness.
But he's
I'll
falling. We
make it
believe he is a
Pelagic.
dead.
Oh,
Larvae.
Yes, there's one of your protists stuck on the
Oh, there
end
is
of
a
it.
protist stuck on his leg.
And you can see the fans are not moving yet.
Right, yeah, I don't think this guy made it.
This
But
is
there is
hilarious.
silver lining. He is going to become part of the marine snow that continues to fall down to the seafloor, providing nutrients along the way for other organisms in the deep sea.
And carbon sequestration.
Exactly. He can sequester some carbon, move it down into the ocean.
Probably even collecting microplastic particles on his way down.
Nice job. And the scientific jokes ensue.
The image some phylosomas in
That's
the lab
great.
last night.
The,
Yeah.
the tissue is still pretty intact.
We have caught some alive
It's a little
in the
opaque.
nets.
I mean, they're normally
two
completely
live.
transparent.
Yeah, we've got plenty. I don't think we need to collect this, but some video would be great, and then we'll move along.
What I would want to see is why make such a flat body.
So he's
Talking about this, that doesn't make much sense. So
yeah, it
let's see
does.
move in
It's
the
a
water.
surface area. You want surface to volume ratio, right? So I'm not gonna
Yeah,
sink.
passively you can drag along.
But how much is he gonna move against a current? He's not. Those little legs aren't gonna do much.
All of these
It's in
things
pretty good
move
nick.
a ton.
If it's sunk for like
Yeah.
400 meters.
It's
or whatever.
yeah. I was gonna say he's usually collected in what the top
Nothing's
50 to
eating it
100
on the way
meters.
down, right?
All right. If we get one more on this, we're okay after that.
Yeah, I'd still like to knock it into something to see whether it starts to swim.
Okay.
It looks dead,
Pilots,
but
I leave that to you to tappy tap lightly onto some piece of something.
The science book,
Sure.
It's gonna be laser.
the old science book.
It's touching the cameras. Yep, we have touch camera.
and it's not doing anything.
No, it doesn't look like it responded from what I saw.
So it is dead.
Yeah,
Okay.
I'm going with a.
It's good to know.
Yep. Okay.
Someone's got to write a paper about the contribution of dead phylosome larvae. to-
Yes, this could be a thing. All right, we're gonna head on down. We've made it yet another 7 meters, which is still great.
It might just be planning
I
for the
know.
fjords, Russ said.
You're joking.
No.
Look at that depth trap.
That's the phylosoma we got in the lab.
Yeah.
The mosaic,
We're gonna get
but
there, team.
if
We're
we
gonna
keep
get down
zooming
there.
in, zooming in, I have the
I think it was my
entire
goal
view.
last night. By the end of my
Every
shift, we
one
should
of
be down
this
at the
is
bottom.
Like a 20x mag.
Oh, wow, okay.
Yeah, so all
Oh, yeah,
the muscles,
should be.
all the tissue. That's why I want to see it swim. Like, that's the resolution that zoom is at.
Okay. Cool.
remarkable to kind of do histology without killing anything.
Right. Yeah, because they're so transparent.
Exactly. And so flat, so it's perfect for imaging. But you can still see it swim.
Thanks.
They have them in.
I found the paper you mentioned them riding the jellies, but to me that doesn't explain the flatness. Something else is going on.
Nope.
Of the what?
Yeah, I think
The
when you
phylosomes?
passively sink, phylosomes.
Halicreatid.
Right.
Oh, to the right.
See what it is. It looks like it might be Halascaraconica.
If we could get some zoom, see how we're looking. It literally is almost every 7 meters. Oh.
We're making great.
Maybe there's a
time I go to
pattern.
the
Maybe
lab
there's a set
thinking I'm
pattern
gonna
that
go
these.
finish
Maybe
the
we're
work
finding
I
a new
need to
pattern for
do.
gelatinous
And
organisms.
then I
Yeah.
They
just
need their
come back.
territory of 7 meters around them or something.
Maybe this is the zone that we just needed to be in.
Is it between 400 and 600? Yeah, it probably is.
But we don't know until
Yeah, he is small.
we do a survey.
Nope, nope, nope. We're
We
not
should
doing
just
that.
go
If
all
we
the
can
way
get a
down
zoom,
with
do you want to
the
zoom on
cameras.
this?
Capped
Just want
off.
it in focus. and
Yeah,
then
yeah.
where
If
it's
we can get him in
gotta be.
focus for a
I
second
mean,
or two.
technically, that's
What
the right way.
type is it?
Because then we rush up.
I think it's
By the time it's
Halascaraconica,
night,
a kind of
it's
Trachymedusa.
very
Okay.
dense.
Yeah, do what you gotta do.
They're all
Oh, he
tired,
just
yeah,
dropped.
like the night now we're fresh.
It's right in the center
The
now.
dive
That's
should
in
be
there.
really like a dive.
Just leave them all on for the
That's still a
Yep.
sinking Phyllosome.
Oh, it's the same one.
Yep,
No?
yep.
Yeah, that's the same.
creeping ahead.
Unless all of them
Coming
just
on
die.
in. There he is. Almost.
And an activity
Working
as
on
well.
focus. At this rate, the lobster larva is gonna beat us down there.
Yep, Halascaraconica.
There you go. Halascaraconica has been the ID for this Medusa. Fabulous. Nice job. Thank you.
Thank
And we
you.
can move down.
Another 7 meters, please.
I would love to go 14. I know I'm going bold.
Gonna fight for every meter.
I'm going bold, but I'm trying for 14 meters. See? He's still falling.
That's Mr. Lobster.
Yes.
You can get some sinking lobster speeds
We
here.
could get sinking speed.
Yeah.
Paper there. How fast
There
is
is-
our speed? Apparently.
Yeah, it is the speed of the ROV, I think, is
But
where
it's
we're at.
the extra drag from the Rhizarian you'll have to factor in.
Yep, good point. Mm-hmm. Possible density differences as it gets deeper.
Are measuring that.
We are.
So if we dropped a hammer and the Phyllosome at the same time,
I think it's nailed to the perch. Here we go, Calicophora. A few siphonophores there. Oh!
Oh,
Something
that was a
just-
squid.
Ah, I think it was a shrimp.
But
and
it
it was
ain't.
bioluminescent.
Oh, maybe it was a bioluminescent.
liquid.
It did swim like a shrimp.
Yeah.
You're right,
Yeah, it could have been one of the bioluminescent shrimp. Isn't that a Canthophyra?
Yeah, or an Oplophorid or a...
Aquaphorid,
yeah.
right. Yep, there's a few of those groups. Go with shrimp, and then you could put, go to the shrimp options. Just go shrimp, and then just write bioluminescent.
Has it been 7 meters yet? Go a tad more forward as we're descending, if possible.
What was that? A little more forward?
Yeah.
Yeah. We could go a little bit more forward.
one more forward.
Speed's good. A little forward would be great.
Got a still misses?
Oh,
No?
stop.
No, something else.
No. that was-
Halicreatid, maybe.
Sorry. My bad.
Keep going.
We stopped?
Yep, keep going.
Okay, we're good. We can keep going. Is that mixed messages at all, gentlemen? That was really clear, right? That was super clear for
I'll
you?
tell you a funny story about that later. It's not really control for influence.
Did you get your
Mm-mm.
7 meters?
I'm not telling you.
I was told I had to go 14 before I could stop again.
Yep, we have, believe it or not, gone 14 more meters.
That means I can stop at any
Not
time.
yet. Another 14, Dougal. Another 14. Not even halfway. We're supposed to be at 1200 by. Oh, we have some time still. All right.
Oh, here we go.
Yep,
Something.
middle.
I refuse to be constrained by.
Nope, you should not be constrained. I said stop as well and put the pointer out.
Oh,
Yeah, but some
nice.
horses can't be tamed, you know?
That's right. That's right, Dougal. We got you.
It's a collapsed.
It looks like a
mucal
house.
structure of some kind.
I don't see anybody in there, do you?
No.
No.
Must be a bad neighborhood.
Yeah.
We've seen several of these. They don't, they're disc-like, but they don't have any structure.
No, yeah, you're right. Does that mean they may have been falling for longer?
Well, no, a house normally kind of parachutes more down and it
Yeah.
consolidates and gets some of these
It's got a
weird
double layer.
vertically
hasn't
stretched structures.
Yep.
So, this is kind of weird because it's like a dead
it? Yeah.
Thalassachilus or something. It's just what's left decaying.
Yeah.
Oh, and maybe
Cool.
smaller part.
Okay. Moving on. Hmm.
Fish.
Fish. We can get a zoom on him for quick,
Oh,
just because
It's a
we've seen a bunch.
cyclothone.
Yeah, it's a cyclothony, but we haven't zoomed in on one in a bit, so we'll give those vertebrate folks a little
Okay.
quick zoom on a fish. And oh, he apparently just wants to come say hello.
Hello.
All right, well, that was a nice dream. Okay. Oh wait.
Yeah, a key grab over
He's
here.
doing the
All
sandbar.
right. What do you think?
Yeah.
If he settles enough, I don't want to spend too much time, but if he settles enough,
It's a cyclophone.
okay, maybe. Maybe not.
Cyclophone.
We're okay. He's a cyclothony. We'll see more.
Cyclophone.
Continue on, my friends. Thanks.
Oh, siphonophore.
That was a siphonophore.
That was a praying siphonophore.
You're good. Dougal didn't scream at us, so
I didn't scream enough.
Yep.
Yeah,
Is that what he'll do?
yeah, possibly.
I will.
You'll hear it. It just sounds more like you'll hear. You'll be able to tell. It's not terrifying.
That you're like a screaming goat.
Oink.
Not screaming.
That was a hiccup. Cryptopod.
Yeah. Say amphipod on that?
Maybe.
Or a copepod? It was pretty big. If it was a copepod.
Oink. Lots of little crusties. Copepod.
Copepod, I think. Look at this guy. We could zoom in on the middle guy real quick. I think it's a copepod, right?
Yep.
Yes, it's a goat.
Okay, we can keep going.
Keep going?
Yep, we love the copepods. They always get their day in the sun. Extremely important organism. We're zooming on.
Straight ahead in the middle. Is that a oik or something else?
Oh yeah, down low middle.
An octopodema maybe?
No, another halibut.
I'm not sure. I think it's an octopodema. A little bit closer.
Cyclothony, I saw the chat real quick, is spelled
Octopodema.
Cyclothone. So you guys are close. Dougal, what do you think on this one?
It's an octopodema. ARCTOPODEMA.
Arctopodema.
See, it's shedding its tentacles there.
Oh yeah.
I think it probably does that as a defense mechanism to get them caught in the gill raker in the gills
Yeah.
of fish.
Of whoever's trying to eat them.
Thank you.
Great.
Cool.
Thank you. Moving on.
coelodendron.
Bless you. Again. Again.
Be alert to those things. In the middle. Yep.
Middle top.
That's that. Yep. Is it salps or
This looks like salp chain. Doliolids.
Doliolids.
I like doliolids.
Looks doliolidy, doesn't
Uh-huh. Agreed.
Totally
it? Let's
opposite.
look closer.
Like
Doliopsid,
umbrella.
is it?
Oh,
Yeah,
nice.
it's like
That is great
Doliol.
zoom.
Oh, nice.
Great
What
zoom.
is this?
They're all joined.
They're-
Oh,
it's a-
it's shed. It's like
They
the.
look like an IUD.
No, it's a piece of stolen along along it. We
Wow.
still don't know how they really do it.
Let's look at this for a
I'm
little while.
sorry.
Doliol.
So doliolids, yes?
Doliolidae.
Can we get
Yeah, I
a,
don't think we can get
can
much.
we get a scan of
Oh
It's-
yeah,
it's
we could try a scan
consistent.
on these.
Yeah, that's
Yeah.
like
Yeah,
a
yeah.
scan.
I would like to
If
get a scan.
what type of scan would you
Kinda
like, my
small.
friend?
A
but-
DPIV one.
DPIV, please. That would be great.
Yeah, this is pretty.
Yeah,
cool.
these guys are pretty cool.
I have no idea how fragile
You have the
color
right-
is.
All right, we're okay to put the lights like we did for the, yesterday for the scans, right?
No, so it's the laser one?
Yep.
Brand is the laser one?
Right, but so remember yesterday we had the minimal light so we could still kind of see them, right?
Yes,
That's
and
what we'll do
then
that
just
again?
turn them off right before you get to the.
Like just before it's that
All
flash
the-
stop.
All the lights off this time, or like we were doing yesterday?
All lights when it's at the shoot. But right now.
We need the
I
light to find them.
recommend Aligning it in that camera.
Go ahead.
So just play
Where did he go?
Why is my feed flickering?
And then if I can run a data task here, which is log deployment annotations, and this now polls in real time. It's put on the database. And
I saw
it
it
goes
too.
to
I'm
try
not
and
seeing it
log
all the
files
time.
then.
It's kinda
After a
random.
while, it's
Flickering on which screen?
Her screen.
after I activate it. And then
See it? Every once in a while it's a blip.
Yeah.
Yeah.
file.
It's
Do we-
happening just on
And
the
then when
feed
you
in
click
general.
on
Do we see the doliolids?
real-time annotation, it'll take
Wait.
the video to play it.
No.
So you can the original video.
Escape really
I
well.
have not seen that.
I think we lost the doliolids.
You see that's in the database.
Yeah.
It's pulled from the database.
Unless there's a. This might be a.
And so that's our doliol.
That might be a.
So
That's a
if I
Tina
go to
4.
the videos file, if I point
You
at
guys, what's
videos
in the middle of the
file,
screen right now is not the doliolids, correct? That's a Tina 4?
That's correct. That's it looks
I
like a
think we lost.
Bathuroi maybe?
I think we lost the the doliolids.
May as well scan the Bathuroi while it's right in front.
Well, while this guy's here,
Tina 4?
this is a Tina 4.
What type?
Low bait.
Haven't had it in focus zoom yet, so not sure.
He's super close, I
I'd
think,
say
to
it's a
to us.
Bathuroi. It's got a darkly pigmented gut.
Okay.
I think it's
Yeah,
right
he's caught a little
inside,
suction stuff. Okay.
Yeah. Okay.
Should we
Keep
cut
going.
cameras and keep back to go?
Yep.
Sorry we lost those doliolids. How they can escape in this crazy environment. Crustacean. Do you want us to keep your cameras on, Connie,
Yeah.
while you're looking at this?
Yep.
Grill.
Yep, we've got Falzid over on the left. No. No. He's gonna say yes.
Oik.
This guy. Oh, that's what it is? Middle?
No, it's
This
a
looks
doliolid,
different.
maybe.
This looks different. I'm going doliolid.
So,
Yeah, but this is a solitary.
Yeah.
my Taurus monitor is
Yeah.
The deep thinkers, the ones
Yeah.
If we can get some good zooms on this would be great.
Oh, look at
Yeah.
the red spots on it.
I know, what's that all about?
Plugged into that wall there with that one? Yep. Yeah, go ahead and unplug that and then slap it back in. We'll see what happens. Otherwise, I can reboot that either.
Working on the zoom.
All right, still jumping. I'll just reboot the gibbets. That's good. That's good to have that to compare.
Yeah.
There's certainly a lot happening at this depth, isn't there?
Yeah, there is a lot. And it's below the deep scattering.
Wow, it
Deep
looks
scattering
like,
is up at 250 at right this
Right.
second.
It looks like the Dahliolid has eyes.
It does. All right, this.
Just focus, focus.
All right, let's see if we can get this guy. Is he getting too close?
It's a big jelly there,
Where are you at? Oh, yeah. He's not on my screen yet. Wait till you see this, Dougal. Okay.
Nice one.
I think we're okay on Doliolid, yeah?
Yep, yep.
Let's go to that jelly before Dougal has
What jelly? What jelly? What jelly? What jelly?
Yep, if we can go to that jelly.
Say jelly.
I don't see him on my camera yet, but I see him on yours.
I know you're struggling.
Oh, there he is.
Halocreatid. Let's take a looky. Take a looky.
It's
looky.
very pretty.
Ooh.
This guy's awesome.
Yeah.
What do you got going on, Dougal? What you got for us?
You gotta come up a little.
Oh, stand by, Dougal. There's a lot of marine snow in here, too.
Down.
What is that?
You pay me out a little. Come on, show me your gonads.
You really say that on the street?
Great at parties.
This would be awesome if it's. There you go. Almost, almost. There it is. Almost. If only the water would stop moving. Or the ship. There you go. That's great, you guys.
It's a Haloscera.
Haloscera Medusa.
Which one? I'm still deciding.
See if we get that
Why
zoom.
do they have to create two
I know,
genus
I know
names
you are.
that so close together?
Thanks, you
Oh
guys.
yeah, there's an eagle like that.
There you go, Dougal. How's that?
It's really
That's a
just to
good
confuse
shot.
those
You've got some gonads. I see some gonads there, Dougal.
Oh.
6, 7,
Should be 8.
8. Yep, I was just counting around.
Is there like a big egg in the middle of each? Or?
I can tell you. Oh, he just changed position. Stand by.
Oh, and for folks following along at home, we're running into some video here. There we go. I got you back some.
Okay, what did you just ask me? Look at right here. There's a definite
So I might need to reboot our system at some point. I'll let you know if that's going to happen. But we're working on video here.
Photos that are gonna be going into Fat
Okay.
Very
Dougal,
nice.
you good?
Yep, we're good.
Okay.
Oh, is he going in the D sample? Look at that. No, he's going in the suction sample. Look at
No,
that.
no, he's not.
No, he is.
No.
No, he is. Look at that. Look at that. Look at that. There he goes. He's going in. Look at that.
Opportunistic suction. All right, thanks, pilots.
Put in the D, and if we find something better, we can.
Yeah, that's true.
We said that yesterday, and that didn't go well.
I'm just
throwing it out there.
It was opportunistic.
Just saying.
It was opportunist.
Just saying.
All right, we're moving on, my friend.
Moving it down.
Yes, please.
Yeah. You may make 550 soon.
Hey, don't be so optimistic, Russ.
South.
Covering some distance with this current.
That works. I was gonna say, how is the current? Pushing at us? We going into it at us?
Check my copy process again. Be back soon.
20 minutes away from dinner.
Yep, got it.
I mean, we could try a different HDMI cable, but it's weird that it just.
Yeah, that's the one we were using yesterday, though, right? Yeah. Just slap this.
I can move if you need me to.
All right, good.
Okay. It's nothing now.
Slept through lunch.
You slept through lunch.
It was
We
delicious.
almost didn't make the dive.
Yeah.
Jittery for
Why
us.
didn't anyone come and get me before we went to go?
Well,
Are you serious?
you had
There
the
were like
illusion.
2 people that were like banging on your door.
Yeah.
Did you not know?
I think Karen was the only one who
And
banged
Sarah.
strongly enough that
Apparently
I.
Sarah did.
Middle, right?
Yeah.
Come on
We
in.
were on the-
Okay,
Yeah,
down there we go.
both.
Just coming in
is that a
lower right.
heteroparamus on the top in the middle?
We're going top, middle,
Yes.
and then
Top,
there's a
middle,
jelly
top,
below
middle,
it.
yeah.
Yep, top, middle's more important at this point.
And then if we're lucky, we can drop down and
We're
get
lucky
the
we can
second
drop
one.
down.
Yep,
but that's
agreed,
the
Russ. I
first
gotcha.
one. Oh, maybe it's a cunifees.
Oh, who's this guy?
Oh, that's cunifees multidentata.
Yeah,
Do
what
you
does
want
that
me to
mean?
annotate that?
What does that mean
Did
for
you-
the
can
rest
you
of us?
capture one?
Yeah.
It's a Chalcophoran siphonophore.
Thank you.
Yep,
A
there we go.
Calicophoran siphonophore, my
Yeah,
friends.
family Clausifiidae. It's got these ridges
Very
running
cool.
along the nectaphore,
Yep, I
which
see that.
bifurcate up laterally.
Yep.
It's
Cool.
looking a bit rough around the edges. I
Yeah, I
think
was
it's
gonna
had
say, shouldn't there be
it's had a
feeding
hard night
tentacles?
out, I reckon.
Are there feeding tentacles on these?
It's retracted.
Right,
they're
they're retracted
retracted,
right
yeah.
now? Okay.
Cunifes.
They normally string out multiple times, a dozen times the length of the animal. or more.
Right. That's why I feel like I've seen that. Okay. Thanks.
Wow, it's got that green tinge on it as well.
Yeah, around the edges.
It's nice. Okay.
Yeah, it's cool.
What's the white in the middle?
That's the somatocyst where it keeps oil and food reserves and is also used for buoyancy.
Balance.
That's how you can tell what species it is. It's perpendicular to the long axis of the bell,
Totally thought you were
that thick white bit, so that's what makes it
He might have had a
multidentata.
chance.
It's like a spaceship for real.
It does. Very spaceship-like.
And isn't it weird how the jelly, a soft jelly, can make those hard edges and ridges like that? I think that's really
That jelly's still
interesting biomechanically.
there? I don't think
There was
so.
another jelly right
You
around
might,
it. We just
yeah.
dropped down.
We just go down
Yeah.
and see instead of going down and forward.
Can we
you know?
drop down a little bit?
Yep. Yep,
And
that's what we were just talking about to see
Yep.
if that jelly was
that
there.
second jelly, yeah.
Yep. We're with you.
It looked to be a repellinomatid. The second jelly, that is.
I don't know about that, Tuchel. Oh, there he
Oh,
is. There's
that's
one over to the
a-
left. I don't think it's the
That's
same
how
one.
they're created.
What's that?
What's that over there?
And then what are these two in the middle?
That's- Yeah, more. Oh, that's the Doliopsid with
These are the-
Three animals.
Yeah.
three animals
Let's
and
go
a
to the
skull.
3-1.
We're not going deep anymore. Let's get rid of
Kind of
that.
ducks.
Yeah. Yeah.
Wash here.
Okay.
Moving along.
There's a bunch more just
There,
down to the-
yeah, there's a whole
the
bunch
right.
to the right that were just falling.
I think that might have been part of the same colony. and
Right, maybe it
refracted.
was.
Oh yeah. One there, one there, one there.
Yeah.
We're helping them disperse.
Exactly.
How kind of us.
They are probably the most poorly known family in the Pelagians. because it's just not recognized and not abundant.
I beg to differ. Look at them all.
For a second, I thought that lobster caught up.
He probably did.
Put him
Probably
in,
did.
You want to put him in a D-sampler, and because we can always.
Okay, how's that, you guys?
I mean,
Nice.
would the allow us to check if it's described species, if it's Doliolunacris or not?
I can't see any red pigment.
This was a colony.
Certainly
There's a bunch
consistent
around.
with that. But we can't see the muscle down the side.
They're connected by that muscle, isn't it?
The
What connects the
stolon?
two?
It's called a stolon.
The stolon. Nice zoom.
Is it D-samplable?
There's D-samplers available.
It should suck up too, it's not that delicate.
You two make your choice. We have 2 D-samplers and 4 canisters.
We're not gonna know what species it is until we collect some. It's a couple of individuals, so you'd have a couple to use for genetics.
We can suck it, and then we could put something on top of it if we need to later, because it's hard, right?
No, they're pretty soft. They get rolled up on stuff real easily on a plankton. They're sticky on the
Is this
outside.
one?
I think we'll
What are we doing?
suction.
Are we
Suck
suctioning?
him up, yeah.
Yep, suctioning him into canister number 1. Is that correct?
Yep.
That's the one we're on this second. Yes. If we could suction these guys, that'd be great. Thank you.
Just above the
Oh yeah.
thing right.
A lot easier to work with than
Julieta.
siphonophores.
Can you read the chat?
Yes.
That would be great.
It'd be a good
Some
animal
folks are
for
asking
that
for some Spanish.
laboratory imaging system, probably.
If you're up for that.
Ya vengo. Momentito.
What are we doing now?
No, yeah, but what are you looking for? Ah, okay.
Did we lose him?
No, I think they're right up here.
Oh, I
Above
see
the
him.
suction
Yep,
sampler.
I see him.
Yep, coming in.
I think I know what it is. Well, I think that's
Buenas tardes a todos los que están del otro lado que
Sucky, sucky,
estaban esperando por un poco de traducción en español. Bueno, acabé de llegar al estudio, mi nombre es Julieta. Así que a medida que vayamos viendo qué es lo que estamos yendo atrás, creo que era un nidario, un tenóforo.
Too far.
Yep. On the other side now. Coming back. Euphausid in the middle while we're watching.
Hay un pequeño camarón en el medio de la pantalla.
Off a little bit.
Cream yellow.
We're trying to make him about 7 meters at a time. We'll get there. Yeah, no worries.
Yeah,
They're
there we
in.
go.
They got in.
Yeah.
Yep. They went in the sampler. Take a look. Canister watch.
I'm watching.
Thank you, Russ.
Just go in?
I haven't seen it yet. Yeah, it takes a couple.
It's just one, right?
It was 2 doliolins. It was the 2 that were.
Suction sampler one. Yeah.
Oh yeah, I'm sorry. Yeah.
What was captured?
Small Theliacea.
2 or 3 joined together on the stove.
That is from the
Correct.
group of the
They're a pelagic tunic.
Not in the canister yet.
Like a
Bueno, acá confirmamos
Like a
que pudimos capturar el tunicado que estábamos buscando.
Not seeing anything in there.
Not yet.
El ROV hizo su inmersión a la 1 de la tarde más o menos
Are we
y
still
vamos a estar
sucking or just
transmitiendo directo 12 horas hasta la 1 de la mañana y luego después tenemos
Taking his leisurely time.
los momentos de las redes, que las redes son lanzadas al agua también a diferentes profundidades para seguir estudiando el zooplancton, los invertebrados de la columna de agua.
Want to try a little pulse with the suction?
Yeah, kick the suction for a second to give it a kick. Get some flow.
Cualquier duda que tengan, tanto en español como en portugués, a lo largo de la tarde y de la noche vamos a estar traduciendo, tanto yo como el profesor Renato Nagata, que es especialista en nidarios,
Do we want to keep it on
o el
one for
profesor Marcelo Melo,
now
que
and
es
not switch?
especialista en naves,
Pump once more.
en peces de profundidad.
I think we got it, and I think it'll come in.
There's full.
There.
Well,
You
he
got
just had it
something.
on full.
Is that it? Really?
That
Well, there
full
was
is
something
what he
just
said.
bounced around.
Something came
Let's
in.
stop it for a second and see if it comes back down.
I didn't see anything go up.
It's a couple of things bouncing around in it. Who knows what.
So not the,
Can't see it. How about we keep it
no,
on
there's
this,
little amphipods or somebody in there.
There we go, this stuff.
You're getting a bunch of stuff in here.
But little stuff,
Still
but
can't
not
see the
Doliona.
Just leave it on low while I'm flying around.
Something came in pretty quick.
Do you want to just leave it on low and keep it on this canister
Yeah.
for a while?
Yep.
Okay. All right, I'm gonna make an executive call that we are not gonna stop until 600 meters.
Wow.
I know it's all of 40
Ahora la
something
profesora
meters.
Heather como dice que vamos a seguir directo hasta los
I'm
600
trying to go
metros
50 at a
sin
time right now,
parar
kids.
para
We've got to
nada.
get down to
Es
the deep
bastante
water.
difícil porque siempre
I would
aparece
like a
algo
chance
y
for a vampire
todo,
squid today.
alguno o que otro siempre está interesado.
And we don't have one yet.
Siempre aparece algún bicho que alguien va a querer colectar o ver o estudiar con el escáner
Okay,
que
we can take a
tenemos.
look at him real quick. We can take a quick zoom. I don't want anyone to say I was biasing our dive.
I should have put money on it.
All right, there you go. That's your
qué
zoom.
linda.
That's from the first day. It's got the nematocysts on the outside of it.
Great.
Same dude. So
Got a frame grab and we're going. Go ahead, my friends.
What is it?
It's a Medusa.
Narcomedusa.
There you go. Soul
Narcomedusa.
missus.
He wants to collect it, but I'm hitting him down to the floor.
Thank you, Russ.
Yeah.
I need all the help I can get.
Make it sound like I'm out of control.
Not at all. There's lots of good stuff in here for you. Not at all. Just close your eyes.
A single cranchid or a single parakeet.
I know.
What's going on?
There are some things missing that you think we'd be seeing.
Keep the faith.
Yep. Close your eyes for a few meters, my friends. Take a little nap. Take a little nap, my scientist friends. We're all very passionate about our organisms.
Heather le está pidiendo a todos que por favor cierren los ojos
No,
para poder
not
seguir
with them
bajando hasta
inside.
los 600 metros.
Is that what you're looking for?
I would love that.
That would be fun.
I don't think we're here.
No, it was small. I think the host would have been
Did you say from this video? Oh, 5:30. Dinner. That'll be deep if I'm not here. All right, what's that?
Middle.
More totally opposite. It's
Just
rosacea,
checking.
maybe?
Just check. Oh, it's
600 meters.
I know.
It's a rosacea.
There you go. You get a quick
Oh.
zoom.
Turn
There's your frame grab. Putting it in as an observation. Bye bye.
Bye-bye. All right, okay.
Love you.
Love you.
Thanks. Gotta go.
Calicophorus rosacea.
Because
They just did a skull.
some would consider that cruel and inhuman punishment.
Is that what you want?
Our
Is that
viewers
it?
are
Is that it?
correct. It's going to be hard for me to get us down to
Teníamos
600
en
meters.
pantalla un
I
roce
don't want to get
siciliata,
things, I
también
don't want to get yelled
otro
at by my scientific friends over here.
siphonophore.
I know. I'm trying to stick to them. There's a house.
Obviamente
bye-bye.
paramos para dar un
Was it a siphonophore?
pequeño,
Oh, yep, there
una
it is.
pequeña aproximación
Siphonophore,
y que
bye-bye.
los especialistas que están a bordo puedan
We're noting them. So if you guys
identificar
are
la
really
especie.
interested, we'll have it in the video.
We're gonna go in Fathomverse.
Perfect. Fish. Little fish.
What's the vertical one there? Is that a fish?
The vertical one. Can I slow down on that, or do you guys want me? Okay, we're slowing down on this fish.
Ahora
I'm
estamos
gonna take
aproximando
crowd
un
concern.
pequeño pez.
All right,
Oh.
we're gonna catch that fish now.
Well, the good news is we're still moving downwards.
More than anything,
So,
we gotta catch this fish.
If we follow the fish down, we'll actually get
Fish has
down
gotta keep
deeper-
going.
who's this
deeper than
guy?
we would otherwise.
That is not a cyclophony.
No, it's not.
It's not Sarah
No
Vollmer.
es un ciclotónido.
I don't think.
No.
Están intentando identificar aún.
Okay. Thank you, gentlemen. We're moving on down.
Okay.
I don't know what that was. We're getting close. We're getting close. I'm gonna ask for 650 once we hit 600. So, get ready, team. Get ready.
Lots of targets.
About to catch up to the lobster larvae.
Yep,
Yeah.
almost.
Wait.
We're good.
You're good.
Yep,
Move
yep.
along,
We
move
can
along.
keep moving. Keep moving it. Thank you. They're looking out for me up here.
Wow. You see.
Nothing to see. Nothing to see. Yeah, if we could go a little more forward, that would be great.
Just gotta kind of wait for the boat to catch up. Too.
Oh, yep, yep. Absolutely. You're good? Yeah, do what you gotta do.
Un copépodo, pequeño copépodo podemos ver.
I was gonna
Con-
say that was different, yeah.
Out there hunting, center.
Center, we can take a quick look. So
Hmm.
close to 600.
Here on the right, this is a larval.
What? You think that's a larvation?
Yeah.
Yeah, people got not crisp.
Estamos intentando
Working on it.
decidir
Working on it.
qué es lo que,
Looks like something different.
qué especie es, o qué grupo pertenece, lo que tenemos en pantalla ahora.
Yeah, it's small.
And is that a boat here on the right as well?
Stand by. Can't get you on the right yet. Stand by. We got focused on this. What do you guys got?
It's got a very big in it also. It's got bumpy bits on it.
Yep.
It's probably like
Should have missed it.
or if it's lumpy, but I'm still not.
It's
I'm
not.
not getting a good angle.
It is tiny.
It's a great shot.
Mhm.
It's not out of.
Thank you, pilots, for this.
Most of them, only Bathycordia seems to have that really big outer filter. I think that's empty.
That's what it looked like. Okay. Moving on.
Ya casi llegamos a los 600 metros. Vamos a ver si seguimos.
On our way to 1200
Sin parar
meters is the
o-
goal. We're going in small increments today for small successes. We will get to 600 shortly. Yeah, the zoom would be great. We could see on the science camera. Thank you. That helps.
If you keep it out of
gonna say. You can just turn off the
That's the
camera.
trick. Just turn off the camera for all us scientists in here.
Para tratar de no distraerse más con todo lo que aparece, están
Yes.
queriendo apagar las cámaras para que nadie
That's
más
what- I'm
preste
gonna
atención
ask
y
that
puedan
we
llegar a
keep
los
doing it.
600
We made it to
metros.
600.
Pero no, ya
Congratulations,
llegamos.
team. We made it 600. Do not stop. Keep moving along, my friends. We are halfway.
Mm,
We're supposed
Todavía
it's
tenemos
5:30.
We're going till 6 a.m., right?
600 metros más para bajar
We're
hoy
not going to
Vamos a
6
llegar.
a.m.
hasta los
Little.
1200 metros de profundidad,
Yep,
como
yep.
llegamos ayer.
We're slowing down for it. We have officially passed 600. I can slow down if you need to. Oh, there's somebody in there. I see him in there.
Oh, it's the
He's in
guy
there.
with the bumps, the four that the miss
We
we
can
could
zoom
we
in
were
on
doing.
that. As you position as needed.
Oh, I see.
He's in there. Do you guys see him?
Yeah, it's definitely
Yeah, there's a larvation in there.
someone in.
O en
Yep.
pantalla tenemos un larvacio,
Me too.
ayer creo que también habíamos visto varios. Estos larvacios pertenecen al grupo de los
There's no bad
urocordados.
guys. We're at 600. If this is important. Did you say disagree?
You in the scanner, right?
He is small. Yeah, he's pretty small for sure.
Ready?
Are we doing it?
Sure.
DPIV?
Yep, let's.
We're gonna DPIV
Laser
this.
scan it, please.
Cameras on.
So, my recommendation.
Oh, sí, lo van a escanear. Como estaba
Yep,
contando,
if we can get it as soon
los
as possible,
larvacios son
if possible, through the sheet, and we gotta keep it inside her frame.
organismos
So it's gotta be
solitarios que
closer.
pertenecen al grupo de los apendicularios.
Oh, that was it?
Ellos
Okay.
secretan un muco alrededor del cuerpo que les ayuda a captar partículas para... Esas partículas se pegan en este muco y los ayudan a alimentarse. Una vez que esta casita que ellos construyen está
Oh, you got it. in her.
llena
Oh,
de
yeah,
partículas y
Yeah,
sucia,
no, it's perfect. I was
la
almost
descartan
so-
y empiezan a secretar una nueva. Ahora están intentando escanearlo con el láser.
Oh.
Okay, so. No, it's not.
Go deeper. No need.
Ahí están intentando
So whenever you're ready.
que pase más veces por el láser para tener imágenes de diferentes ángulos y poder recrear
Zooming in.
la estructura 3D.
But yeah, that was exactly the kind of.
Para los que están del otro lado y hablan español y en portugués, a las 7 de la tarde los profesores Renato Nagata y el profesor Marcelo Melo también van a estar acá en el estudio traduciendo para portugués. Así que cualquier duda que tengan sobre estos animales de media agua,
Sorry.
tanto nidarios como peces de profundidad, pueden dejarlas en el chat.
Yeah, he's low. This guy, right? There you go. Motivation, and he's in his house. Oh, he just went low again. Here he is.
I got nothing.
Yep, no, I understand. Just above. Now we're good. Yep. Went high. Now he's coming back in. Yeah, no, you're good. Nope, he's in the middle. Right there, you got him. I don't know if you got him in her screen, but I see him out here. Okay. And now he's low. There, he's back in. Yeah, he is low. Yep. He's good. I don't know how far he is for you, but he's in the right area. Yeah. I just don't know.
I don't quite see him on here.
Yeah, that's what I'm saying. I don't know how far away he is, Phil.
Come down a little bit.
He's up.
Go ahead.
I think he's just telling you every option. Oh. That was it. Right on the edge. That, oh. Yeah. I don't know where he is now. Oh, there he is, coming through. Oh, that was good, right? Yeah.
Easy.
You're still good. You're still good.
Right before
He's moving up to off her screen.
There we go.
Here he is, right there.
It's like you're driving on the highway and both your headlights blow out at the same time.
Yep. There you go. Yep.
We're getting a big.
Yeah, what's that?
Oh, we might.
Or below us.
Yep. Keep an eye on it, Russ. It's a cell chain. No, he's right here.
Can you check it down a little bit?
I think he's here.
Yeah, I see that.
Yeah. Maybe?
Keep going down.
Now I'm just seeing things.
Yeah. Okay, go ahead.
Go ahead.
Easy.
Oh, here he is.
Sometimes easier to see it on the cross section.
There he is.
Does it matter to you if it's a reverse scan or a
does
forward?
not matter. So
Reverse the video.
do
Huh?
2 quick shots. It's not this yet.
Do you align these frames from all these different scans and average them to get rid of all that stuff?
Down low. Do you have him in your camera? He's here in the back. Yeah.
You pay me out some?
Mm-hmm. Pero. Oh, maybe. He's right in here.
On the other side.
Yeah. Sorry. Yep.
It's closer to us.
Yeah.
Okay. Okay. Backing up.
Mm-hmm.
So that's speed.
Jerked around by my boat.
Yeah.
They
It's
can
moving.
out so much because the current's blowing the.
Yep. Could Connie want to give it, oh, I was going to say another shot or two, or what are you thinking? Sure.
Take it. Thank you.
You sure?
Yep.
Sweet. All right.
So we think that's it.
Thank you on that. We are continuing down. Now that a lot of the scientists are at dinner, we might be able to move faster. Just joking, scientist. I know you can hear me. I will stop on all cool things, I promise. Siphonophore, just zoom in quick for it. We'll get a good zoom and a shot, and then we'll move. Right? And you guys are logging this, yeah? Perfect. I don't know about that. Siphonophore, Russ, do you know what kind?
I'd be happy to get a scan on this.
Okay. You want to scan, Kakani?
Yeah.
Yeah, same, DPIV?
Well, are we happy with the views that we got?
Yeah, well, happy with
Yeah, DPIV.
DPIV scan, please.
How's it work with those, Russ? How does that work?
It's I can put it to Bisonex. It's one of the ones with the air flow.
Trying to keep an eye on the.
But this is
Build the
really
phone,
good.
okay?
It reminds me of the Arcadian, but
While we're scanning this, I see on the chat the max depth for this dive is 1,200 meters, and we're diving until about 1 o'clock this morning, local time in Brazil.
Yeah, the exact recommendations to watch that screen.
Ooh. Yeah? What wasn't good about it? The angle of the animal or the speed or?
And that's one we really want to change, get both direction scans. That's a little bit better.
Okay. Oh. Uh-oh.
Whoa.
Is that a pear?
Hair?
No, you keep doing yours. Don't do anything else. Yeah, don't pay attention to me. Nope.
In a pear tree?
We're good. We're all good. I think it was a feeding tentacle. We're good. Oh, that was nice, no? Okay, okay. I'm trying to get an idea of what you're looking for.
Yeah, I think it's just, like I said, it's kind of
Yeah, the current has done a couple of things.
wrapping my float around. I try and get it as
It's
steady as I
the
can.
continual every 10 seconds. It's taking it right now. But you're good. One, yeah, but I think it is yesterday. One. Yeah. Leave it. This way, there's something. I think these are these. These are just what we see, and it's doing it every 10 seconds, whether we're seeing it or not. I think these will change.
What's the question?
The A snap button at the bottom. It's the video. It's the frames we see, right, on the top up here. The frame grab and the A snap.
So, yeah, if you turn on that A-snap, like A-snap's always running.
Right.
You just don't see the events in the event history. If you turn that A-snap
So
on,
you can
you'll
turn
see
that off so
every
we don't
10
see that.
seconds.
So that's all it's doing. It's yep. Thank you.
Yeah.
Because
It's
then,
more of a nightmare.
yeah,
when we're
because then
doing
we
that
just
once
won't
every
see it.
1
We'll see the
second.
observations. Yeah, exactly. Thanks, Zach. Or no, Alex. Yeah, I was gonna say. I'm like, no, that was not Zach. That was Alex. Sorry, Alex.
Zach's flying. He's doing good too.
Kakani, how many more you wanted this?
We just zoomed in a little bit more.
Yeah, sure.
Oh, there's
What? There's a salp hanging on, coming on around.
No selves.
Nope, don't you worry.
Run into each
They
other.
are gonna pretty. Oh, that was. Oh, and you got the salp at the end.
it was out of frame though.
It was out. Yeah. Yeah.
Self a day.
I don't think I sweat this much in the petroleum war.
Yeah. That's what happened. This is what Jen, wasn't it Jennifer, who was doing the same thing, right? It's intense. It's focus. It's just so much. Okay. Almost in. Now, he's in mine, yeah? Yeah, he's moving around now.
Okay.
We're good?
Thank you very much.
Thanks, you guys. Zach's like, okay, I can breathe, loosen up. We are headed back down towards our target of 1,200 meters. Thank God. We are doing great. We are finding many, many cool organisms. We are using a lot of our technology that we've come on board to try and practice with, which has been amazing. And the pilots haven't left us yet, so we're sticking with this dive for a while.
All right.
All right, yeah. If we could do our search, head down into it and forward.
600 again.
Oh, and two. We're back up.
Yeah, we did. Everyone's closing their eyes for the next 5 minutes.
It's like climbing on sand.
It is like climbing on sand. I will agree with you right now. There's great organisms for sure.
Is this mic on,
Yes.
Alex?
I think that's the one.
They're all on, my love.
Another siphonophore?
Yep.
Center, another Mars?
Is that the one we just scanned?
We're gonna
Like
keep
we just
moving.
looked at?
Yep.
There's
Are we zoomed
a
out?
question in the Twitch.
Are we? Why am I seeing that?
So we have two different instruments.
Oh,
One
it's just
is
different. Got it.
DKV,
Sorry about
which
that.
is the bright red laser sheet. and then iris, which looks like an umbrella on the front with a bunch of red lights. And these two things are essentially they are capable of imaging different things. DPIV is great because if we're looking at transparent things, gelatinous animals, even mucus structures, we can not only see exterior surfaces, we can also see internal ones. And so by the act of us scanning, right, moving the laser sheet through an animal or mucus structure, we're collecting all of that imagery, much like a CT or MRI scan, and we can use it to create three-dimensional models. And so we've done that on a number of different animals, and here we're hoping to do that on a number of siphonophores, jellies, ctenophores, etc. Now iris is a little bit different. With that, we're able to image the exterior surfaces. And so we can get three-dimensional models or, 3D surfaces of the exterior of these animals with a single frame. So really high throughput types of data that we're collecting. And so the idea is that combined, using both of these approaches, we'll be able to get really great quantitative measurements of all sorts of animals we come across.
Our whole menagerie. You can do all kinds of flavors. What do you want to do? You want mango stuff. Ooh, mango. Okay. You want flying or co-pilot? I'll fly. All right. You ready? Yeah.
Rosie again? I prayed that there's too much.
We have a switch out of pilots happening right now.
Okay. Seems like you kind of, sometimes when you stop, it tends to push you away from the vessel, and then when your umbilical gets tight, it kind of strums you around. It starts strumming you a little bit, so it can make it a little challenging, delicate stuff.
Ice cream up there.
If we could go forward and down is the current situation. Forward and down, forward and down.
Forward and down.
Yeah, at a
It's dark.
little better speed than search speed because
Because you want to go down?
yes, we actually need to get towards our target.
Like it's, just a little floaty, but not bad. Forward and down.
Forward and down. Let's do it.
Like on that last one.
Yeah.
Ready? Yep.
Nope. Yep, we want it out as far as possible, and then we want to move forward and down. So the snow's coming in our face if possible.
Yeah, I'm going. Forward going.
Yeah.
I can't see it.
Yeah, like that was great.
We finally get rid of most of the siphonophore parts off this middle of the screen. Wow. Fish.
Cyclothony.
Wow, that is beautiful.
Another oik.
There's still a lot of work that needs to be done.
Jelly of some type.
Small medusa.
And the parent, is it computationally crazy? Put it together, or?
No, I mean, it's actually really straightforward to make these models. It's the
Mm.
post processing. How do you make sure that
Oik.
Yeah.
so once you have your images to work with, it's really easy to make.
Yeah. Wow.
There's ice cream.
I heard.
It's like
I've asked them to speed
that
up a
was it.
little
It's all
bit
in yourself.
because we aren't getting anywhere. So I'm gonna hand it over to Karen so I can grab some quick dinner. I'll be back.
Wait, ice cream is for dinner.
There it is. Go get it. Or whatever it is. I missed it too.
Did you just get here too?
Yeah.
How long does it take to?
All right.
About 30 seconds ahead of you.
It doesn't take very long.
Ooh, jelly.
That's perfect.
Oh.
Both make this. What frame rate?
Oh, copepods.
Are you?
Doing events? I could, but I'm never usually
Have you
good, very
eaten
good,
or
fast
someone
enough at
is
it,
coming
so.
for you?
And I get the IDs wrong, so.
So funny that it always looks to me, this picture, like the glow off of the tubing and stuff down there is like there's this little green glow happening there, but I know it's just the light bouncing off of it. Oh. Yeah.
You know, like.
Probably when you got up 1200, it might fire.
Well, first I was like, God, is there something wrong with the camera?
No, fortunately, it seems like it's on. Oh.
See, having somebody new sit down is not good for continuing down fast because everything looks fun to me. That's a nice area.
Silo
What's
Denver.
this? Can we look at this?
You're welcome, have my seat.
And what is that?
It's a Calicophryn.
Is this another one of his siphonophores?
Yeah. Okay. We can leave
Yeah,
that.
it's a Calicophryn. It's been saying they're clausal or
Little
tunnel?
rocket ship.
Is that the one you ID by the orientation?
We're gonna pass it up.
Gotcha.
Yeah, a little bit, and the shape, overall shape of the rocket.
Back.
Oh, too late.
On a mission to the deed.
Okay. Sorry.
Even if you have to pass all of those
What?
head into the deed.
Yeah, they just don't get
There's
their
some pretty
fish.
big ones down here.
Yeah.
Like a hatchet fish, maybe. Need to find some
ID stuff
ultra
off
black
the
fish
pictures I
down
took.
here.
Euphausid.
Euphausid, Ketigna.
It's so hard. Blurry, the lighting's bad.
I feel like we can see so much more than
Down
we could
center.
earlier. Maybe
There,
just because I
straight
finally got a
center.
little sleep for a half an
No.
hour. Mm.
She's on a mission.
It is in the ice cream.
Whoa. Is that ink or?
It does look like ink. Could have been a scythe. Can we do a little more forward?
Yeah, we're heading down to 1200 days travel. Okay.
Oh, there is a little jelly, tiny jelly in the back.
Cyclostone top corner. Jelly coming up over there.
I always have the feeling when we're doing Midwater, there's something just outside our range, just outside the light there.
Yep.
Constantly strain my eyes. Whoa. What's that? Oh, that was a worm. Tiny one. Probably a lepidarincid, Pelagopia or something. We are blazing.
Six.
0.6. Wow. Another little Calicophryn. Certainly no shortage of things in the water here, despite what our toes seem to pull up. Look at that. white thing. It had an interesting flicker beneath it. Oh, it's probably a res area. Kind of reminded me of a spider, but it's probably it's down just off the bottom. Outside of it. Almost thought it was a spider. And by spider, I mean an isopod. Midwater isopods look like spiders, the Bunuricopy ones. The Menopsis. The Canthomenopsis. There's a whole group that live up here in the water column. They hang there like a spider hanging on its web. And I haven't seen a single one yet. Kind of amazing. A little bit more forward. Now it's 680. Almost. Cyclophone or Prisma. Ooh, a little
Totally
tiny.
opposite.
We don't have to watch that guy. It's cute. Are we still zoomed? All the particles got big. Do you look at that? This one?
That one? Yeah.
Like a small bisonect.
Isn't it?
I think
Yeah.
so.
Yeah, go for it.
That was beautiful reconstruction you did.
Ah, yeah, you saw it?
Yeah. Really nice.
Another one of those small mirrors. This looks Apolemia-like.
I don't see the tentacle.
No, I just saw the
Shape is
Had a
similar
couple of
to
other little
Apolemia with
mirrors.
a short tail, but I don't see the tentacles either. Okay,
This
we can leave it.
Yeah.
We won't be able
Yeah.
to scan it or do anything
Not quite this small.
with it.
What did we think that was?
Marus, Marrus. Yeah, I'm not a fan of real spiders either, although they are pretty cool. But midwater spiders, those guys, They're really cool. They stride through the water using their legs, stepping on the water with big paddles on their feet made of hairs. So, cubic now. Yeah.
Fish. Another fish.
Larvation up to the right.
So when you do a scan like that, what kind of resolution?
Well, the video is 1080p, so it's
Interesting,
that
right?
times the number of frames we put
Oh, the fish,
together.
with
That's usually a couple hundred. Because we recorded 60 frames per second, so 3 seconds of scan is 180 frames.
Yeah.
So you lose some resolution based on because of how the laser light shows up, but we can zoom. So what that relates to in terms of millimeters can change quite a bit. A good baseline is 0.1 millimeter.
So many
per
sites.
voxel.
Per what?
Per voxel, so it's a 3D pixel.
Oh.
And oftentimes they aren't symmetric, so you might have like pixels in X and Y,
Yeah.
and
And
then in
different
C,
depths.
those frames may not really be spaced a little further. And then the first step is kind of what you saw, is to create a volume membrane. That's what I showed. It always looks a little deceivingly good because to make a true 3D reconstruction that you can turn into like a mesh model, you need to really clean up that data.
Yeah.
But to do a volume membrane, you don't need to do quite that much.
So, but you need to figure out, because if it's going through really fast, it would seem like it would be squatter. Is that right?
Yeah, so that's why I kind of multiply that dimension sometimes to fit what it
But
needs to
you
be,
know
and we
what it
use
looks
that
like.
feed from the side, and we use different views as a reference to, the symmetry. So very often with these, it's pretty easy.
Oh,
All the jellies they
have a
have,
look at this guy.
they're supposed to be round.
Nice rise there,
So,
yeah.
Dendrus.
Right, so,
Dendrus.
so you know stretch out that third
Just a
dimension
single.
until the circle is round,
Right.
Single-celled organism.
instead of like a squished limbs.
We can move along. We're looking for one that's paired.
He wants a paired one. Just a single one is not enough. But
Yeah, they didn't all work out that great. The Galaxy side was pretty good. Like a third was a little bit harder. Dolio was okay.
So, but you reconstructed it not from the PID, right? Right, you're doing it
From the PID, yeah.
From the PID. So, how is the iris different? I mean, the iris is just a snapshot at the same time, and that gives you kind of that surface topography.
Siphonophore?
pretty little
So
site.
like you said,
Let's
the
look at
surface
the head
topography,
and see what
so you
kind
have
it is.
full, like 3D every point in space in this volume.
Yeah.
Nicely spaced.
Some requires the laser to shine through whatever it is. So iris is really good for
Just a quick look at the head is enough.
the demo example.
Oh, unless he decides he doesn't want to be here. Psychic helicopter.
looks like a what?
It has two
Another
swimming bellies.
Prayed?
So it's a prayed if they're side by side, essentially, which is, I think, what it was. Okay, we can leave him. There's a fish out there. A little more substantial than a cyclothone. Oh, stop on this jelly. Is that Tetrorchus? Something like it.
Ooh, that's cute.
Is that Tetrorchus? I'm asking the jelly people in the back. It'd be nice to get some video of this. Very pretty gut there. Unusual way to hold the tentacles.
Need some relief to get to dinner?
Yeah. Kind of just try and grab a picture of this when we zoom back in.
Oh! Tetrorchus!
It's easy for you to say.
Tetrorchus erythagaster.
Thank you for confirming.
to say that.
Red stomach.
Funny.
Does it only have 3 tentacles?
It only has 3 tentacles,
It
but
says
it
one
knows it
chunky
needs to have 4.
What is it?
It's a Trachymedusa. Trachymedusa.
Got it. Thank you.
It's pretty little
I didn't
gonads
know if
there.
that one prayed what it was, and I didn't know if the place in there was in fact another.
Are you putting a
Mars
demo up?
Or
Yeah.
Might be nice to set this up.
So I just want to check the annotation.
Okay. Just having trouble finding this file.
Sure.
Oh, don't sweat it.
Add to Larvasian might
I have
this.
Don't
a
sweat it.
slight
Okay, I
memory
suspect
that
this
you
is
guys
more
like to jet
It's kind of a
suddenly.
good example of what it's normally used for.
But if
was
we can
instrumentation.
suck them up.
Should get this hand.
We're gonna try sucking him
Try.
up there, Jocko.
Right.
We're gonna try sucking him up.
Oh, we're filling the buckets quick today.
Rom, extend the ROM for you. Yeah, it's gonna bring it out a bit, please.
Oh, lost him
So
for them.
this is the type of thing they like to use this for like measuring
Oh,
things
it's up
on
there.
circuit boards. If you look at the total focus version, you see this depth map on here. You can measure all these individual features on it. In this case, actually the 3D model I think works pretty well. And you can see how like these raised elements over here.
Is this
all
gonna be an
stick out
8?
pretty nicely. You can
Oh,
measure
the T-bars are
if they're all
not.
the right height. So they use these in kind of a production environment
Just
where
down just a little bit out front.
the lighting is fixed. The camera takes a picture every time like the new board moves over your picture, moves picture, and it is for like screw, thread, pinch, to verify. So
I
they
know.
really set up these.
This is the thing, I looked away as well. The
With these
T-bars
types of
are not
instruments. Especially at the
oriented
cheers that it
well.
requires like a single lens. So you can look into things
Yeah.
pretty well.
I think we lost him.
Yeah,
Because in this
I
case,
shouldn't have
lots
looked
of
away. He was a tiny little guy.
Yeah.
Sorry.
It's alright.
Is there anything we can do about
The
those
image in the
T-bars?
back.
Yeah.
Yeah, it's a
we'll-
background, it's
we'll,
a
uh,
total focus image.
We can
Jump
him
as well.
Nudge him a little
From
bit
years
with the
ago.
one.
Oh, it's in background.
So, in using that, I mean, the benefit of it is you can control the speed perfectly. So by running it through controlled speed, you never have to do those kind of fixing algorithms that you had to do to
Yeah,
adjust
absolutely.
kind of more
You
quality.
put the camera on a tripod relative to a plane that you care about. So we have kind of some challenges that people are probably have.
Yeah.
This is more of a lab, like typical setting on this.
Did you have
So-
dinner, Karen?
I did, yeah.
Okay.
Why not on the back side of your laser sheet, just put a, like a
Center
uniform
bottom?
suction. So as soon as you get there.
Sorry, what was that?
Medusa? Center bottom, there's a big
pull it through at
Medusa.
constant speed, and you blow it back at constant speed.
Yeah,
not rely
outside
on
the frame,
moving the
somewhere?
bus.
Yeah,
Below
we've
us?
considered
And the other
like,
camera.
the whole laser is
In the science
yeah,
cam.
that can move back and forth.
Oh, yeah.
Sadly, the
Possibly
way
down
these
there.
actually
Up, down?
usually with a lot of vibration and like not really constant speed. We've tried it. It's not perfect. A different approach would be a galvanometer.
Might be a site, that one.
Kind of move the laser sheet around more dynamically.
Or I think
So
it's a site.
these systems also get complicated quickly, so we kind of went back to a more simple design. But yeah, it has clear flaws in like operating.
Yeah, yeah.
I
Yeah,
don't
I
see
mean, you
a
have a
head.
lab equipment set up too. And there
Maybe
it's
in
one button,
Apollonia, a bit
and
far
it's-
away to tell.
And it's all calibrated.
I don't think there's a tail. A head.
Yeah, you could do it like a TSA thing.
I see the chap you were putting fathom while doing this excellent use of your time. You put some images in from the dive 2 days ago, and yesterday's dive we just pushed live about an hour ago. So some pretty fresh imagery up in
People are identifying.
Right.
Yeah.
I don't think it has a head. Can leave it.
Oh, it's doliolids, maybe.
They're pretty tight. Could be.
Yeah, I think it's. No? I gotta get my other glasses. It looks like doliolids. Yeah, it's a big doliolid colony.
Come up. Does anybody need anything while I'm up there? Coffee?
Thank you.
Chocolate?
No, thank you.
Okay.
Is Russ interested in these?
Yes, he is.
All right. Got a little bit of
Tried
video of
sampling
them, and then
before,
we'll
but I don't think we did.
suck up this chain. Bound to get at least some of them. Nice. I haven't seen them arranged like this before. All right, let's try to suck them up. Looks like we're on bucket 8. Never
about
mind the
nine.
scythe. Never mind the scythe. Although if you want to get both,
Okay.
you could. But the doliolids are the priority.
You sucking the doliolids?
Yep.
Yeah.
Can we do a quick scan of it first? And we've got it on.
Can try. Quick deep PIV on these guys.
Yes, please.
And there we go.
What are they, by the way?
Doliolids.
Yeah, there's 3 we were trying to catch before.
Yep.
Kind of like that, a chain of
Yep,
doliolids.
yep, yep. They
I
were
think
all
it's
just-
different.
It is though. It looked
Yeah,
very
it's
different than
not-
what we saw before.
Not the usual chain.
Right.
These back.
Yeah.
Yeah.
The DPIV camera keeps glitching on the levers, but it's fine on my side.
Hey.
on the left. And
it's a little low.
Maybe.
When we actually scan through, can we dim the ROV lights?
Joel, did you hear that?
When we
When-
scan through, can you dim the ROV lights?
You know how we turn the lights
Or
off just
partially.
as it gets there, like in this, just before the laser, cut the light.
One way to do it too is for wait for Marco, and then Marco can say when it's a good time and it feels like he's about to push through the sheet. Looking good. It's there.
There you go. The Doliola chain.
For the scan, it'd be good to have even less light, less ROV light.
Yeah. Yeah. You're still good. He's low. He's coming up. Coming back. You're still in the middle. Going low. Back in the middle. If we could do it a smidge slower, but I don't know if that's possible. It went pretty low. Like out of frame low for me. Yost, do you want to do one or two more? What are you thinking?
Yeah, let's try a little more.
Sure.
That was close. If we just go a little bit slower,
Yeah.
then we'd have the money shot.
It's like bouncy
I'm
today.
aware that it's very easy for me
I know, right? Every time, like,
Just slower.
just focus, just go slower. Just seems so easy. It is definitely not.
The appropriate response from Marco would be just go higher frame rate, which I cannot currently do.
Coming in.
But it would have the same effect.
Got a new 4K camera to replace this DP camera, but I wonder if we need 1080p 120 that actually would be better.
Pretty low. And we're collecting this after, yes?
Yes.
Oh, I also have
ROV lights? Not yet. There.
Okay, yep, still in the middle. We're in the middle.
Yeah,
Let's go in a little lower. Back in the middle. Up top. Higher. Coming back in. Going back. You're good. He's lower. He's coming up. He's a little low. He's low out of, not out of my frame, but. One more, Yost?
one more, please.
Yeah, absolutely. Going low, coming back, going to the right, coming back. Now we just went up. There you go. Yeah. Do one more pass, that'd be great. Yep, you're right at the top edge. He's up at top of it. He's above. It's coming back down. Coming in, maybe? Oh, nope. I can't tell if he's inside or outside. He's off screen top. Yost?
Yeah.
You good? You want one more? What do you need?
Love one more.
Yep. Let's do one more and then we're gonna go collect. Marco's like, great. You've got this. You're doing
Ship
great.
roll
This is not an easy assignment.
Ship roll on that last
We are definitely challenging our pilots on this trip.
It's lined up nicely.
Do that.
Straight ahead. It's a little low. Comes.
You're in a good line. Oh, now he's high. Right, Yost?
It's high.
Yeah. It's coming back. He's coming back. I got him. Oh, now he's going low. I think he's still on the outside. Yep, you got him. Great.
It's high and inside.
Inside?
Yep.
Thank you.
Off screen. Coming back.
Inside, outside.
Outside, I think.
Apart.
Oh. Okay.
It's getting a little lush around. Why don't we just call it and go section it? So,
Yeah, if we could. Section that if we can get it.
Did we get the dually options sample?
No, not yet. There may not be a lot of doliopsids to collect. There might not be many to collect.
Did we just blow it away?
A little bit. There's some, there looks like there's a piece out here that maybe we could try.
is that what the
See what I'm saying?
glitching is from?
I don't know
Maybe.
if that's.
We've tried to restart it, like reboot that encoder, and it doesn't seem to fix the glitching, so I figure it's swap the water.
Get a little bit of zoom in there, please.
Yeah.
Sorry. Thank you, Papa. About the thing?
Yeah, I went and swapped it, so just center it
It's just like a piece of something there. I think it's it.
Oh, even worse, that one.
Oh.
Hold on,
Missed
I do
it.
have the heated still.
I don't think. Is that in?
It really doesn't like it.
Huh.
Oh, other cable's better. Oh,
There it is.
Press it.
A couple of pieces of it.
I know.
Something really warm.
All yesterday
Could be
was
this
good.
device, right?
It could be the output, but at least y'all's monitor's good.
Yeah, I got the data's right.
Let's see what it looks like this time.
One thing I could try too.
Maybe we scared it
That-
to behave.
oh, no.
Alright. Well, it's better that with that
Yeah,
light.
let me, so this device runs really hot. I can turn it off for a little bit and then I'll turn it back on.
Yeah, there's a little piece down there. Let's try that, and if we don't get it, we're moving.
No, I just plugged up with it. I kept one we're not gonna get back. Simply as well.
I didn't either. You went behind it again. I don't see it. Okay. Thank you, pilots. We are going to move on. If we can go into search mode, down and forward at a moderate clip, we'll stop. We are still working our way down to 1,200 meters. We are at 715. Also, pilots, I've got Niskin requests. Do you want me to tell you them now? What would you like me to do? We want to fire all bottles, but they are going to be at different depths. And we've got one bottle at 1,200, one bottle at 500, two bottles at 50, and then two bottles, I guess, as close to the surface as you guys feel comfortable. That would be great. Thank you.
You like remind us like
As we get there. Yes.
right, but so many things.
And
Normally
Niskin bottle
forget.
1 is what fires at the 1,200, right? It goes 1, 2, 3, 4, 5, 6. So
Yeah.
5 and 6 will be at the surface, right? Okay, I'm making sure. I'll take notes here too. I'll just put notes for this. Yep.
Down.
Yes, that'd be great. At a lovely pace is great. We're gonna stop for only amazing.
All right, give me a
Well,
minute.
everything we stopped on's been amazing.
I agree. That's why I was like, this is hard to quantify at this point, right? Hard to
You're
figure
just
out.
looking for a shortcut.
No, not me. What did we just say? 2 at 50 and 2 at.
I snacked.
And we're zoomed out as far as we can be?
Yeah.
Thank you.
Oh. Now we are.
It didn't feel like we were. I was just checking. Just like to check. We need as big a field as possible. Thank you. Chat with Karen.
Yeah.
Fixable. Hi. Yes. Yes, we are.
You made a lot of progress.
And we continue to do so. We have 20 minutes to get to 1200. We were close yesterday. We did it with about 7 minutes to spare. We'll see. We're good. There's good stuff.
Not too many. Probably do 4 polyhistory.
Okay.
Beautiful copepod. A beautiful copepod we just passed. Fish. I think that was a house, Russ. We're still here. See.
Probably looking in the wrong place.
Yeah, it wasn't clear. Middle, low.
What's that coming by? Is that
Looks
house
like a
with you, or is
ctenophore to
that
me. A ctenophore or a house. If we could zoom in on that, that'd be great.
I think it's a ctenophore. A little bit.
Yeah, that's what it looks like to me. But let's get in closer. Do some good zooms.
No,
It's not a
big
ctenophore.
house.
Yeah, it's not a ctenophore.
So who do you belong to?
It looks actually. A lobate ctenophore is what it looks like. But it's a house? Larvation house, perhaps?
Yeah, I haven't
Yeah,
seen
that's
this
great.
structure before.
Yeah,
though.
this is great. If we could get some good image or video on this, this would be super helpful.
Yeah, I think so. I just don't. The structure seems kind of foreign.
Right.
It isn't like a pleurid.
Yeah.
It's awfully big.
What do you think, Dougal? Is that Dougal that just
I'm
walked
still
in?
looking for the animal swimming. Did you see
I didn't
it?
see any animal in it.
It's look too narrow to be Mesocordius, right?
I see no
No,
animal.
it's not Mesocordius. It's Oikopluridae.
Yeah.
Like the family.
I love when you guys agree. I think that's good. If we can get to him
But it's
again,
not Oikopluri,
he's
right?
down lower.
I'm not sure till we get a little closer.
Yep.
I don't reckon the house seems funny. It just doesn't seem wing-like.
We had some good
but I-
scans.
Angel-like.
We had a yo
angel-wing-like.
scanned
Think of
something.
an Oikoplura.
Siphonophore, maybe?
And
Or do you mean Marco scanned it?
Yes. Well, there was DPIV. How's that?
I pressed a button once.
Doliolids was doliolid chain. Up top, middle. Let's get some zoom close up on this again. Yep. When you guys get a second, it just broke.
Just broke into.
It just broke.
Got in the wash. If
Okay.
there was an animal in it, there's no longer one.
Yeah,
now.
I didn't see one in there. All right,
Yeah.
let's keep moving.
I turned off to see what
All right, moving down and forward, please.
Silly me, I should have taken a grab of it.
You're in charge now.
No, man, I'm curious.
Oh, because I will have to.
Dougal sent a
Seven
viewer.
were doing Portuguese and Spanish.
Yeah,
Okay.
if you'll talk.
If you need Japanese, we could do
Yeah,
Japanese.
actually, no, me just Portuguese and Spanish. Japanese, okay.
I think Cheryl said something. She's gonna come down Dougal at like 8 o'clock to do that.
8 p.m.
8 p.m. tonight for Japanese, I believe, and I think we
Okay.
heard Spanish and Portuguese coming in soon.
We missed it.
Rumor on the street. So for our viewers out there, we are still working our way down to 1,200 meters. We have some of our scientists are going to come in as we have lots of different countries and languages represented. So that way we can share all of our thoughts with everybody who speaks all these languages. So thanks. Go pod, keep going.
Go ahead, breeze on by it. Nobody's getting offended by that.
I think his name was Orlik. Orlik.
It's not
Orlay.
It's going to be a fish.
I get it.
Did you get it? That's a good old Latin joke.
Yeah, Keatignath.
It's a keat.
Keatignath, I think, not a fish.
It's a keet fish.
Another Keatignath. Headed over to, oh, that was actually a fish.
Give me this one. Pull it down. Pull that one. I'll move over.
That was Schmidt
I find it's just too
without
tight to see.
drama.
Right about now, I would like a big bowl of
Is that possible?
Yeah. This one's much pain in the mind.
recess.
No, I mean, like, oh, I do have a buff.
I know what's weird. I've got the wrong glasses on. They're in the other room.
These don't help with the identification. So they would
I
help.
will like it. I will do the later.
So you don't need to shut up? Is that what you're saying?
Yeah, but I've had them.
No.
Are those just readers or they
Yeah.
prescription cheaters?
5 of them for $15.
Here,
Yep.
middle, might be a fish. Oh, it's another Keatignath. We're okay. Keep moving, my friends. Thank you.
Ah. I didn't need glasses.
Yeah.
4 years.
Yeah, 5
I was starting to just find that I
I
was,
need a glass every night.
you know,
Yep,
long
we're
days,
good.
just was, things just
Oh, coming in the bottom, we have a phycynect of some kind.
This low
Looks
middle.
a bit like a Phrylogalma. Oh, I wonder if it is. Does the bottom of it look serrated?
We're zooming in.
Zooming in, zooming in.
Yep. Working it.
Zooming in. Oh,
Getting
it
there.
looks very Frilligalma-like, doesn't it?
How do you feel about that, Dougal?
Frilligalma? You says it's a Frilligalma, so it must be.
Okay.
Yeah.
Are we good with that?
I think we're good. Are we?
All right.
Is that okay?
We're moving. Are we good? We're going on.
I only know of one, but
Off to the left, that house.
that was a house. It was very deep for a Frilligalma. It's doing all the way down there.
Fish and copepods and chaetognaths. We're seeing a lot of that.
Frilligalma would be a good 3D scan. We've got some.
Yeah, we saw a bunch in San Diego.
Middle. Oh, copepod. We're good.
But they are really cool. Just a lot more frills.
Middle.
Yeah.
I think we're good. Chaetognaths. We're good. We're good. Thank you.
Yeah.
Yeah. Down and forward.
Actually, I didn't need the extra light, put aluminum foil behind it so that I could get some arbitrary backlight on it, and did 1,000 frames a second.
Did we have to
Just like a
color
store
screen the full width, or did we also compress
I'm
it?
gonna try to do it.
Sideways.
Yeah, me too.
I feel like it's a certain order of operations.
Another 50 to 100 meters, and we'll start hitting the dead zone.
Right. Like yesterday too, we were like messing with a bunch of things. Well, there's a P thing.
You're
exactly where it's
65% there, Heather.
I like your optimism. I think this is good.
Just saying.
I know. I know.
She's close to 75.
We're working at it.
Oh, and aren't we going to 1,200?
We are.
Yeah, so.
We're getting there. We're getting there, team.
8, you're right, sorry.
Slow and steady.
Is that a kiwi, mate?
That's a chaetognath.
500.
Yeah.
I have an issue with it.
That was a kitty now.
Yes.
Orange kitty.
Red fish, blue fish. You just made me type in orange. Orange is not Latin.
Oh,
like that.
we need to stop.
No,
Oh, that's a chaetognath. We're good.
We're good.
Aren't we?
Yeah, we're
Okay. We're
yeah.
collecting them in the nets. So
Yeah.
Yeah.
There's quite a few oiks and chaetognaths and copepods.
yeah, lots of copepods and chaetognaths.
Haven't seen a single Pyobius.
No,
Remember,
me either.
remember the mission to search for the pair, okay?
Yeah, we're looking.
Okay. So a single down bottom.
Down the
Bottom
pteropod,
middle,
pteropod.
bottom middle, bottom
Pteropod,
middle, bottom right.
Okay.
You really think she's gonna go for
Bottom
that?
right.
Yep.
There's a keat and a pteropod.
I think it's a pteropod, right?
Yep.
Looks pteropodish to me.
Definitely pteropodal. Pteropod.
This guy.
Oh yeah.
We can get a middle and zoom. That would be great.
Oh, yeah.
This is a pelagic sea
It's a
snail.
gleba.
Gleba, yeah.
Oh, is it?
I don't know. I need to see.
Corolla?
Pseudo There's no shell, right?
I don't see a shell, but I need a little more zoom on that, or a little more.
Some of the deep sea shelled ones, the Pericles,
Pericles.
they have a big.
They don't look like this though.
Parapotiod, a relatively small shell,
Yeah, I don't know that that's this guy though.
and they're supposed to be deep water.
Stay in there, buddy. Come back. All right, we have a pteropod coming in and out as we're working a zoom.
It's a gleba, I think.
I think you're right. I think it's gleba. I don't
It's a
think it's
gleba, right?
pericles.
No, I think you're right. It's Gleba, Corolla.
I would go. I can go as far as gleba species
It's
and be
gleba
comfortable.
SP,
Yeah, gleba SP. So these are shelled pteropods. There are not shelled pteropods as well that we find in the midwater column. We're good. We can go on. Thank you guys.
Stick your tongue out, Gleba. Stick your tongue out.
They are pretty. So pteropods are also known as sea butterflies. They are one of the groups of mollusca that I work with in my lab with my students and the Deep End
Be
project.
surprised what you'll see.
And there's
Was that a
also
pseudo
another
cell?
group of pelagic snails, which we haven't seen yet, I don't think, on any of these
It looks like a
ROV
pseudo
dives,
cell.
which are the heteropods. They are
Smells
the
like a
sea
pseudo cell.
elephants. So sea butterflies and sea elephants are two pelagic snail species that we find down here in the midwater. We are moving on down to 1,200 meters. Forward and down, my friends.
Cyclophone. Cyclotone.
Using all kinds of accents back there. Nice.
Algeilas. Lang may Lumsriek.
Going down? Yeah. Perfecto. Thanks.
Come on, squid mama, bring 'em on.
Let's do it. Trying. I got close. I have a pteropod.
Yeah, that's
I have
close.
my phylum that I
It's
work
as
with,
close
so that's
as we-
close.
We've been for a while.
One of these days.
Straight in the middle, fish maybe?
Yep. Fish middle low. I don't know if it's fish, but middle low. It's somebody big.
It looks like a cereboma maybe.
It does. It's not Keatingna.
Just come in a bit closer. Cereboma is my pick.
Yeah, his little head's up at the top.
It
Yep.
was. Oh, and I forgot to capture.
That was a cerevomer, which is one of the midwater fishes. Is Marcelo here? No?
Yeah, I'm here.
Do you want to talk to us about cerevomer?
Cerebomber is a kind of unguiliform, typical from midwaters.
Yeah, we can keep
and
going.
it's the names come after there's a middle row of teeth that looks like a saw, and it's located in the vomer. It's a
You
bone
want to
that's in the
check
roof of the mouth, and that's how it calls cerebomber. It's a pretty cool one.
Forearm? Or the forearm that's not a forearm?
The forearm is not
That's
a forearm.
a forearm.
The radial area is formerly known as forearm.
And he did say around 800 meters.
I just heard a door
Yep.
open.
Okay,
Not mine.
heading down and forward, my friends, down and forward.
We're still looking for those coelodendrons holding hands.
Correct. Yep.
Really?
Yeah, we haven't found the partners.
They've all been going it alone. We haven't stopped playing it.
We didn't get them.
Didn't get 'em. Yeah, we tried.
We can go. Which one? An option? Yeah, a little down and a little faster, just a smidge, so we can still search. Thank you.
Okay.
Forward. Forward, down. Is that what you were gonna say, Dougal?
Bottom left forearm. Yeah, that was what I was gonna say. Sinker.
You're good with that? Yep.
Yep.
We're good.
I'm not screaming, I'm just saying.
Yeah, no, that's perfect.
Key fish. Copepod, 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 schmopepod. Amphipod from amphipod. Amoeba, ciliate, virus, bacteria.
It take you a long time to catch up. And to be 7.
Yeah, I got a little quicker and it goes up by order of magnitude.
Ooh, what was that?
I don't know.
Oh,
Stop.
low bait.
Low bait 10 forward.
See what he is.
Yep, coming in. If we could get a zoom on it, that'd be great.
It looks kind of Thalassocallis-like, but not.
Came off my screen. Down low.
Daddy.
Down low right
Oh,
still.
It's got a dark pigmented gut.
Working on it. Stand by. Went up screen. Back down in the middle. He's not quite in the middle yet. There you go. There is some current. Gone off screen. Still in the middle. Coming in. Out.
Appears to have lobes.
Coming back, Dougal.
Still can't see where the tentacle
Yeah,
bulbs
we need a
are.
little better focus if possible. Looks like there's a lot of movement.
Looks like they're at the around the mouth, so I'd say it's a Lampoctase.
Stand by. There you go. Oh, there he is. Okay, Dougal, is that better? Almost. Maybe.
Yeah, getting there.
Going off again.
Make sure it has oracles. It looks like a Lampoctase, an undescribed species.
Down off. Down right bottom. Coming back in.
Me? Sarcastic?
Yep, that's Lampoctase. Currently only one species described in the genus, all others have been recorded.
Right. Oh, we keep losing it when it gets that close, huh?
Yeah, George. Tell us.
Yeah,
Give us
to
a
say the
name.
current.
What's he called?
Yeah.
I don't get a name, I'm calling him Roger.
We'll give it another couple shots, Dougal.
Yep.
There's a current that's we're messing with down here, so.
We want to get close enough to see what color it is, because it
Okay.
seems color might be diagnostic.
Perfect. It's up top.
This is what I want the hyperspectral camera for, these guys. You have? Yeah.
These are what's your favorite?
How does the gut happen? Like, do you get what's in the shadow of the gut when the laser hits it?
Don't see a barrel eye. I've never seen a barrel eye.
Me neither.
And I've seen the video. Technically, it isn't any different. But being in the control room.
Dougal? Oh no. It came in. Here you go, Dougal.
Really weird.
Yeah, let's go nice and close. It's looking good.
Red. I see red, green, white. Actually, the green was refraction. Red.
Wasn't it spotted for the first time by the MRI?
No, the tentacle bulbs are on either side of the mouth opening. Otherwise, it looks like Bathyceroid, but that's how you tell that it's a
Dougal?
Lampoctase. Yep.
You good with this?
Just waiting for an in focus. Yep, there we go. That was in focus. Do you want this? It's a new species, so we could, yeah, for sure. Do you want to scan it first?
Yeah.
Okay, are we gonna?
Yeah.
Can we scan this, please?
Are we scanning and collecting?
Yeah,
or are we
scanning and collecting, yeah.
Scanning and collecting.
If we can, that would be great.
Yeah. DPIV?
Yes, please.
DPIV.
Man, I wish the shadow cam still had battery. This is perfect for him. Taste crude event, a red pigmented guy with your red laser. You managed to get a scan?
Scatters the light, so you get a big shadow.
Right.
I'm not happy with those scans.
Coming back into DPIV.
And it seemed,
Middle low.
yeah, it seemed to be one of the most difficult fits should try, really.
And Joel, if you could cut the lights when it comes to laser again, that'd be great. Thanks.
If they're too reading, then necessarily scatter some laser light.
Getting out of frame low. Coming back.
So they haven't seen this heat.
Yep. Low, but in there. Now it's out. Low again.
But reality is, they're getting your
In. Oh, so you got them on your screen? Yeah.
Pessoal, boa noite.
That was the
Fast.
Bring the lights back up again.
You're like, where's the animal?
It's not that big.
pretty small, isn't it?
A professora Silvina e a Julieta, que são lá da FURG também. A Julieta tem acompanhado vocês bastante aqui falando espanhol, né? E agora é a vez de ouvir um pouquinho de português.
It's not going to survive suction and the shadow cam won't get any sense.
I don't think so. He seems pretty fragile.
Sometimes, not at the same time. But sometimes we'll do today
Yeah.
one animal.
Yeah, I've had that happen.
É.
It's for DPIG here. Iris is off to the side, parked, the lights are collapsed. Collapsed. There's like the light shroud. Yeah, so if I close it here.
Dougal, if we can't scan it, do we want to collect it or?
Quer se apresentar aí?
I think we would want to hold off on.
We don't want it with the suction sampler. The only possibility would be a D-sampler to go away if we don't need it, but it's small, isn't it?
It's small.
Boa noite, pessoal. Renato Nagata, da Universidade Federal do Rio Grande, aqui. Vamos começar uma sessão agora em português e espanhol. Depois vamos ter uma sessão em japonês, às 8h30. Agora o pessoal do Monterey Bay Aquarium está fazendo um escaneamento, usando um equipamento que é o DPIV, né? Eles podem ver uma luz laser vermelha, né? Que ilumina um corpo de um animal. Esse escaneamento faz fotos em várias fatias, em diferentes profundidades de campo. E um computador, né, um software, processa essas imagens, essas diversas imagens e integra essas imagens dentro de um único modelo tridimensional reconstruído dessa imagem. Eu estou aqui com esses pesquisadores, estava conversando agora com eles, né? Esse é o que vocês podem ver mais ao centro da imagem do ROV, né? Que é um equipamento que tem esse laser que ilumina da esquerda para a direita ali. Tá?
You got this, gentleman.
Both of those are set up wrong because the
The lids will stay open.
That's why we're losing things.
Yeah. It needs to be 90 degrees to that.
Uh-huh.
The lid closed.
There's one, go in the middle one, that's the bloody belly.
There's only two.
This does not have the distinct.
Yeah, got him. Nicely done.
Okay, is that D1 or D2?
That's D1, right, Joel?
Yep,
D1.
Opa, agora acabamos de conseguir fazer a coleta de um quitenóforo, Lampoctea,
Lobe 18-4.
um quitenóforo lobado, dá pra ver ali dentro da garrafa. Foi coletado. Opa, um peixe apareceu aí, Marcelo.
You get a better shot of it in there. Yeah, zoom in and let's.
He's down here on the right.
Yeah, let's get some morphology.
Parece uma ciclotone que apareceu agora. A gente tem aqui a bordo do Falkor
Nice.
pescadores de diferentes especialidades. Eu trabalho com peixe, sou especialista em peixe mar profundo. E a gente está um pouco dividido agora, porque tem um pessoal prestando atenção aqui no quitenóforo que está dentro da armadilha e eu nos peixes. Geralmente quando a gente vê alguma coisa, a gente dá um foco maior, dá um zoom, para conseguir ver direitinho o objeto, mas a equipe está mais focada agora na captura do quitenóforo.
Yep.
E só para a gente se situar, nós estamos aqui ao largo do Nordeste brasileiro, a expedição partiu de Salvador. A gente saiu da Zona Econômica Exclusiva Brasileira, então estamos agora em águas internacionais. E a gente está dando volta no Nordeste. A gente está agora, deve estar na altura mais ou menos de entre Alagoas e Pernambuco. E a gente vai seguir rumo ao norte, até mais ou menos Rio Grande do Norte.
Undescribed. So this is an undescribed species for this group. Very fun to be able to collect it.
O pessoal está super empolgado agora. Esse quitenóforo provavelmente é uma espécie não descrita ainda.
Dougal, you get 30 more seconds, my friend.
You can be descending right now. We're all distracted.
He is a shy little
E daqui a pouco vão ter duas pessoas aqui que falam espanhol.
There he
Por enquanto
is.
estão só eu e o Renato que falam, nós dois falamos português. Um pouquinho de portunhol.
Right, it focuses great behind it. There you go.
It's very clear. I can see no yellow.
I don't. Nope.
Os quitenóforos são muito
I
legais
don't
que a
see
gente
any other
consegue ver
colors. You're right.
esses cílios, né? Eles têm essas linhas de cílios e isso dá esse efeito, parece uma purpurina brilhando neles. Ver que esses cílios se movem, eles se locomovem e pra gente dá um efeito muito bacana, assim, porque quando a luz bate nesses cílios tem essa iridescência.
Tentatively sneaking out from behind the equipment. Oh.
E eles são extremamente frágeis também, é um organismo muito difícil de ser coletado, porque eles são, né, qualquer coisa que bata nele, uma rede, uma coisa assim, destrói ele todinho. Então esse é um método de coletar o organismo sem causar nenhum dano para ele. A gente consegue depois recuperar ele, trazer para a superfície e fazer outros tipos de estudos, de morfologia, genética.
It is off. Okay.
No, it's yeah, check this. Yeah. Take it off? It's alright, leave it off now. Because everything I...
Take the glasses off, it's all in focus.
Here he comes back. Last looks. If it focuses okay on him. It is not. He's down low.
It's kind of funny because that eventually...
He's not going in focus.
Yeah, I think the autofocus must just...
I don't know if the autofocus is on or off, but I'm not touching anything.
São diversas câmeras que, agora o Renato falando, boa noite aí para quem chegou agora. São diversas câmeras que existem no ROV. Opa, alguém com o telefone tocando aqui. E aí, nem sempre o que a gente está falando, o que a gente está vendo é o que está passando no YouTube. Agora, eles estão tentando filmar dentro da garrafa para ver se o animal foi coletado. E aí, agora, opa, acabamos de ver que sim, foi coletado. Pode ser uma espécie desconhecida da ciência que eventualmente vai ser descrita, né, a partir dessa expedição.
Would be fabulous. Yep. Up, forward, scan. Down, forward, progress.
Agora, então, estamos a 840 metros de profundidade, descendo até 1.200 metros. Essa é a área de foco dessa expedição, né? O ambiente mesopelágico, o ambiente epipelágico e um pouquinho do que a gente chama de ambiente bate-pelágico. Nesse momento, né, a gente está no início da noite, os animais devem estar fazendo o que é chamado de migração vertical diária, né? Eles nadam para a superfície quando fica escuro, né, quando escurece, e através da escuridão eles se escondem, né, de predadores visuais. Então, é um movimento generalizado de vários animais do plâncton que ascendem à superfície ao final da tarde, e nós estamos indo na contramão desse movimento, tá?
Completar um pouquinho essa informação que o Renato falou, né, esse movimento de migração é chamado de movimento de migração diária, vertical diária. E vários organismos fazem isso, tanto peixes quanto invertebrados, né? Eles se escondem, eles têm vários mecanismos de camuflagem na escuridão. Eles usam da escuridão para se esconder de outros organismos. Então, assim que começa a escurecer, a anoitecer, eles começam esse padrão de migração, sobem para próximo da superfície, onde tem mais alimento disponível, passam a noite se alimentando, e ao amanhecer eles começam a descer de novo para a escuridão, onde eles se escondem. E esse é o maior movimento de migração do planeta. Né? A gente, quando pensa em migração, pensa nos gnus africanos, em migrações de aves, as migrações de baleias também, as jubartes, né, que saem da Antártica e vêm para a América do Sul, para as regiões tropicais. Mas em termos de biomassa, nada se compara a esse padrão de migração vertical diária que os organismos do mesopelágico fazem. Então eles saem de grandes profundidades, durante o dia eles estão entre 800, 900 até 1000 metros de profundidade e vão para mais próximo da superfície, às vezes em torno dos 200, 150 metros de profundidade. É uma caminhada bem longa, é um longo caminho, né, de 1 quilômetro, quase 1 quilômetro, 700 metros, 700, 800 metros, que eles percorrem todos os dias para poder se alimentar e se manter assim protegidos. Ó, tem um brasileiro ali no chat.
Yeah. It was the one we saw yesterday.
Yeah.
Kirignath.
Hit 1200 yet?
No. We're on our way, my friend.
Almost there. Are we 75% there?
Getting there.
No, 2/3 there. I
Well,
can't do math.
we're working our way.
2/3 the way there.
We're working our way.
Big house?
Big house.
Ué, cadê a...
Do you want to stop?
Looks down, no one's home.
I don't see anybody home. We're continuing. If we do see one with a home,
Or it could be
an owner. What was that?
it could be a dead jelly just kind of
Hmm.
decaying
Yeah,
its way to the bottom.
could. The good news is we haven't caught up to the lobster larvae again.
Yeah. You know, we should have put a beacon on it.
We're calling that a win.
Medusa, Susan?
Medusa, yeah. Tracking Medusa.
Those are the littlest ones, right?
Agora, nesse momento, a descida está sendo liderada pela professora Heather Judkins. Vou deixar o...
Oh, yeah. Okay. Makes sense.
E a professora Heather Judkins tem um telão aqui, não dá para vocês enxergarem no estúdio, mas é ela que aponta qual animal que deve ser observado de perto, né? E aí, óbvio que tem uma série de pesquisadores especialistas aqui de vários grupos animais.
Yep, in between.
E a professora aponta, e aí a tripulação direciona o ROV para aquele organismo. Óbvio que cada um quer ver o organismo de interesse, mas quem dá a palavra final é a professora Heather Judica. Opa, uma coisa
We're gonna
interessante
zoom in on
apareceu.
this. Yeah.
Red ones are faster than other ones.
Tomopterídeo, que é poliqueto
So this is a
planctônico.
midwater pelagic worm species.
Nós coletamos
We
desses
saw
ontem,
one
media
similar
mais
to
ou
this
menos uns 40
yesterday.
centímetros, um animal bem
I don't know similar,
interessante,
but
bem
we saw
bonito.
Tomopteris yesterday. If we could zoom when you guys are ready, that'd be awesome.
Eles são bem rápidos, esses poliquetos. É difícil coletar. Ontem, felizmente, conseguimos coletar uma espécie grande dessa.
There's some great copepods coming in and out. I wouldn't have skipped it, Dave.
What do you think's more important?
Ketignath over here as well. Still working on this Tomopteris.
He's orange. He's new.
He's different.
Deixa eu ver se não dá pra deixar
Get
mais
him
alto
in.
foto, não.
He's up to the right still, in my view. We don't have him in ours yet. Yeah, he is definitely, yep, he's off to the right. There he is. There he is. We get him down in our bottom left. So then he scooters across. He's off ours again. Okay.
He's calm down.
Yep, he's calm for a second. Need him centered.
my breath back. There you go.
Like, there's the oxygen. Time to go. Yep, if we could focus on that. Autofocus is not on, right? Or is it on?
Off is good.
Off is better for these.
Can we get a speed?
A swimming speed?
Yeah.
If it's going directly up or directly down, yes.
Yeah. But do we have to be sitting still, or can we compensate for?
No, we have to be
our-
following it.
our motion?
We center it and then we look at depth versus time for the ROV.
Yeah. Or sit still and just watch it move across the frame.
That's harder to quantify.
Yeah. If it stays in plane.
Because we don't know how far we're focused and because the
Oh,
camera's going to
You can do it to body length though.
Okay, we could focus when you guys get a second. That would be great. There's a lot of movement. We don't have him on our screen. Yeah, take your time. Oh, and he went off. Coming now, he's up on the right corner, top right. Could come down in the middle. Yeah, I was gonna say there's a swirl going on. Water is definitely swirling.
Agora vocês estão vendo uma perseguição a poliqueto
He's out of
planctônico,
our
tomopterídeo.
view.
Um animal que tem uma natação rápida,
he's
ele está
lower.
fugindo para o fundo, né? Ele já reconheceu a luz aqui.
He's right by the
E
section.
vamos tentar chegar.
There he
Opa!
went.
Agora ele desviou. Vocês podem observar o batimento metacronal dos parapódios dele, né? Essas ondas que se propagam nas laterais do corpo dele. Através desse batimento, esses animais nadam.
We need to get some
E
video on this if we can. with a
interessante
focus.
que foi recentemente
know the best way
publicado
for you to do
um
it,
artigo
so-
sobre esse batimento metacronal deles, é que esse
Zoom back
batimento
out,
gera
so
uma
we're-
pressão negativa no corpo dos organismos, né? À
Here's in the
medida,
middle.
o modo com que o animal dobra os parapódios e gera essas ondulações produz uma pressão negativa na lateral do corpo desses animais
So at the
que
very
eles
bottom,
usam para fazer uma
not
natação
focus, though.
que é chamada de natação por sucção.
Focus,
Então
if you could zoom.
ele na verdade ele é
Focus again.
sugado, né, através do modo com que ele dobra os
Not
apêndices, os parapódios na
Almost.
lateral do corpo.
Almost. Better. Better. You guys see that?
Yeah, it's
Let's try it again, yeah.
Slightly out of focus. Still.
Yep, it was getting there. Not in ours. Coming up. Okay. We could zoom and focus a little each time. Maybe step it slow. I know I'm asking for the impossible.
Yeah, that would be.
Better. Any chance for a little more zoom and keep them in focus? Or is that too much to hope for? This guy is definitely moving. He is not making it easy. Maybe. Oh, better. That's nice, you guys. I was like,
How it
yeah,
looks
peace
like
out.
they're going this way, but it's actually
There he is.
coming
Still there.
down. It's called metaclinical.
Are we collecting this?
I think Garen probably like it.
I would assume that's a yes. All right, we're gonna collect this in the suction sampler. Do you think we have enough image? We have a couple of shots.
Yeah.
You want
No.
to try more? He's not an easy character.
Okay.
All right.
I think the characters you need to see, you'll need the catches anyway.
Right, we're gonna have to catch that. Okay. So you guys, are we in container 1?
Is there already a doliolid in container 1?
The doliolid's in container 1, right?
So we should go to container 1.
I believe so. You guys, the doliolid went into container 1, correct?
I don't think you sucked up the big long chain though. So
We missed the chain,
yeah,
right?
So on number 1 is fine.
So we can put him in 1.
Because we were sucking.
Okay, so we're gonna put him in 1, the one that's currently there. We're ready when you guys are. Yeah, start some of it. He's up back here. You got him. Yeah, yeah. Let me know. I'll keep my eyes on him.
Yeah.
Coming in. Maybe he'll just swim in like the squid did last night.
There was one of those 2 paired cells that just flew off screen.
I see the dendron guy.
Oh?
See the dendridae.
Nope. Went a little low. So suction's on, yeah? Gentle to middle suction would be great.
Let's get a coffee.
Yeah.
He's right in front.
That'll be it.
Yeah, it was pretty badly melted by some of them.
Okay.
Oh, was it?
Yeah? Yeah. He is fighting you. Is the suction on, yeah? There he is. He's
He's
in.
in.
He's in. We got him in. Watch for container 1, my friends.
Opa, a gente conseguiu
Nice
coletar
job,
agora
Marco.
animal. Opa,
Wait,
entrou,
don't let it settle
entrou.
for a second.
Ai.
Or is she gonna go up? Yep. Okay.
Ok, ok, entrou.
Switch it. Right.
Entrou.
Switch it.
Index.
Yep. Switch the container.
Número 1. Isso.
Okay. Sorry.
Animal coletado aqui, poliqueto, tomopterídeo.
Got it.
E agora uma festa, né? Quando se consegue
We need
coletar
to
um
go to
animal
8, though.
diferente, bonito desse, aqui,
Perfect.
todo mundo
Thanks, you
comemora.
guys. Nice job, team.
Nice catch.
Yeehaw!
Yeah, it was number 1. Great. Thank you. All
Group
right.
4 of the
If we
adult
can
is radial
scooch
area,
down.
right? The non-radial area and radial area.
Keep going down and.
Oh, there's a big hybrid. Look at that.
Oh, stand by.
Suck him up.
Can we suck that guy up by chance?
Opa. Que ali é anfípode perídeo vermelho, ó que bonito, gente.
I don't know. What do you think, Google?
É um animal que a gente está decidindo se vai coletar aqui. Essa cor vermelha é bem comum agora, vocês vão ver alguns animais com essa coloração vermelha, né? São uma coloração que nessa área dos oceanos, né? O vermelho é um comprimento de onda de luz que não penetra tão profundamente. Então, esses animais que são vermelhos, eles são virtualmente invisíveis, né? E a invisibilidade é uma característica que provê uma vantagem interessante, né? De passar desapercebido por predadores, né? Então, vários animais que vocês vão ver são transparentes, né? Principalmente os animais de áreas mais superficiais, dos oceanos, né, onde a luz penetra. E os animais que são de áreas mais profundas têm uma tendência a ter essa coloração mais avermelhada e que também dá uma camuflagem, né, nesse ambiente escuro dos oceanos. Aqui vamos tentar observar de perto primeiro esse animal, esse anfípode, para decidir se ele vai ser coletado ou não.
Complementando isso que o Renato falou, né? Então a luz solar ela penetra pela água dos oceanos de maneira diferente. As luzes, os comprimentos de ondas extremos, né? O ultravioleta e o roxo, eles são absorvidos logo nas primeiras camadas, assim como o vermelho, o amarelo e o verde e o azul conseguem penetrar até regiões mais fundas. Para quem mergulha, né, isso é muito comum, a gente sai de águas mais rasas, ali, a gente está fazendo um snorkel, uma coisa assim a 1 metro, 2 metros, e a gente consegue ver as cores dos organismos bem vivas. E aí quando a gente vai mergulhar a 10 metros, 15 metros de profundidade, a gente percebe que os organismos já estão todos mais parecendo marrom, assim, mesmo bichos que são muito amarelos, muito coloridos como alguns peixes e corais, eles já 10, 15 metros já ficam todos marrons, essas cores já não estão mais presentes. E em águas oceânicas, elas, mesmo sendo muito claras, né, essas águas são absorvidas, de forma que só o azul consegue penetrar além dos 50 metros de profundidade. Então, o vermelho é uma cor que não existe lá. Esses organismos que são vermelhos pra gente, a gente está conseguindo ver eles vermelhos porque eles estão sendo iluminados através dos holofotes aqui do ROV.
He's in? No, he's in. Oh, he's right on the outside. Sorry, my friend.
HD4.
So close. Yeah, he's out.
Mas, como o vermelho não chega nessas profundidades, o que a gente vê agora com o vermelho fica completamente preto lá embaixo, né? Então ele se camufla com a escuridão das profundezas. E aí vocês estão ouvindo esses gritos aqui, assim, porque todo mundo muito empolgado aqui de tentar pegar esse organismo aqui. Então é difícil, né, por causa das correntezas, então ele fica se movimentando na água, né, e a equipe de pilotos aqui do submarino não tripulado tem que fazer...
Marco, you were awesome. Stay with it, my friend.
Né, tem que ficar tentando encaixar o bicho nesse funil, que é um funil de sucção. Mas é mais ou menos como colocar uma linha numa agulha ali, então é bem difícil. Isso de maneira remota, né? E essa sucção ela não pode ser muito forte também, senão ela acaba destruindo o organismo. Então, se a sucção for muito forte, acaba destruindo. Tem que ser uma sucção bem lenta, bem delicada, para poder preservar o organismo sem causar nenhum dano para ele.
Esse é um dos animais de interesse particular aqui dos pesquisadores que estão a bordo, né? O professor Ian Hemi, da Universidade de Western Australia, está estudando a visão desses animais. É um tipo de crustáceo que tem olhos grandes, enormes ali na frente do corpo, e é um animal que muitas dessas espécies são especialistas em se alimentar de águas vivas, né? De animais gelatinosos, na verdade. Então, como que um organismo no escuro, né, nesse ambiente escuro, consegue localizar uma água viva que é transparente? É algo que é bem surpreendente, e talvez estudando, né, a visão desses organismos, né, o professor está fazendo experimentos com animais vivos, né, experimentos fisiológicos para entender as respostas do sistema nervoso desses organismos a estímulos visuais. Então é possível que a gente possa estar entendendo melhor, um pouco melhor, opa, entrou, parece, um pouco sobre a visão desses animais e a visão, né, de como, opa, entrou aqui, e, opa, e conseguimos coletar. Então esse aí vai para os experimentos do professor Ian Hemi.
You might not. He's still up there. He's still there. Yeah, absolutely. Nicely done, pilots. Nicely done.
Good.
Yes, I think we can change the canister.
Yep.
Yep. We're going to 7. We have 2 canisters left and 1 peak up there. Right? Yeah. The one with you.
Agora eu vou chamar a professora Silvina Bota para nos juntarmos e fazer a tradução para o espanhol, né, aos colegas da Argentina que estão nos ouvindo, aqui nos assistindo.
Yes, I am determined not to go to sleep until we hit 1,200 years. But Dave is going to come and replace you.
You want to get us down there?
No, I think it's going to be midnight.
It's not going to be much down below 1,000.
But we still have to get down to that.
We're going to get down to 1,200. 1,200 is goal. Dave and Russ, just so you guys know, there's Niskin bottles to do. I wrote them down at 1,200. We're going to drop 1 bottle, so you have to find the pilots. I told them to. 800 or 500. Yep. 50 meters, 2 bottles, 0, or as close to surface as they can get, 2 bottles. So 12, you're going to be around for maybe 1,200, 800, and then Russ, yes, 500. All right. I'm handing it over to Dave Fields.
Good job. Good to go. Way
Good
to go,
team.
sir.
Well, we're not there yet. We're getting there. We're getting there. Thanks, thanks. Everybody.
Buenas noches a todos. Bueno, ahora vamos a pasar un poquito para español. Justo el profesor Renato y Marcelo salieron y entramos Silvina y yo al estudio para hacer algunas traducciones e ir contándoles lo que estamos haciendo en este momento con el ROV.
¿Dónde está?
Yeah, we need a learn vision still.
No está. Buenas noches.
The High Bird is very happy.
Mi nombre es Silvina Bota, soy profesora en la Universidad Federal de Río Grande. Soy argentina igual que Julieta, pero las dos trabajamos en Brasil. Y estamos acá ayudando con la transmisión en español para las personas que están viendo esta transmisión en vivo por YouTube. Ahora seguimos bajando. Vamos a bajar hasta los 1.200 metros. Por ahora andamos por los 850 metros de profundidad.
We'll get there eventually. Yeah, stop is the best word, Marco has decided.
Not too often.
I didn't say that. One. We went in, I think it was 1 or 1:15. So it was 12 hours. 1:30 at the latest. Should be 10. Yeah.
Starry night. Is that a verb?
Hola Julián. Julieta ahora te va a contar qué es lo que estuvimos colectando hasta ahora.
But it's single. Oh no, it's not. It's a livid. My bad. Good. I didn't yell stop.
Stop saying
Bueno, las últimas cosas que fueron colectadas, que son de interés de los investigadores que están acá a bordo del Falkor 2, fueron un poliqueto de la familia Tomopterigi. Los poliquetos vendrían a ser parientes de las lombrices, nada más que con muchas patitas. Y también colectamos un anfípodo, que es pariente de los crustáceos o de los camarones dentro del mismo gran grupo, y es un animal carnívoro que se alimenta de algunas medusas. Una vez que lleguen acá a bordo, cada uno junta sus muestras. Oh, un agua viva pasó, supongo que vamos a ir a seguirla o no. Creo que más temprano también juntamos algunos tenóforos. Sí, actualmente estamos en aguas internacionales que están fuera de la zona económica exclusiva de Brasil. Oh, ¿qué está pasando ahora? Ahora estamos yendo cada vez más profundo. La idea es llegar a los 1200 metros de profundidad, que son aguas muy profundas. Como pueden ver, si bien estamos a la altura de Brasil, la temperatura a estas profundidades alcanza casi los 4 o 5 grados en comparación con el agua superficial, que está a unos 28, 29 grados. Y eso está influenciado básicamente por las masas de agua que se van formando en otras partes del océano, que está todo interconectado, tanto en Antártida como en el Ártico. Se forman las grandes masas de agua submarinas que recorren el resto de los océanos y van cambiando sus propiedades físicas y químicas y de temperatura.
Bueno, acá estaban preguntando también por qué no estaban colectando peces o camarones, crustáceos, animales que por ahí se mueven más. Justamente los peces son más rápidos, entonces son difíciles de colectar, y la bomba de succión tampoco no es tan fuerte como para poder succionarlo. Y también porque el propio motor del robot y las luces que tiene los ahuyenta. Entonces no solamente no los colectan, sino tampoco aparecen en pantalla grandes peces ni otros organismos que tienen una capacidad de natación obviamente importante. Además, podemos recordar que este grupo de investigadores se dedica específicamente al zooplancton, a los invertebrados. Muchos de ellos trabajan con animales gelatinosos, como los xenóforos, las medusas. Entonces, además de Marcelo Melo, que es el profesor de la Universidad de San Paulo, que trabaja con peces de profundidad, el resto de los investigadores todos trabajan con invertebrados. No es el foco de este crucero.
Like a sawtooth kind of a feel to them. See how they're going, they're pointy on the end. This. And the nectaphores are also very chunky. They're not like flat, pointy or one shape. They're much more chunky.
E das operações, a gente chama ele aí pra...
Oh yeah, that was a money. That's a money shot. There we go. Yeah, that's really good.
Creo que más temprano habían preguntado cómo funcionaba el ROV y los propulsores, entonces podemos invitar a Leo Alcántara, que es piloto del ROV, acá en el barco para que nos cuente un poco sobre cómo funciona, cómo es la colecta. Él es brasilero, así que los que sean brasileros van a poder entender perfecto, y si él habla despacito, los argentinos que están del otro lado o los que hablan español también van a poder entender. Si no, cualquier cosa podemos traducir.
Tem um momento de glória. Eu vou responder o que você perguntar.
Não, manda um oi só, né? Se apresenta.
Alô, boa noite, pessoal. Eu sou o Leonardo Alcântara, eu estou aqui a bordo do Falkor com a galera aqui, brasileiros, argentinos, representando a América do Sul. E vocês quiserem saber curiosidades ou interesse na carreira de piloto de ROV, fiquem à vontade aí no chat para mandar. O que quiserem saber, eu estou aqui disponível para responder. Nesse momento aí, a operação de coleta de amostras é um pouco mais delicada, né? Porque a gente tem correnteza, tem alguns desafios que tornam bem desafiador. Então, fiquem à vontade de perguntar o que quiser.
Eu tenho pergunta.
Então pergunta.
O que tu fez, o que tu estudou para chegar a estar pilotando o hobby?
Então, eu sou formado em eletrônica. Geralmente eles pedem formação em eletrônica, pode ser técnica ou engenharia, ou mecânica, mecatrônica. Então, no Brasil a gente tem uma indústria de óleo e gás que usa bastante ROV. Então, tem bastante gente lá, principalmente do estado do Rio, que acaba seguindo a carreira da Petrobras, é muito grande, né? É um grande empregador lá. Então, acaba outras empresas de suporte demandando muita gente. E foi assim que eu comecei. Eu tinha 19 anos só quando eu comecei minha carreira. A vida toda trabalhando embarcado. Mas, assim, tem seus prós e contras, né? Aqui a gente tem oportunidade de viajar o mundo todo nessa carreira. A folga é boa também, que você fica um mês e meio, 2 meses de folga, dependendo da sua escala. Aqui a gente faz geralmente 5, 6 semanas de trabalho, 5, 6 semanas de folga. Então tem esse tipo de vida que é um pouco diferente, mas é interessante. Paramos para a foto aqui.
Opa, interessante. Ficou meio esquisito.
It's got a parasite. It's got a
meio esquisito. Tava toda torta, né?
Really go, With the schema sitting on it. Oh, cool.
Efficient.
They're lovely.
Silvina.
But they have killer antenna. And they bioluminesce. What more could you ask for?
You don't want this for us?
I wouldn't mind. Mas não sei, estavam aqui. Eu acho que era a bolinha.
Tipo de parece.
You can barely tell something apart. Under a microscope.
That's
E os ROV que usam para a Petrobras do Brasil são iguais a esse aqui? Não, porque esse aqui está modificado.
Sim, esse aqui é especialmente feito para ciência, né? Então, assim, o conceito básico é o mesmo, mas aí você começa a mudar a questão de equipamentos que usa, força, né, potência do ROV, porque esse aqui é um pouco mais delicado, então você não precisa de muita potência. Enquanto no óleo e gás você move muito equipamento pesado, então eles são bem, tipo, comparar um trator com um carro, uma SUV. Então, dependendo do trabalho, você precisa de mais força. Mas é o mesmo princípio, tem os braços, os propulsores, tudo bastante similar. Aqui que tem essa câmera 4K, que, por exemplo, do óleo e gás a gente não precisa disso.
Não.
É uma câmera bem porreca. Isso é uma das coisas mais legais que tem aqui, que eu acho. Essa câmera, você vê lá, é incrível, velho.
Incrível. As imagens são incríveis.
São. Isso aí é uma das coisas que eu acho mais interessante.
E quando vocês estão trabalhando no offshore também aparece muito bicho legal? Vocês também se divertem com isso assim? Conseguem...
Parece bastante. Só que a gente não consegue ver o detalhe, né?
Como aqui, como a câmera.
Eu já vi uns bichos. Eu não entendo nada de biologia. Eu já vi uns seres bem diferentes. Eu já vi tubarão, baleia.
Baleia?
Uhum. Marlin.
Esse marlin vai nas profundidades.
Todo quanto é tipo de animal já vi.
E engraçado que você falou do trabalho na área de óleo e gás, mas eu também já usei em aula imagem que foi feita por pessoas que estavam trabalhando em ROVs, né, da indústria de óleo e gás, justamente porque eventualmente se encontra um animal bem interessante, né?
Sim,
então é bem comum. E vai pro WhatsApp a imagem e aí chega na gente.
Entre a gente circula a galera do ROV.
Sim.
Quer falar?
É siphonophore ainda, não é?
Volviendo para el objetivo del ROV, le agradecemos a Leo que estaba acá contándonos. Si tienen más preguntas, obviamente pueden dejarlas en el chat y él las puede ir respondiendo. Lo que tenemos ahora en pantalla que estamos buscando es un tenóforo o sifónoforo. Un sifónoforo, no había escuchado lo que estaban diciendo. Está me diciendo acá Ciel que parece que se ve un anfípodo dentro del sifónoforo que podría haber estado alimentándose. O ser un parásito. Lo tiene, tiene un parásito agarrado un anfípodo parásito agarrado del lado de afuera del sifónoforo. Están intentando tener una imagen mejor.
Sería esa bolita que está ahí abajo, ¿no?
Sí.
El flotador,
tiene
la parte de abajo tiene una bolita blanca.
La parte más transparente es el flotador del sifónoforo y la bolita blanca más oscura que a veces aparece, no sé si es lo que estaban viendo en pantalla, esa bolita es el anfípodo parásito. Como, bueno, solo para recordar, porque obviamente no todos los días están las mismas personas. En el inferior izquierdo de la pantalla tienen las informaciones de dónde estamos actualmente, por ejemplo la profundidad, que es a 890 metros de profundidad, la temperatura del agua en este momento, la salinidad y las concentraciones de oxígeno. Como estaba diciendo antes, la temperatura en estas profundidades no es la misma que en superficie por las diferentes masas de agua que se forman a lo largo de la columna de agua. Estas masas de agua provienen desde otros, o se forman en otros océanos y viajan en profundidad por diferentes alturas dentro de la columna de agua y van circulando a lo largo de todo el océano. Esto es lo que se conoce también en oceanografía como conveyor belt o cinturón termoalino. Cada una de estas masas de agua tiene propiedades físico-químicas que se mantienen a lo largo de toda la trayectoria. Entonces, a lo largo de una columna de agua, en un punto determinado, vamos a tener el agua superficial, como pasa acá en Brasil, que tiene una temperatura de más o menos de unos 29 grados. Y a medida que vamos descendiendo, o sea, a medida que nos vamos más en profundidad, vamos a ir pasando por distintos grupos de masas de agua con diferentes características tanto de salinidad como de temperatura. Llega un punto que no se puede tomar más ese té. Pegou? Bom, parece que capturaron el sifónóforo. Estamos esperando a ver si entró en el colector número 8.
All right, so we are a little early on needing to restart the streams, but we need to restart the computer. So I'm going to restart stream now, and it will take a bit longer. It should kick you over into part 2.
Sí.
But you're just going to have to sit tight for a few minutes while we reboot our systems here, and we'll see you over in part 2.
Dos acompañantes, dos peces. Oh, entró un peixinho. Dos, no.
Ah, es anfípode.
So do we have one bucket left then? This is
No, we still have 2 left. 6 and 7.
Okay, but we can't close the bucket after the last one.
The last bucket never gets closed. So you're very likely to
Uno era achei que era um anfípode, eu estava pegando nele, porque era igual e tal, e depois não estava mais. Passou, porque não era peixe, aquele outro primeiro. Do jeito de nadar, parecia.
Mas aí tu tava trabalhando fora, quando você tava trabalhando fora quando você ficou sabendo aqui emprego, porque teu inglês é bem já fluente, assim, já, né?
Desde 2007 que eu trabalho. Mercado fora.
Eu sei.
Comecei no Brasil, 2001.
Um tilt no cerebro. Eu tava pensando em ave, eu falei que Marcelo estudava aves.
Fiquei lá até 2014.
Ave de profundidade.
Uma empresa de UK.